thank you and welcome to the second hearing for the inquiry into the use of primates and other animals for medical research in New South Wales. My name is Greg Donnelly and I'm the chair of the committee. I'd like to acknowledge the Gadigal people of the Eora Nation, traditional custodians of the land on which we are meeting today. I pay my respects to elders past, present and emerging of the Indigenous members in our community. I also acknowledge and pay my respects to any Aboriginal or Torres Strait Island people who may be joining us today, particularly over the internet. Today we're hearing from a number of stakeholders, concluding with evidence from the New South Wales Government. I thank everyone for making the time to give evidence to this important inquiry. While we have many witnesses with us in person, some will be appearing via video conference today. I would ask for everyone's patience through any technical difficulties we may encounter over the course of the day. If participants, for one reason or another, lose their internet connection and are disconnected from the hearing, they're asked to rejoin the hearing by using the same link as provided by the Committee Secretary. Before I commence, I'd like to make some brief comments about the procedures for today's hearing. Today's hearing is being broadcast live via the Parliament's website. A transcript of today's hearing will be placed on the Committee's website when it becomes available. In accordance with the broadcasting guidelines, media representatives are reminded that they must take responsibility for what they publish about the Committee's proceedings. While parliamentary privilege to witnesses giving evidence today, it does not apply to what witnesses say outside of their evidence at the hearing. I therefore urge witnesses to be careful about comments you may make to the media or to others after you've completed your evidence today. Committee hearings are not a forum for people to make adverse reflections about others under the protection of parliamentary privilege. In that regard, it is important that witnesses focus on the issues raised by the inquiry's terms of reference and avoid naming individuals unnecessarily. All witnesses have a right to procedural fairness according to the Procedural Fairness resolution adopted by the House in 2018. If witnesses are unable to answer a question today and want more time to respond, they can take a question on notice and written answers to questions taken on notice are to be provided within 21 days. If witnesses wish to hand documents, they should do so through the Committee Secretariat. Finally, for those participating in today's hearing by video conference, can I please ask everyone to state their name when they begin to speak directly into the microphone and to mute their microphones when they're not speaking. In terms of audiability for today's hearing, I remind both committee members and witnesses to speak into the microphone. And finally, could everyone please turn their mobile phones to silent for the duration of the hearing. So we welcome our first witnesses. Thank you very much for joining us today. We know that you're all very busy and we appreciate you carving out the time to be available today. So what we'll do, we'll go through the formalities of having you sworn or affirmed to your evidence and then we'll move on to the opening statements and questioning. So we have joining us today, and perhaps if you just move from my left to right, it just might be the most convenient. So we have Dr. Sarah Toole joining us by video conference. Welcome, Doctor. We have Professor Kevin Dunn, and we have Professor Wayne Hawthorne. So I'll get each of you, starting with Dr. Toole, to state your name, your position title, if you're representing an organisation today, and swear either an oath or an affirmation, whichever you prefer, the words of which should have been provided. If for some reason you don't have them, just put your hand up and we'll provide them to you. So starting with Dr. Toole. Doctor.
My name is Sarah Toole. I am the Animal Welfare Officer at the University of Wollongong. I solemnly, sincerely and truly declare and affirm that the evidence now about to be given by me shall be the truth, the whole truth and nothing but the truth.
Thank you very much. We'll move to Professor Dunn.
I am Professor Kevin Dunn. I'm the Pro-Vice Chancellor of Research at Western Sydney University, and I swear that the evidence now about to be given by me will be the truth, the whole truth, and nothing but the truth. So help me God.
Thank you very much. And Professor Hawthorne.
My name is Professor Wayne Hawthorne. I'm the Chair of the Animal Ethics Committee at Western Sydney Local Health District. I solemnly, sincerely and truly declare and affirm that evidence now about to be given by me shall be the truth, the whole truth, and nothing but the truth.
Thank you very much. Well, what we'll do, I'll ask each of you to, if you wish to do so, make an opening statement. With respect to submissions to the inquiry, can I just note for the record that with respect to the Western Sydney Local Health District, your submission's been received. Submission 233 has been processed and stands as a submission to the inquiry and has been uploaded to the inquiry's webpage. With respect to Western Sydney University, specifically Professor Dunn, submission stands as submission 212 to the inquiry. Has been processed and stands as a submission to the inquiry and been uploaded to the web page for the inquiry. And Dr. Toole, we don't have a submission, but we very much welcome you today to provide some insights into the work being done at the University of Wollongong. So with that brief introduction, perhaps if we just start, I might start with opening statements with Professor Hawthorne, if you'd like. So, Professor
Sure.
Hawthorne.
My name is Professor Wayne Hawthorne. I'm Professor of Transplantation at University of Sydney. I'm the Director of the National Pancreas and Islet Transplant Laboratories at Westmead Hospital as well. I run the Xenotransplant and Surgical Research Unit at the Westmead Research Institute. I'm currently elected the World President of the International Xenotransplant Association. I'm also the President of the Australasian College of Biomedical Sciences. I'm here to provide support for animal research, and my views and opinions are my own from some 30 years of experience in clinical and experimental research work. As an active researcher, I've seen our research translated from basic laboratory research to become federally funded clinical transplant programs. We've successfully transplanted now more than 600 patients with kidney pancreas transplants. We've transplanted patients with type 1 diabetes, more than 300 islet cell isolations, and more recently transplanted 13 patients with severe hypoglycemic unawareness and severe pancreatitis. Our Animal Ethics Committee oversees and provides counsel on all animal ethics matters relating to research utilising non-human primates and other animals across the Westmead campus. We oversight and promote the ethical, humane and responsible care and use of animals for scientific purposes on all active animal research projects for the many research staff on our campus, as legislated by the Animal Research Act and the Animal Research Regulations. We work to the Australian Code for Care of Use of the Animals for Scientific Purposes. As part of my role, what we offer at the Westmead campus for all students and researchers that attend the campus, we provide a workshop as an introduction to research. Fundamental part of this is their understanding that medical research in the future will undertake research to investigate ways for them to diagnose, evaluate, elucidate, treat, cure or prevent various diseases or conditions. We do not do animal testing. We do not undertake LD50 tests, nor DRAIS tests, nor do we test pharmaceuticals, nor cosmetics, nor has our Animal Ethics Committee ever had any researcher approach us to do so. We undertake our research to treat our sick and dying patients, to improve their quality of life, to improve their overall health, and we look at them very holistically. And we do this and question the best ways to improve their health care, to save them from dying or undergoing problems with their health, and we want to improve their lives for them, their families and the local community. Thank you.
Thank you, Professor. Appreciate the opening statement. Professor Dunn.
Thanks, Greg. My name is Kevin Dunn. I'm Professor of Geography and Urban Studies, but substantively Pro-Vice Chancellor of Research at Western Sydney University. Thanks for the invitation to participate in this important hearing. I do so on behalf of Western Sydney University. As we stated in our submission, Western Sydney University welcomes the opportunity to assist the committee here today, and in summary, I'd like to make a few key points. Obviously, everyone's had access to our submission, but the five I'd stress are, first, that Western asserts the importance of research involving animals in advancing medical knowledge and innovation, and the current Western Sydney University approved medical research projects have significantly improved human health. Our second point would be that the current competitive research and government grant funding processes do ensure scientific rigour through independent peer review, and we feel that funding body processes, though, could perhaps lift their attention to ethical considerations, so that when researchers are seeking funding, that the visibility and importance of animal ethics perhaps could be raised, as well as the costs associated with both the work involving animals and maintaining their welfare. So that's our comment in regard to funding. We also would submit, and you would see it in our submission, that we regret the notable lack of funding in New South Wales and Australia to support innovation in the 3Rs. We are of the view that, in contrast to other countries, where they have nationally established bodies such as the National Centre for Replacement, Refinement and Reduction in the United Kingdom, they provide research and they provide resources for research, for innovation, to encourage researchers to adopt more proactive and innovative approaches to directly address the 3Rs, and we would recommend the same in Australia. Fourth point, the integrity of the research efforts in this area is governed by legislation, including the Animal Research Act, the Australian Code for the Care and Use of Animals for Scientific Purposes, which we believe are working very well, and we have, I think, world's best practice in animal research as a consequence. Respect for animals underpins all of the decisions and actions involved in the use of animals for research, as I perceive it. In this state, the Code and the Act provide a robust regulatory framework to oversee and authorise animal research in Australia, would be our position. And fifth point is that internationally, the regulation of medical research involving animals is an ever-evolving space, and we should be dynamic and innovative, and we recognise that there is a current debate around sentience, and we support the continued intellectual debate around that, and we think that that is part of an important step forward again in the welfare of animals, but we are of the view that some level of consensus should be reached on the definition of sentience before it can be effectively operationalised in legislation and codes, etc. But we look forward to that occurring, and I look forward to further discussion today. Thank you.
Thank you very much, Professor. Dr. Toole, can I invite you to make an opening statement?
Thank you. My name is Sarah Toole. I am the Animal Welfare Officer at the University of Wollongong. I am a registered veterinary surgeon in New South Wales. I also hold a postgraduate certificate in laboratory animal medicine from the University of Guelph, Canada. UOW has one biomedical animal facility with housing for rats and mice, which supports researchers at the university and the Illawarra Health and Medical Research Institute. UOW animals are sourced primarily from ADR and ARC. Any research-specific genetically modified lines are generally held and bred at ADR. As such, we have limited surplus animals, many of which have been rehomed through the Liberty Foundation. We established a small grant system in 2018 to support 3Rs research. The small amount of funding that is available is insufficient to support any major initiatives. Further funding for refinement, reduction and replacement models is urgently required in Australia. I am the only veterinarian employed at the university and have been here on a part-time basis since October 2015. I am responsible for the program of veterinary care. I have oversight of all training, and I conduct formal competency assessments prior to researchers commencing work with experimental animals. I closely monitor the welfare of the animals, as well as compliance with AEC approvals and the code. I work closely with the researchers and the technical staff to create descriptive monitoring and intervention sheets for projects, which are regularly reviewed and refined. I attend the AEC meetings and advise the AEC on the welfare impact of proposals and any areas that I feel could be modified to improve animal welfare and standards. The chair of our ADC is external to UOW and is a veterinary pathologist and consultant in laboratory animal care and management. In addition, our 2 Category A veterinary members, Category C welfare and Category D lay members are external. Our 3 Category B scientific members are all UOW research staff. UOW has been audited twice since I commenced in 2015. The 2017 ARP audit resulted in a number of commendations and some recommendations. In 2021, ARP was unable to conduct our audit due to resourcing shortages, but approved an arrangement whereby we audited another institution and members of that institution audited UOW. Whilst we believe both institutions conducted the audits with the required rigour of an independent audit, we feel that additional funding and resources should be directed towards the regulator so that they can conduct this important process. UOW is embracing openness and has provided details about animal use, including numbers and types of animals and categories of use on our public-facing website, and will be expanding on the information we provide as time and resources permit. We would like to thank the committee for the opportunity to appear at this inquiry and welcome any further questions you may have.
Thank you very much, Dr. Toole. We'll commence the questioning. Now, represented today are members of the government, opposition and crossbench, and we'll share the questions around between the 3 groupings. So we'll commence with the crossbench, the Deputy Chair, Honourable Emma Hurst.
Thank you. I'd like to start with some questions to Dr. Toole. The University of Wollongong stated in a survey that it had determined 2 years ago that forced swim tests were not ethically justified and would only be approving if they were being used to develop alternatives. Can you explain why the university reached that decision and what the animal welfare concerns are with forced swim testing?
So forced swim test is a test where an animal is basically placed in a large barrel of water, and the water height is such that the animal can't keep its nose above the level of the water. It has to actually actively swim. And so the test, it's used by some researchers as a model of anxiety or in some cases depression, and it's used in both rats and mice. So in the time that I have been at UOW, it has been used in a number of rat studies, and we have had some adverse events with that particular test. So we've had quite a few rats that were large male rats that had been housed in conventional laboratory housing for a period of a number of months. And when they were in the forced swim test, we had some drownings occur. Now, the test is conducted with researchers watching the rats, and the rats should actually be actively swimming. And what they say is that when the rat decides just to float or gives up trying to make escape attempts, that is a helplessness or a way of assessing depression or helplessness, anxiety. And so because we had these incidents where, so the rats didn't die straight away. Basically it was aspiration of water that wasn't detected, and the rats subsequently died after the test, and it was confirmed by post-mortem examination and histopathology on the lungs. So those issues were brought to the attention of the committee, and the committee reviewed some videos of that test and subsequently decided that they would only allow that test if it was being used in parallel with an arm to look at alternatives to that test. So since the committee made that decision, the test has been conducted once this year, and that was a study that was designed particularly to look for alternatives.
Thank you, Dr. Toole. Professor Hawthorne, in the same survey, your ethics committee had said that there isn't any applications for a forced swim test, but that such procedures would be unlikely to be approved by the AEC. Can you explain why that particular view was formed on this particular experiment?
We've never had an application. and that's based around the fact that we do different types of research. Ours are very much clinically directed. We don't have many mental health studies, so from that aspect we don't necessitate utilising those sorts of tests. The reason we would avoid that is simply because we don't have appropriate facilities to undertake those sorts of tests. So without the appropriate facilities, appropriate training and appropriate oversight, then we wouldn't want to undertake those, obviously.
Thank you. I'll go back to Dr. Toole. Another experiment that's drawn a lot of attention during this inquiry is the nose-only smoking experiments. And again, looking at this ARP survey, it says that the University of Wollongong Committee concluded it would not approve the use of nose-only smoke inhalation procedures because it would not meet the code's requirements for refinement and ethical justification. Can you explain some of the welfare concerns that the university has with these smoking tower experiments?
I actually have no experience with any sort of smoke inhalation tests. I've never seen one performed, and we haven't had them happening at the university in the 7 years that I've been there. So I really don't think I can comment on that. I know that the chair of our committee has worked in institutions with that test, and those comments came. The committee did discuss it, but most of the members of our committee have no experience with that test.
That's fine. Thank you, Dr. Toole. We've heard a lot about facilities with non-disclosure agreements. You mentioned in your opening statement that you do run a rehoming program, which is good to hear. There was an article printed on the weekend in the Sunday Telegraph that talked a little bit about these non-disclosure agreements that often come with rehoming of animals, and that they can actually serve as a way to silence rehoming groups, and that sometimes some of these rehoming groups felt as though it was almost like a threat that animals would be killed or not rehomed if information was disclosed. Are you aware of this, and do you think it should be a concern? And should there be certain species of animals like cats and dogs where we have, a mandatory rehoming regulation put through to make sure it's done properly?
So I'll start with, I guess, talking about the University of Wollongong's position with rehoming. So we only really have rats and mice as our biomedical animals, and we do have an arrangement with the Liberty Foundation to rehome any suitable animals through that organisation. I was involved initially when the legal agreement was drawn up, and our agreement does state that the Liberty Foundation is not to disclose that any particular animals have come from our institution. My personal opinion, and I'll make it clear this is my personal opinion, I don't see any issues with people knowing that the animals have come from our institution. I also personally would say that animals that have animals like cats and dogs that have been used in research should be made available for rehoming, but those animals would need to be assessed as suitable for rehoming given their behaviour and what procedures that had been performed on them.
Yeah, thank you. That's really useful. One issue that's been raised in this inquiry is honour students using animals for research. Do you feel that there are concerns about honour students using animals for research, particularly given some of the concerns raised is that a lot of these projects are reruns of projects with a known outcome?
The situation we have at UAW is that we do have a number of honours students doing projects in our biomedical facility. Because honours is a very short time frame, most of those honours students are actually brought in to an existing research program. So it would be very unusual at UAW for an application in biomedical research to be submitted purely for an honours student, simply because of the time that it takes to go through the ethical review process and get approval and then get training underway. All our researchers have to be trained before they can order animals in. And and then to actually complete that within a year. So the situation we have is that honours students will come in and be added to a project, and they may be responsible for a small arm of that project and looking after them. But we also at UOW have some guidelines surrounding what sort of procedures honours students can be trained in because of the limited time that they're in the facility. And so mostly our honours students are only trained in general handling of the animals and health and welfare checks. It's very rare for us to actually train an honours student in anything invasive such as restraining animals. Well, so they will be trained in certain restraint procedures for other people to give injections, but it's quite rare for us to actually train an honours student for injections or anaesthesia. We do that sort of training for PhD students, but generally not for honours students.
Do you think that should be across the board?
No. I'm going to stick to the times.
Thank you, and good morning to all of you. I guess just picking up on that point then, in terms of, I guess, students undertaking animal research from a sort of training perspective as opposed to being something that is part and parcel of an existing research project. Dr. Toole, do you think that there should be restrictions imposed on that sort of pure use of animals for a training purpose?
So I'll make it clear that I'm voicing my personal opinion, not the university's position. My opinion is that honours students should not be trained in invasive procedures. I don't believe that there is justification to subject animals to training for such a short period of potential use time, and that the cost to the training animals is not outweighed by the benefit.
Thank you.
Well, it's outweighed by the benefit, I mean. So there's too much cost for the animals for a short period.
Thank you, that's very clear. Could I ask, sorry, yes, Professor Hawthorne?
I could add my own personal opinion to that as well. Just as a blanket rule, honours students, as Sarah was just saying, they're there for such a short duration, they're never assigned to undertake active research work per se without direct guidance, direct support by a supervisor, and they do not undertake any type of invasive procedure. What they generally are assigned to do is take part in an experiment from a peripheral standpoint, as Sarah was saying, to hold an animal. They may be involved with health checks, as in they'll go down and be taught how to handle the animals and then to weigh them on a daily basis. So they're simple things. They are not taking part in doing major types of experimental work. They are not there for long enough, and as a general blanket rule, we do not support that sort of thing. It's different when you've got a PhD student who's with you for 4 years.
Can I clarify, when you say that they're not involved in experiments and research, that also mean they're not involved in these sort of training on invasive procedures as well?
Sorry, I should clarify. They are involved with the research projects. They're assigned and they are signed off as being part of a research project. But they do not do anything that is of an invasive nature per se. And it's more the handling, simply because they're there to be part of an experiment. As a researcher, I would never get an honours student to do anything such as injections or take bloods or anything like that, because we want it done how we've been doing it with our trained staff. They will help analyse the samples, and that's generally what you get them assigned to do, is to take part in the other aspects from the laboratory standpoint.
But to be clear, there's outside of the research project context, is there any separate training that goes on?
So in my
animals?
opening statement, all honours students have to undertake a training program. They all have to undertake the ITAR course before they actually are allowed to even enter the facilities. We also have our own in-house training, and as part of that, it's assigned to a training module, and then they're assigned to their individual senior principal investigator and group to train them how to handle a mouse or a rat or whatever and then to do things like weighing the animals.
Okay,
So
but not invasive.
non-invasive, no. Not
Could-
in our facilities.
Okay, could I ask you the same questions then, Professor Dunn?
Sure, Abigail. I mean, as a principle, in the area of research with animals, research and training go together.
Mm-hmm.
That would be my first principal comment, and that a researcher in this area needs to have training in animal care. Now, some of that training might be at a very basic level, and for students at lower levels. Animal care, husbandry principles, 3Rs principles, all of those sorts of things. That's essential. Deeper levels of training around other types of experiments, invasive procedures, would be attached to where the research was necessary or where the research was involved. You wouldn't do that sort of training, particularly if it affected an animal, just for the sake of training.
Okay. Thank you. Could I ask you, Dr. Toole, I understand that you're a vet as well as an animal welfare officer at Wollongong, but I understand that some institutions don't have a vet. Does that concern you? Do you think that should be a requirement that every institution researching on animals should have a vet?
In my opinion, yes. The code states that there has to be a program of veterinary care and somebody with either veterinary or other appropriate qualifications responsible for veterinary care, but it does not stipulate that that person has to be a veterinarian. And I know that I believe that it was a recommendation to UOW from R in an audit before I commenced that they have a veterinarian on staff. And I believe that all institutions should have a veterinarian on staff.
Thank you. Professor Hawthorne, do you have a veterinarian on your staff?
We have my own personal staff. I've employed more than 2 or 3 vets at any one time to take part in our research work, to oversight. But in terms of our campus, our Director of Animal Care is actually a vet. We also then have a consultant veterinary anaesthetist, or several veterinary anaesthetists that help with the research projects and advise in animal care. And there are a number of other 4 or 5 vets on our campus that are part of the programs. So I think that's integral to research projects. There's also consultancy through universities, etc., to help those facilities that don't have vets on staff, so they can actually have access to vets for research.
Is that the case as well for you, Professor Dunn? Do you have a...
Yes, our animal welfare officer is a trained veterinarian, so was the one before, and so was the one before that. And many of our approved ACEC research projects involve veterinarians as chief investigators.
No more questions?
Thank you so much for your evidence so far. Just wanted to start with a question about, do you think we have a strict regulatory regime here in New South Wales? To all 3 of you.
Yeah, well, I'll go first. I'm sure that Wayne has a bit more to say about this, but we have a good and robust regulatory regime. Strict's a particular word that I wouldn't use. Many researchers may see it that way, but that's tough because it has to be that way. I think we have a high quality regulatory regime and it delivers good outcomes.
And you'd probably, there's probably where the regulations are, and then probably you would go in your various campuses as well, probably a bit above and beyond that.
Absolutely, and in fact, and that's the role of our ethics committee too, to continue to lift the bar as well in this regard. And that's the why, and that, I mean, as you'd be aware, the way animal ethics committees are constructed in terms of its membership is intended to do that as well.
Yeah. Maybe to you, Dr. Toole, about the forced swim test. I'm just trying to get my head around why are those tests used, or what potential medical benefits would you say could come from those tests?
So I'm not an expert in the field, in that field of research, but generally they're used by researchers that are investigating mental health disorders. And so the way it has been used at UAW is that a researcher may be looking at a new or alternative medication that may help with depressive states or anxiety states. And so what they do when they have the test is they would have a control arm of animals, so animals that are not receiving any medication. Then they would have animals that are receiving a medication which is known to be effective, for example, something like Prozac, and then they would have their test medication and they would have a cohort of animals on that. And they may also use different strains of animals, so there are a number of strains of rats and mice, and some of those strains might be more inclined to exhibit depressive or anxiety states. And so each animal would have the same type of treatment and a number of behaviour tests, not just the forced swim test, to assess how their behaviour is changed by that medication that is given. It is a very contentious test, and a number of pharmaceutical companies no longer use it because they do not feel that it actually provides robust information. Does that answer your question?
Yeah, no, it does. I'm just trying to understand, because it's come up a few times, in the first day of the hearings, the forced swim test and also smoke inhalation, and trying to understand, is opinion divided within the profession about the whether there are benefits, or is it the ethical concerns that outweigh the benefits? I'm just trying to understand. I think the committee would appreciate an understanding of that.
I think that there are a lot of ethical concerns about it, but we have had researchers who have been very adamant that if that test was to be banned, that they would no longer be able to do their research. We did have some very robust discussions about it at UAW. We had some research groups that were quite upset about the fact that they would have to provide further justification or include an additional arm of the study to look at alternatives. And so we actually, since UAW came up with that position about having to only approving it if there was an alternative arm to look at alternatives to it, we haven't actually had any researchers apply to perform that test. We had one of our technical officers who received a scholarship from an industry partner and her project was specifically looking at alternatives to that test, and in performing that project she did have to conduct the forced swim test as well as her alternatives, and that has only just been conducted and the data hasn't been analysed as yet.
Okay. And maybe Professor Dunn, Professor Hawthorne, any views you might have about on forced swim test and the smoke inhalation, the ethical concerns compared to the medical research benefits?
Can I just say, from our perspective, we've never done and we've never been approached to, obviously, because we're a different type of health and medical research institution. So I've got no comment on that, but I'll take you back to your previous question with regards to how we legislate and how regulations are. I think they're appropriate for what we do, and with regards to non-human primates in particular, there's extra barriers there and extra assessment. So if you go to apply for a non-human primate research project, you have to go to your own institutional ethics committee, then the ethics committee from where the animals would come from. You also have to report to ARP, and you also have to report to the NHMRC Non-Human Primate Committee. So there's a number of checks and balances there for you to undertake any of those specific types of research. So I think the appropriate level of guidance and assessment is there. Regular assessment by the ARP also oversights our committees. So I think it's appropriate. Obviously, looking towards the 3 Rs, I think obviously we can put in more infrastructure and more support, as Kevin said earlier, but we need that financial support to be able to do that.
Any observations?
I just am not aware, sorry Chris,
No,
of any
okay.
protocols at Western. I will check on notice and get back to you, and and on animal ethics matters, any experiment needs a very credible and considered response, so I'm not going to just...
Yeah, no, that would be good just on those two tests, because it's come up a few times. And
Yep.
I think it'd be good to understand what they're used for and what the benefits are versus the ethical concerns with using them. So that would be great, thank you.
Thank you very much for all appearing today. I just wanted to put things, I guess, into a wider perspective. Professor Hawthorne, can you just provide us some real-world examples of how the research and also the procedures that are undergone in your areas are actually benefiting people and making a difference in their lives?
Sure. That's an easy statement to make. As per my opening statement, I've been involved with a number of programs that started in their infancy, looking in the laboratory, trying to treat patients with type 1 diabetes and renal failure. And the wonderful thing there is obviously we've been able to take them through from the bench side, from laboratory basic cellular testing, through the murine models to porcine models, to then develop the specific ways of doing the diagnosis of, the management of, transplantation, diagnosis of rejection, use of the various drugs, and ways to which to minimise rejection, immunosuppression and so on. As I said, we've transplanted now and got federal funding for our national pancreas and also a second program, a national islet cell transplant program, benefiting almost 1,000 patients directly. And that's affected them dramatically because they've had a kidney transplant and a whole pancreas transplant, changed their life, changed their quality of life for them and their families. The patients that have received islet cell transplantation, specifically for type 1 diabetes and hypoglycemic unawareness, that's where they can't detect a low blood sugar and will collapse and die from their low blood sugar. We've done more than 300 of those islet isolations for transplantation and benefited those patients. They no longer need to take exogenous insulin and are cured from their hypoglycemic unawareness, so essentially saving their lives. The children that have undergone more recently, we've done 13 auto islet cell transplants. This work was all done in our basic laboratories, even the simplest things of how we do the surgery, how we actually then isolate the islets and are able to transplant those patients. Most recently, there was a program on SBS. One of our patients, little Miller, of great benefit, she was dying in acute pain, agonising pain from her severe pancreatitis. We removed her pancreas, her duodenum, and the lower end of her stomach, and then took the pancreas. Through our research, we're able to isolate the islet cells and put them back into her liver, and she is now off of all exogenous insulin. She doesn't require any insulin, and she suffers no more chronic pancreatitis pain. So they do have dramatic effects. Again, this is just my own personal work. We have many hundreds of other of these sorts of things that have come through Westmead, not to mention every other research institution in New South Wales, Australia, and around the world.
Mm. And is it fair to say that animal research played a large part in being able to reach those milestones and achieve the outcomes that you've got?
Absolutely. It was the underpinning reasons that we were successful in being able to do it, and again being nationally funded. Worldwide, the institutions that are very similar to us and have got similar programs, we all utilise the background of our research work. We go to Congress, present our data, and we share. I went overseas to actually train in a number of institutions, and we have other people come to our institutions to train on the same sorts of things. So we share our data, we share our expertise, and give these benefits around the world to as many patients as we possibly can.
Would you have been able to achieve the same outcomes if, say, for argument's sake, animal testing wasn't permitted?
Animal research, if it's not undertaken,
research, correct, yes.
we would categorically, absolutely never have had our transplant programs happen. There's a huge interaction. It's fine to say we can test things on a chip. We can look at organoids, organelles, petri dish cell assays. They're appropriate for basic understanding of a simple single pathway. What we're talking about in our context, certainly for transplantation, is thousands of pathways that you cannot mimic, you cannot change or have them happening in a single entity such as a microchip or an organoid or any sort of cell culture. You've got to think you've got immune system, hematological system, biochemical pathways, endocrine pathways, neural pathways, let alone the surgical aspects or the aftercare. There's so many interactions that happen within an animal that you cannot mimic in cell culture or any other model at this time. Obviously, we'd like to have the ability to do so, to pre-test and so on, and we do a lot of these pre-testing in our various models before we go to mice or larger animals. But categorically, no, we would not have our programs that we have got.
Professor Dunn, is the experience in your centre similar? I'll just ask that simple question.
Yeah, look, we would point to brain cancer treatments that have been delivered. Some of those are mentioned in our submission, but also medical medicinal cannabis use from experiments with or animal research with mice. Yeah, absolute clear benefits.
Apologies, I ran out of time, but thank you for your insights.
Okay, I'll now move to some questioning, and I'll draw on my questions, some content in the submission, and also some comments made in the helpful opening statements. Professor Dunn, in your submission, you on page number 3, just under the heading D, just go down 1, 2, 3 paragraphs.
Yep.
The comments made, I'll just read it. Open quotes. Within the research community in New South Wales, there is growing consensus that the current method used by the New South Wales Department of Primary Industries, open brackets, DPI, close brackets, to collect data on animal usage statistics in research and teaching activities requires revision. I'm just wondering, can you elucidate on that and help us understand this emerging view and the thoughts about how that's best addressed?
So there's a little bit of detail in there, Greg, but thank you for drawing attention to it. The big distinction is between wildlife-based research and research which is occurring within our facilities. To me, they're categorically different in terms of the interaction with the animals involved, and at the moment, treating them the same in terms of the count of animals inflates the extent to which animals are being used in research for medical research or for research generally, particularly where there are very low levels of observation. So that's just a recommendation with regards to the data gathering that we're making.
Okay. If I could just stay with you for the moment, Professor. Equally in your submission, you comment about or reflect on the lack of funding opportunities in, and I'm talking about page 2 here now, under C, a lack of funding opportunities in New South Wales and Australia to support research into the 3 R's. Mm. Perhaps I'll ask all 3 of you, perhaps starting with you.
No, thanks, Greg. Yeah. First, I'd say that as a sector, I think in Australia and New South Wales in particular, we've done very well. It's just that I jealously look at other jurisdictions around the world where there's a coordinated investment to encourage universities to even push this further. We could go further. We could do more in terms of our innovation on the 3 R's if we were provided with the resources to do so. And I think we could coordinate that effort as well in New South Wales, for instance, if the resource was there. And I know that because of the great advances we've already made. And in our submission, we make reference to some of the examples of each of the R's anyway, at Western Sydney University. The UK offers a funding scheme annually. It's a two-part scheme. One of it is for major projects up to £75,000. There's also a smaller scheme in which people can get salary subsidies and focus on innovation in this area. It does expressly prescribe funding for facilities and equipment, because that's appropriate, because that's in the commercialisation realm maybe. But you know, I would encourage in Australia for us to look at something similar.
Okay. I don't know whether Professor Hoyle, you want to...
Sure. I guess I'd echo Kevin's comments. Certainly we struggle as it is to get appropriate funding. Funding levels are around 8% success when you apply for an NH and MRC grant. So you're struggling to get your basal research funding these days. It's highly competitive. So outside of the current schemes, I think, and certainly this is my own opinion, that we should have additional funding, directed state-based funding, to help support the 3 Rs. And we certainly would welcome that to allow us to undertake any sort of research that we could help develop changes to the 3 Rs.
Thank you. Dr. Toole, would you like to comment on that matter of the 3 Rs and enhancing and improving that position in New South Wales?
Well, yes, so UAW has, as I mentioned in my opening statement, a very small grant, and it's in the order of $3,000 to $5,000 that we have been able to make available over the last couple of years. But we receive a number of applications, a small number of applications. Because it is such a small amount of money that we are able to offer, it really isn't sufficient for anybody to really look at replacements. Often the researchers that apply for those grants at UAW are looking at ways to refine their research. So for example, they might be wanting to look at can an analgesic be used in their model, and so they want to run an additional arm of the study using an analgesic. So yes, we definitely need a lot more money available for research and funding into refinements, reductions, and ultimately replacements.
Thank you, Dr. Toole. Professor Hawthorne, if I could just take to your submission, and particularly on page 4, under B, in the second paragraph. I think you helpfully give some perspective here about the matter of progressing to clinical trials and the steps leading to that. I mean, it's all compressed into one paragraph, but just halfway down that paragraph, about fourth line, you say, given the timeframes and costs involved, only those drugs that stand the best possible chance of success can proceed to clinical trial. All of these have been developed and trialled in animal studies at several levels of testing. I'm wondering if you could just elucidate on that a little bit, because there has been some evidence to the inquiry, both in submissions and oral evidence, that seems to suggest that it moves relatively quickly and in sort of, well, I mean, I guess it's my words, but perhaps less than, shall we say, rigorous process before something gets to a clinical trial. In other words, it can move pretty quickly and all sorts of things can be done in a clinical trial, and particularly in the area of I use the term vernacular, the wastage of animals in experimentation. So I'm just wondering, in terms of the rigour around the clinical trial process and getting to that, could you just please, I know there's not much time, but just give us a sense of what's involved?
Well, certainly I'm no expert in this area, but I can make comment from the point of view that certainly our own experience trying to get agents that we use as immunosuppressives. You mentioned time scale. I guess it's all relative. 10 years, 15 years for us is a quick time. Certainly development of vaccines like we've seen for COVID, that happened over a period of only 2 years. Now the difference therein is you've already got decades of research that led to a number of things that underpin the development of those vaccines. Likewise with a number of pharmaceutical agents. Certainly immunosuppressives can be tested in various other models prior to then going to being used in research projects or then getting to a clinical trial. So the clinical trial process is obviously human patients that undergo trials with those agents. Certainly to get there though, it's only the best drugs that have the appropriate use on that specific target will get to those trials.
And perhaps the other witnesses comment about the phrase best possible chance of success. That's very hard to define with any specificity, but is that essentially a process of distillation, of looking at what the possibilities are and the likelihood, and a judgment made over time to go in a particular direction with a particular sort of drug?
Look, I'll make the example from my own research, and that's basically we try and define a number of immunosuppressive agents that have been developed and used for different organs. We may start with models that have been developed and we know clinically that kidney transplantation sort of leads the field. Specific immunosuppressive agents, one called belatacept, has been trialled clinically in kidney transplantation. We felt that that would be a benefit potentially for islet cell or pancreas transplantation. So it's already been tested or trialled clinically in the kidney model, but we then went back and utilised it in our animal research projects, looking at a number of other immunosuppressives with that agent, and we've also shown that it has effect for pancreas and islet cell. And it is now being used in a clinical trial. So things can be streamlined by looking at different aspects. It doesn't mean that we've tested 5 or 10 other agents to get to a specific agent. It's more how we look at the overall modelling. So that's what that broad statement is sort of trying to say.
Okay. Well, time's beaten us. I've certainly got more questions, but they'll need to go on notice. So if you're agreeable,
Absolutely.
myself and others will put questions on notice. So once again, I know it's been relatively short the time, but it's been an opportunity to ask you a range of questions, and we appreciate you making time available. So thank you very much.
Thank you for your time, and thank you for the question.
Thank you. Well, good morning and welcome to you all.
Thank you.
Thank you very much for making yourself available. We know you're all very busy in your professional role. No one's on video, so it'll be easy to sort of work across the group and ask the questions. It's always hard to coordinate a bit when people are participating by video. So what I'll do, if it's agreeable to you, turn to the formal part of the swearing and the affirmation. It's easy to sort of go from my left to the right and do it that way. So get you in that order: name, position, title, and the organisation you're representing. Swear either an oath or an affirmation, whichever you prefer, the words of which should be before you, and if they're not, just put your hand up and we'll provide them to you. So thank you very much, Professor Double.
Kay Double, the University of Sydney. I solemnly, sincerely and truly declare and affirm that the evidence now about to be given by me shall be the truth, the whole truth, and nothing but the truth.
Thank you very much. And Dr. Mesrick?
Good morning. My name is Susan Maastricht. I'm the Director of Research Integrity and Ethics at the University of Sydney. I solemnly, sincerely and truly declare and affirm that the evidence now about to be given by me shall be the truth, the whole truth, and nothing but the truth.
Thank you very much. Dr. Rob?
Ted Rohr, Director of Research Ethics and Compliance Support at the University of New South Wales. I solemnly, sincerely and truly declare and affirm that the evidence now about to be given by me shall be the truth, the whole truth, and nothing but the truth.
Thank you very much. Dr. McCarthy?
My name is Dr. Christopher McCarthy. I'm the Chair of the University of Newcastle's Animal Ethics Committee. I swear that the evidence now about to be given by me shall be the truth, the whole truth, and nothing but the truth, so help me God.
Thank you very much. And Professor Kelly?
Yes, Professor Brian Kelly from the University of Newcastle. I'm Pro-Vice Chancellor of Research at the University. I swear that the evidence now about to be given by me shall be the truth, the whole truth, and nothing but the truth, so help me God.
Thank you very much. Well, just in terms of the formality of recognition, and thank you all very much for your respective submissions. So if I just group them together, the University of Sydney, Submission 217 stands in the name of the organisation. It's been received, processed and uploaded to the web page for the inquiry, so thank you for that. With respect to the University of New South Wales, submission number 221, once again received, and we're grateful for that, processed and stands as a submission and been uploaded to the inquiry's webpage. And with respect to the University of Newcastle, thank you. Your submission is submission 231, and once again processed and stands as submission to the inquiry and been uploaded to the webpage. So perhaps what I'll do first of all is invite each of the respective organisations. We normally have opening statements, and that would be 5 opening statements. We can do that if you wish, but perhaps there's been a person for each organisation or each university delegated. So perhaps if we start with the University of Sydney.
Thank you, Chair.
Thank you.
As I said, my name is Kay Double, and I'm a Professor of Neuroscience. I'm a biomedical researcher, and I'm also the Chair of the Animal Ethics Committee at the University of Sydney. And I'm joined today by Dr. Susan Mastridge, who, as she introduced herself, she is Director of Research Integrity and Ethics Administration at the University, and Professor Beloff, who's the Interim Deputy Vice Chancellor of Research. Extends her apologies as she's unable to attend today.
Thank you.
So the University of Sydney has a strong commitment to animal welfare, and we welcome the opportunity to assist with this inquiry. So we'd like to reinforce several key points in our submission. First, research involving animals continues to play a critical role in improving human health and in treating disease. Secondly, the university supports alternatives to animal research wherever possible. However, further government support is needed for 3Rs research. Third, the current method of reporting animal use requires revision. And finally, we need to retain federal legislation specific to animals in research and teaching. Animal-based research underpins medical breakthroughs that improve human health. In Australia, the Therapeutic Goods Administration requires evidence of safety derived from controlled animal studies for the registration of therapeutic drugs and devices. But on the other hand, much animal-based research is specifically directed to improve animal health and welfare. We're moving towards replacing animals in research, but progress is impeded because of the lack of 3Rs research support from state and federal governments. So we recommend that the New South Wales Government supports the establishment of a national 3Rs centre, charged with support, training and grant provision for 3Rs promotion and research. Active collaboration with the NHMRC and the ARC for a 3Rs centre could be vested in the Australian and New Zealand Council for the Care of Animals and in Research and Teaching, or ASCART, which would see Australia move more rapidly towards embracing 3Rs, including the replacement of animals in research. Inconsistent reporting of research animal activities in different states and territories, and including all animals involved in biomedical, veterinary, wildlife, environmental research and teaching, significantly distorts animal research statistics in Australia. Other countries report only animals directly used in biomedical research. And this could be addressed by a harmonised national reporting system embedded in legislation, as seen in the EU and the UK. We also recommend that the New South Wales Government urge the federal government to improve resourcing of ASCART to deliver, support and administer an openness agreement to facilitate greater transparency, community confidence and the social licence we currently receive for animal research. Thank you, and we're happy to take questions.
Thank you very much. Professor Roll?
Thank you for the invitation to participate in this important hearing. I appear here on behalf of the University of New South Wales in my capacity as Director, Research Ethics and Compliance Support. Our apologies that Professor Edna Hardeman was unable to accompany me today. With this statement, I would like to outline the core points of our submission. We conduct world-class research on 21st century biomedical challenges, and we are affiliated with prestigious medical research institutes, many of whom have also presented to this panel. We receive significant research funding from NHMRC and MRFF, and our research efforts are underpinned in part by animals other than non-human primates. In this context, we have outlined our strong commitment to ensure that this work is reviewed prior to commencement to maximize its benefits for human and animal health and the environment, and that strong safeguards are in place around any risks that would arise as part of this research. We also have strong safeguards in place for animal welfare, from the review of all research involving higher order invertebrates and vertebrate animals prior to commencement, to continuous monitoring by our animal ethics committees and animal welfare team, to world-class training and emergency care. Importantly, UNSW has decided to commit significant funding towards a dedicated research facility using high-level imaging and other technology to advance the 3Rs through the use of organoids to mimic the behavior of human organs, etc. We have so far set aside $170,000 for staffing and $250,000 for equipment, with more to come. Further investment in this exciting opportunity will come in our collaboration with the Prince of Wales Hospital and other partners. Already 50 UNSW and affiliated researchers have expressed interest in using this facility. I would now like to reaffirm our recommendations. 1, consideration should be given as part of this inquiry to steer a discussion at the national level towards the regulation of animals used in research, equivalent to the oversight of gene technology research by the Commonwealth Office of the Gene Technology Regulator. 2, reporting of animal usage should be considered in the context of openness and transparency and be meaningful for the informed public. 3, the regulation of animal use needs to be considered in the international context and environment to really be able to achieve best practice. And 4, investment from the New South Wales Government towards initiatives such as the 3Rs and the repeatability of animal research would be invaluable to retain our top international standing. Thank you.
Thank you very much, Doctor. Professor Kelly, will you be making the opening statement?
Yes, I will. Thank you.
Please proceed.
Thank you for the opportunity to be here today. I'm Professor Brian Kelly, Pro-Vice Chancellor of Research at the University of Newcastle, and as mentioned in the introduction, I'm accompanied today by Dr. Paul McCarthy, who is the Chair of our Animal Care and Ethics Committee. I wish to acknowledge the traditional custodians of the lands on which we're meeting today and pay my respects to their elders past, present and emerging. The University of Newcastle has a long history of health and medical research, and our studies have a clear aim: to help people live better, healthier lives. We understand that involving animals in research is and can be a challenging subject. We also know that animal-based studies are leading to better treatments for conditions such as stroke, mental illness, asthma, COVID-19 infection, chronic pain and a range of cancers. Animal models are carried out in combination with other technologies to help us better understand and treat disease. They do not perfectly mimic what happens in the human body, but neither do molecular methods, cell cultures or clinical trials for that matter. Animals do, however, provide an essential degree of guidance and confidence that enables subsequent clinical testing to proceed more safely. For example, one of our research teams developed a nasal spray to help fight COVID-19. This group was initially investigating ways to counter severe illness caused by cold and flu viruses. When COVID-19 hit, they pivoted. In April, their innovation entered phase 2 clinical trials to further test efficacy and safety in humans. If successful, this treatment could complement COVID-19 vaccines, particularly where a vaccine may be less effective or in particularly vulnerable individuals. Another team pinpointed a cell that's behind health conditions affecting the uterus, including endometriosis and infertility. Researchers collected tissues from a large number of women while also using laboratory models to test their findings. Their work provides a framework to help scientists better understand potential treatment options for uterine disorders. Our researchers are also adapting medical technologies created for humans, such as IVF, and using them to help our native species, including the now endangered koala. We remain committed to the 3 Rs of animal-based research: replace, reduce and refine. We would welcome greater investment in the development of viable alternatives to animal models, but funding to advance this is limited. We would also welcome greater investment in central research infrastructure, as recommended in the 2021 National Research Infrastructure Roadmap. Given our state's strong track record in health and medical research, we encourage the New South Wales Government to also consider opportunities that could enhance the collective capabilities of our research institutions so we can continue helping people live better, healthier lives. Finally, I wish to reiterate the firm commitment of the University of Newcastle to animal welfare and the highest ethical standards in the conduct of our research. We welcome this opportunity to talk with you here today. Thank you.
Thank you very much, Professor. Well, we'll move to questioning now. We've got representatives at the table, excuse me, from opposition, crossbench and government, so we'll move it around and share the questions between ourselves. We'll start with crossbench, Deputy Chair and Mr. Boyd, and then we'll move to government. Honourable Lou Amato, if you can be on notice, about 12 minutes time or so, your turn, so we'll just share it around that way. So, the Deputy Chair, Honourable Hurst.
Thank you, Chair. I want to ask some questions to Dr. McCarthy and Professor Kelly. Newcastle University has come under quite a bit of scrutiny in this inquiry about the approval of nose-only smoke experiments. Are these experiments still approved and currently running at the university?
That's correct.
We've heard evidence at this inquiry. Somebody actually suggested in the last day of inquiry that one of the members of the Newcastle Animal Ethics Committee had objected to the smoking mice research. I'm wondering, first of all, if that is correct, if you're aware of it, and if so, how it was approved, if one of the, or more than one of the Animal Ethics Committee members actually did not want to approve the project.
Sure. So that was before both our time, so I can't speak directly to the events that occurred around that time. I joined the committee after that had occurred. My understanding was that there had been not a consensus on the protocol that had been put forward to the committee.
Okay. One document highlighted a significant number of deaths, 102 deaths between April 2018 to June 2020, with 73 of those deaths directly related to the smoking tower, asphyxiation, essentially, being a primary cause of death. We also heard this morning that one of the universities actually cancelled or just made a decision not to approve any more projects from 2 deaths, but we're hearing that there are over 100 deaths in these projects. I'm wondering why these projects are continuing to be approved and run by the university despite what seem to be quite significant numbers of deaths.
Sure. So I can speak to a little bit about that in that I was a Category A member of the committee with one of those events. I can't speak to all of those, unfortunately. There had been a failure of the machine at that time, which made the numbers obviously larger, rather than it being a series of smaller incidents, which is why the number was large. At that time, the machine was shut down, and investigations were performed, and no processes were allowed to go forward until the Ethics Committee had received information from the engineers that the machine was safe to use. So after that event, there had been a cessation of all animals in that model and until that confirmation had come through. Since that incident, engineering and Ethics Committee involvement put more stopgaps to try and ensure that incident would never be repeated again. And since that time, the cause of that incident has never been repeated.
And how many animals died in that specific adverse event?
I don't have that detail for me immediately, but I can certainly take it on notice for you.
Yes, please, thank you. And so those 102 deaths occurred from various adverse events, is that what you're saying? But the only one that you were sort of involved in is that particular one?
That was a series, is my understanding. The one that I was involved on, I believe there was maybe 20 deaths from the machine. Again, I'd have to take notice on the exact numbers, I'm sorry.
Thank you. Do you have plans to phase out the smoking tower experiments in the future, or does the university have plans to continue to approve these projects going forward?
Look, I think on behalf of my committee, and I think I speak on all researchers, is the day we don't need to use any animal models would be a good day. And
I just want to talk about...
At this moment, we don't have from my understanding from the researchers, and we always ask the researchers this question in every protocol is, please justify to us the need for an animal model. And at this stage, my understanding is the researchers do not have an alternative that would remove the animal model.
Has the university or the Animal Ethics Committee received complaints about the smoking experiments or anyone involved in the smoking experiments? And can you talk, if you have received complaints, can you talk a little bit about what those complaints have been?
I'm not aware of any obvious complaints, but again, I can take notice on that. My understanding is we have acted upon any adverse events that have occurred from the machine. So the Ethics Committee are involved in any death, obviously, in any adverse event. So every adverse event that comes to us from any animal model, it goes through the committee and we discuss why it occurred, how it occurred, and what could be done to prevent its reoccurrence. And so any complaints that may have come, I guess, through researchers about an adverse event, we would certainly have dealt with. The university does have its own complaint process. The Ethics Committee works independently, as I'm sure you're aware of the university, but I believe there would obviously be that process available.
I also want to ask about the nose-only apparatus, because that's something that's come under quite a lot of criticism. Some people who aren't sort of necessarily against the use of animals in experimentation have been specifically against this experiment because of the nose-only apparatus. My understanding is if the animals move, they can suffocate. If they wet themselves, they can die of hypothermia. And some people within the research industry are saying that it's this actual apparatus that's causing an enormous welfare impact and a lot of animals dying, and there are alternatives. I'm wondering why the university is continuing to use the towers, despite the criticisms for that massive welfare impact, when other people in the same space are saying that there are alternatives to these experiments where animals can be smoked, but not in those towers.
Sure. So again, I can't speak for the researchers who have had negative opinions of it. Within my time as both a Category A member and chair, we have made many reinforced recommendations to the researchers, and all of those have been enacted. So as far as cooling in the units, the mice have also now been placed, and for several years now, are warmed both prior and post any of the procedures that are occurring. Any animals that are seen to be turning in the tubes, both those towers are monitored at all times. And so researchers are able to identify those animals and remove them immediately. And there have been no deaths from turning of mice in certainly the last few years that I've been involved in as chair of the committee.
And have there been any adverse events with the warming of the mice after the procedure?
No, in actual fact, warming has been a very good initiative and has had a much faster recovery from animals from the tower.
And are students involved in working on the nose-only smoking models?
I believe students, if they would be involved in those animals, would have strict supervision. As part of our ethics committee requirement, any student involved in a protocol must do what we call Care 1 and 2, which are modules designed to have them be aware of legislation around animal use, but also around the university requirement for welfare impact to be examined. So no researcher would be able to, no student would be able to do a monitoring role unless they've done both those care modules. We also require that a supervisor be allocated to any student on any project across all protocols, and that their competencies must be explained to the committee before they're allowed to do anything on their own.
Thank you. So does that mean that students are involved in these smoking tests outside of research projects?
No, nothing occurs outside a research project.
Okay. What
Sorry, I may have misinterpreted the question. Did you mean by student as in university students doing their degree, or research students under the ARA of a research project?
So I'm talking about university students, undergraduates.
Oh, no. So no students at all would be used in any of those research groups.
And you often hear stories of undergraduates in psychology or other things doing experiments involving mice and rats. And I know that, I did psychology some time ago, but we used mazes and did all sorts of things with mice. Is that something that still occurs? Are there still experiments and testing that goes on with undergraduate students at your university?
Again, I don't have the knowledge based on that being the chair of the ethics committee, so I only have knowledge based on projects that come through to me. Mm-hmm. We do have teaching projects come through the ethics committee, and so we review all of those teaching projects. I believe there are some of the wildlife researchers have their students obviously observational. They come through our committee, and I know that any of the research that any of the teachers who are using animals in their research are required for us to have feedback from those students. So as part of their annual reporting, we get a feedback about the use of animals in that research and whether they believe that whether the students believe that the animal use has been beneficial or not beneficial to that research, and then it's acted upon by the committee.
Right. Okay. Just coming back to the smoking test then. So my understanding of especially when that nose-only apparatus is used, is that you're exposing an animal to, basically they have to sit there for the time that it takes to burn 12 cigarettes, and they're sort of inhaling that, and that is to induce disease in the animal. Is that correct?
Correct. So
Yeah.
the goal of that animal model is to reproduce a disease called chronic obstructive pulmonary disease, which is a disease that about 1.5 million Australians currently suffer.
So the idea is then they get that disease, and then you can test treatments on the animals once they have the disease.
Correct. So I think the term smoking often is very confusing for lay people. This is not a smoking model. This is a model about COPD. The smoking is purely to try and provide a space for that disease to be present.
Understood. I also understand that there's been quite a lot of criticism as to the validity then of a lot of the tests that follow that, and that there are alternatives. Why are we continuing to use those that sort of smoking model in light of available alternatives?
So again, I can't speak on behalf of the researchers, but the first question we ask in any of our projects is, could you please justify the use of animals in this project? And have you looked at alternatives that may replace an animal in the project? So we can only go based on the information provided to us. And I don't think at any stage we have felt that the justification for the use of this animal model has been unnecessary. Some projects that have had use of this model we have questioned and had more justification provided, whether that be from other papers that have used that sort of research and achieved the same goals. But the committee is very much trying to make sure that there's always justification for why animal models are used across all of our projects.
But given the obvious animal welfare impact, the real cruelty of, when you look at, I've seen stories I've read stories of animals who have had this nicotine withdrawal, and just how awful and cruel this particular method is. Do you think that the committee is failing in its duties to weigh up the welfare impact against the potential benefit of the research?
I've been a veterinarian for over 20 years. I've dedicated my life to the care and support of animals, and I probably do take objection to the fact that I think you are doubting the fact that our welfare committee is in any way putting any animal under a degree of stress that is not monitored well and covered for.
I didn't say monitored well, I said justifiable,
Sure.
given the potential benefits of the research.
And so there has been translational benefits from this research, which has been shown and proven. And I believe every member of my committee would stand here and say that we believe that the animal welfare and the translational ability would be justified in this experiment. Otherwise, the experiment would not be allowed to go through by our committee.
I understand that the University of New South Wales has, just in 2021, put $250,000 into researching alternatives and basically doing things better using the 3 Rs. What has the University of Newcastle done in terms of actively providing funding for alternatives?
Well, a number of our researchers are already pursuing alternatives for studying this conditions such as chronic obstructive pulmonary disease, issues like use of cell cultures or organoids we heard about in other presentations.
Yeah. Is that something your university is funding, though, or is it something where there happens to be people at your university that are funded to do it by someone else?
People will be seeking funding from our external funding bodies like the NHMRC and so on,
Right.
using those technologies. I'd have to take it on notice in terms of the specific funding that's been provided from the university through its internal grant scheme and so on, or other initiatives that would have directly supported those activities. But I can take that on notice and provide
So if I
that
could then
information.
ask you, Dr. Raw, it's correct, is it, that the university itself is providing that 250,000 in research funding?
Yeah, we have a unique system in that we encourage a centralized approach to buying equipment. So rather than having a microscope in one laboratory, we have a central facility where all the researchers can go and utilize that. And the idea of funding the organoids initiative came from that strategic overview of how do we fund all of our facilities for our researchers in general. So that's why we're a bit different to most other institutions. And obviously in that case that's a big benefit.
The Honourable Lua Marto, who's on the screen.
Yes, thank you, Chair. Look, I want to thank each and every one of you for coming today. With your insight and experience is invaluable to this inquiry, and really appreciate the knowledge. Look, I'm going to start off perhaps with yourself, Professor Dunn. In your submission there, what I found quite interesting was you mentioned it about a Bionic Eye. And the reason why I mentioned the development of Bionic Eye, my brother lost the eye when he was 3 years old to retinoblastoma. Can anyone hear me?
Yes.
Oh, good. I just thought maybe you could just give the committee a bit of insight into what's happening in that area. I know back then, this is going back in the 50s when they had the eye removed, but back then a lot of children, the cancer then went from the eye and ended up going into the brain. I just wonder if that's still occurring, if it's not still occurring, and would we've had these advancements if animal testing wasn't available?
Right, well thank you for the question, and I'm pleased to have the opportunity to speak to some of the benefits of the research that comes from the work done by our researchers. So, as submission describes, the research into retinal diseases at the University of Sydney has led to the development of the Bionic Eye, and that technology can be used for individuals who have vision problems from a variety of causes, including tumours. But also other causes, and the development of that technology, and it's now the fact that it's at the stage that it can go into clinical trials to be used in humans, is really the result of the fact that we've been able to progress from a very basic development of that technology in just the electronic sense. But being able to test it in animal models, so in a functional eye, and that is connected to a functional visual system of the mammalian brain. And that's not my area of research. I'm a neuroscientist. But my understanding from the work and what I know of it and what I've read about that work is that we would not be at the point that we are now where we can use that technology and trial it in humans if we did not have the ability to trial it in the animal models.
No, thank you very much for that. A question perhaps to all of you. So what advances have you made, let's say in the last decade, that otherwise wouldn't have been achieved without animal testing? I don't mind to answer.
Where do you want to start?
I think
Shall we start?
Yeah, that's right.
Chomping at the bit. Okay, shall we start? Listen, there's no particular order, but we'll start with New South.
Yeah, it doesn't matter.
No, I think the most convincing argument for me is what we do in our Children's Cancer Institute, in that we take extracts from a tumor that a child has, a hard-to-treat tumor, we transplant into a mouse, and then we use all the current treatments that are possible to test that on the mouse, so that we can apply that treatment on that particular child or future children who have that specific cancer. So I think that's the most convincing argument for me.
Thanks. That's a great case study. How about Sydney, then Newcastle? How's that?
Well, one of the things that has really been one example of the many developments that we've had from our work at the University of Sydney has been the development of a new potential treatments for opioid addiction. So one of our researchers at the university has now developed a compound which appears to be very promising to actually treat opioid addiction that we know is a problem in Australia and is a huge problem globally. And there are very few effective treatments for that, and that the burden of that disease for globally is very, very high. So one of our researchers has come up with a compound. They have been able to test that initially in cell culture systems and other sorts of systems, but based on the data from those models, they then progressed to animal models. They got very promising data from that, and it is now approved to go into clinical trials in individuals who suffer from opioid addiction. So that's one example of where I believe that we've made significant progress in a disorder which has a very global high burden, both for the individuals who suffer from that, but also for the community. And we certainly wouldn't have progressed as fast if we weren't able to use animal models, and very, very likely we wouldn't be at the point where we are now of being able to go into clinical trials if it wasn't for the animal research that was done in that space.
Thank you, Professor. University of Newcastle.
Yes, in my opening statement I mentioned a couple of the examples of initiatives that have occurred through the university in recent years, but in addition to that I've mentioned some of the work that's undertaken around new cancer treatments. One of those is a cancer treating drug called Cavitap, and before undertaking the initial clinical trials with this work, with this new treatment, if the treatment itself required validation using original mouse models to do that. So the animal research was fundamental to that development. This has been shown to be highly effective in combination with what's now called immunotherapy treatments, particularly for advanced melanoma, lung cancer and bladder cancer, and is now entering the early clinical trial stages. So it expands our repertoire of cancer therapies, particularly for some of these very debilitating advanced diseases. There may be other examples my colleague has to bring to mind.
Certainly, I work quite closely with some researchers who are doing some amazing work in endometrial disease, and a disease that cancer is often found very late in life. And some of the research they've done has been able to indicate there may be some markers we can look for earlier that could help the diagnosis of this disease before it's reached a stage but is no longer treatable.
Thank you.
Thank you very much. And my last question before I pass on my colleagues. I just have to ask you yourself, maybe Professor, in your submission there, you mentioned that research is in international effort. And you go on that, you also receive significant funding from international sources, such as the US, the EU governments and charitable organisations, such as the Bill and Melinda Gates Foundation. Can you just tell us a bit more about what you have to do to try and get that that sort of funding, and how much of that funding would equate to what your university gets? So what I'm trying to get at is trying to work out what exactly do you have to show these other organisations overseas to get your funding, and what percentage of that funding would go to your university?
So the actual amounts of funding, they obviously vary, but overall the process is the same as applying to NHMRC ARC in Australia. They have international funding schemes, so the Bill Gates Foundation, for example, has funding schemes where our researchers can apply to, and our Central Research Grants and Contracts Office facilitates that. They inform researchers about international and national grant schemes. In other instances, the researchers can directly approach funding bodies and say, look, this might be a good idea to do that, especially something like the Gates Foundation. But in general, it's a competitive grants process, if that answers your question.
That's right, and also you have to demonstrate a high level of animal efficacy to them as well.
So the animal ethics side?
Yes, that's correct.
Yeah, so look,
You obviously have to demonstrate that to them in order to get those fundings.
yes, there. So there are a number of accreditations that are viable at an international level, but in general, all of the research that is conducted here is reviewed by our animal ethics committees according to the Australian code and legislation. So there is no difference if you get money from the US or from another country. If the work is done here, it's the Australian code that applies, and our ethics committee reviews it.
Thank you.
Thank you very much for that. I'll pass it on to my colleagues.
Thank Leonardo. Honourable Westby.
Thank you, Chair. Dr. Raw, I was just listening quite intently to when you were talking about the impact that the animal research allows you to have in the children's cancer space. And I just wanted to give you the opportunity to provide some insights to the committee and I guess to the broader public about the real personal benefits that that come out of the work that happens in that space, i.e. The parents, the children that are actually affected on a personal level. Can you give us some insights as to the life changes that are made and how people react when there's the ability to find a treatment that works for a child with cancer through the work that you do?
I think there's a very good website that demonstrates the responses by parents and children to the treatment. That's probably the best I could. It's publicly available. I think that's the best statement that can be made in support of this research. There's some great emotional videos and so on, evidence of that on the Institute's website.
Yeah, and look, I think that's really important that we communicate across that the impact on a personal level that this is able to make in people's lives. Can I ask, in the instance that the testing that is done wasn't available to you, i.e. there was perhaps a restriction or a ban on animal testing or animal research, would you be able to have the same outcomes by another means?
The only means I can imagine at the moment is to test different compounds on the particular child.
And that would obviously have I would imagine, a drastic and detrimental effect on that child's welfare potentially. Would that be a fair assumption?
I mean, a comparison is chemotherapy and the different methodologies that are trialled on patients, and obviously the effects are significant.
So, when we're talking about the lives of small children, and their parents who have, I guess, put their hope and trust in the hospital and you, what do you think would be their reaction to not having access to the ability to do what you do at the moment, should we move away from the testing regime or the research regime that we have with animals at the moment?
The equivalent I could imagine is there is medication available that is only available in some countries overseas where it's being developed. I get a lot of complaints in instances where people are ineligible for clinical trials where there is a last resort medication. Mm-hmm. So they are very distressed because they're ineligible because they got some other disease and so on. So obviously very stressful if you can't get that medication.
Yeah. I just wanted to turn to Dr. McCarthy for a minute, and we've heard a lot of criticism during this inquiry, and look, I think it's quite targeted at some of the events that have happened. Could you just briefly, again noting the time, could you just briefly provide some insights as to, on the scale of the amount of testing that and research that happens in your organisation, what percentage would you say would have the events that have been highlighted during this inquiry have made up in the total of the research that's happened? 1%, 0.5%?
Again, I think I noticed the exact percentage, but it's a very small percentage. So
very minute.
For example, 127 mice were smoked in the last 12 months.
Okay.
So that animal model is a very small percentage of what work is being done at the University of Newcastle.
Thank you very much.
Thank you, Chair. I'll move on to some questions from myself. I'm going to quote from the University of Sydney submission, but I think it's a theme that's been picked up by other witnesses to the inquiry up to this point, particularly from the university and the research institute sector. Just on page number 5, just take you to page 5, and I'm going to actually ask each of the 3 universities to comment on this and share their thoughts, but on the top of page 5 there's the heading current initiatives for 3 hours research available to Australian researchers. Specifically, I'd like to just draw your attention to the first paragraph, and I'll read it out. It says, compared to other countries, there is a paucity of funding for 3 hours research, including research into alternative research methods and technologies that is needed to transition away from the use of animals where possible. And indeed that was part of the opening statement. The professor incorporated that observation. I'm just wondering, from the point of view of the 3 institutions, whether you agree with that statement in general about the matter of paucity of funding around 3 hours research. And then perhaps your thoughts about how we may progress doing this better, because it actually has been a consistent theme, particularly with reflections on other jurisdictions, particularly overseas, that appear to be doing this better than we are. So perhaps if we start with the University of Sydney.
Thank you, and thank you for the question. And yeah, this statement comes from our own submission.
Yeah.
So definitely, we definitely agree with this. So the funding which is specifically available to conduct research into the 3 Rs at the University of Sydney is no funding. So we have a small prize available from the DVCR's office, which is
just for the purpose of hands, just that acronym DVCR there.
Oh, sorry, the Deputy Vice Chancellor.
Thank you.
For research, to encourage work that supports the 3 Rs. There is a annual prize available, which is for $4,000, which encourages and acknowledges the work of researchers that do research into the 3 Rs, and that can be any of the 3 Rs. However, the funding is that is set aside for that is $4,000, and that's obviously very, very modest. And because it is a prize for work already done, it does not support the research itself. It is acknowledging research that has been done. So that means that if researchers at our university wish to do research in this space, they have to find the funding for the research initially. The problem there is that we do not have funds available specifically for 3 Rs research in Australia that is from the state or federal governments. And really I would very much encourage the New South Wales government to urge the federal government to support work in this area. Because if we want to move more rapidly towards replacing animals in research, this is what is needed. Because this is the reason why we are still using animal models, because we do not have valid alternatives available. And the reason we do not have valid alternatives available is because we cannot do the research to develop those in most cases. So we do have many researchers that are very vested in animal welfare, and they do spend time trying to refine the work that they do to reduce the impacts on animals. but you've already heard from the witnesses that were here previously that the success rates for competitive funding from the NH and MRC currently is 8%. And those are all going to be research projects which are not specifically towards the 3R. So I've been doing research in Australia funded by the NH and MRC for over 3 decades. And I'm very, not only applying for research, but I spend a lot of time reviewing projects that come through, and none of those are specifically for 3Rs. And the reason for that is because the researchers know that they will not get those funded because there are no specific funds for it. So what we really need is a system for sufficient funding to really enable 3Rs research to occur. And we have suggested in our submission that a 3Rs research centre, which is national, that is similar to the ones that are available in the UK, in Europe and in the US, would probably be the best way of doing that. And so we would encourage the committee to consider this.
Thank you. I'm just wondering from the other institutions, just reflections on the way in which we can drive the 3Rs thinking here in Australia, or New South Wales, perhaps Australia more broadly.
I think the critical points that are coming out of this discussion is that it comes down to funding. So, and also advances in technology. So the only reason why we are able to develop our organoid center is recent advances in MRI and in 3D printing. So there are two techniques that are really critical and they cost a lot of money. And there are no, if you want to apply to our current funding schemes, there is little success rate anyway. So a specific initiative that could be started by New South Wales would be great help. So for example, like the recent initiative of the RNA Institute, so there's a real possibility to dedicate funding and to get something off the ground fairly quickly.
Just on the example you've given, Dr. Did you say NRA or NRI?
RNA.
Apologies.
RNA,
Get my acronyms mixed up. RNA. Could you just elucidate that a little bit? That's been a recent initiative that's taken root.
So that's the recent developments in the COVID vaccines.
Yeah.
So that there is now a research institute in New South Wales that is part funded by the government, as I understand, and the universities to get more of that type of research off the ground.
I moved to University of Newcastle. Perhaps if we just add a layer of further reflection on it, about the federation we have. We've got the Commonwealth at one level doing its regulating and substantial funding for the research that flows down to the universities and institutes. And then we've got the state governments. And of course, across a whole range of domains, there's always this tension and perhaps lack of coordination and clear channels of operation and synergy and all the rest of it. Any thoughts about how that can be overcome? And I just note that I think perhaps it was you, Dr. about the reference to the gene technology regulator as being a potential model or potential framework. Might come back to you, Doctor, but to Newcastle.
Yes, thank you. I appreciate the opportunity to comment on this, and I echo a number of the points that have been made already and reiterate our commitment to the 3Rs in animal research. And I think the issues reflect a number of things. Our existing commitment is indicated in our processes that are undertaken around research already to ensure that not only our researchers understand the 3Rs, but there's an evidence of against those in any application for research. So it relates to research practice as well as research resources and methodology as well, and how we advance and innovate our scientific methods, our knowledge and practice. Researchers are committed to innovation. That's in their DNA, if you like, to be questioning, to be thinking about how to improve their methods and so on. So we need the resources, the support and the infrastructure that encourages and supports that innovation. The sorts of initiatives we've been hearing about are very important elements of that, the infrastructure that can support alternative methods of research. We've heard already about initiatives such as organoids or different ways of clumping cells so they can be studied that might replace the need to do that within animals. It's not perfect, but it's another model. There's also use of mathematical models and biostatistics and expertise in that area, and other areas strengthening biobanking so that tissue banks can be used rather than using in vivo studies, for example. And there are enormous advances being made in that regard. The other is the coordination of central animal resource facilities so that it reduces the variability between different animals, and it creates greater validity and reliability for those animals that are used in research. So the Australian facility that exists in Mossvale around that is a very important element of that infrastructure. So all of those things I think are important directions for future investment in this area to strengthen the 3 Rs.
Doctor, do you have any comment? Sorry, did I misquote you about the reference to the gene technology regulator? Was that yourself or was it Professor?
Yeah, I think I commented. So the Office of the Gene Technology Regulator is a Commonwealth agency that focuses on the Gene Technology Act, which is a Commonwealth legislation governing the use of genetically modified organisms. So as opposed to animal research, where we have the research legislated at a state level. So we have differences between states. And our research is at least national, but even more so international. So I think at the moment we don't have an Australia-wide focus on reporting. That idea of transparency. We have to report different statistics to different states, and then they get differently reported to the public. So and the legislation differs between states. So the Office of Gene Technology Regulator works very well from what I can see, and my colleagues would be an idea.
Perhaps on notice, I'll invite you to just reflect on perhaps a pathway of engaging the change of government, how, and it'd be interesting, a new minister in the role, how there might be an ability for the state of New South Wales, which is the largest in terms of its population and its economy in Australia, to sort of try and drive this with the new government. Just on this, and probably my last question, just on the matter of, and others have raised it as well, the inconsistency of reporting of statistics. It's, as I said, been raised as a bit of a bugbear by other witnesses. I'm wondering, invite each of the institutions to comment on that, and any reflections?
Sure, thank you for bringing this up again, because I think it's a really important point. So as I said in my opening statement, the problem is that there are differences in the requirements across states and territories about what has to be reported and how animal use numbers are reporting. So it's very difficult to really understand what's being used across states. Then it's very difficult to really get a grasp on the scope of animal use in Australia compared to other countries, where other countries in the mains, for example major countries such as the US and the UK, only report animals that are used directly in biomedical research, and they only report the animals that are actually directly involved in the actual experiments. So animals, for example, that are used for breeding, the animals that are subsequently used would not be included, despite the fact they're bred only to produce the animals. In Australia, it's quite different. So we include all animals. So that's, for example, if we're talking about rodents, it's about the animals that are initially breeding the animals that might subsequently be used. Animals, as soon as they get to half of their gestational period, are then counted, even if they are used prior to birth in the experiment. So sometimes we use embryos. They would also be counted. They would not be counted in most countries in the world. And the thing that really blows out numbers, the apparent numbers used for animal research in Australia, is the fact that we don't just count the animals for biomedical research. So if people are doing wildlife research, if they're doing veterinary research, if they're doing stock research, for example, cattle research or chicken production, egg production, which is a large amount of research that comes through our Animal Ethics Committee, all of those animals are counted. Let me give you an example. If someone's doing purely observational research, so they're doing research on, for example, a fish species, and they're only looking to see where the fish are and how many there are, for example, they set up cameras and they're watching the fish swim past a camera. If they have 60,000 fish that swim past that camera, they count those as being animal use. And if those animals then turn around and swim back the other way, we don't know whether they're the same animals or not, but that would then be, they would be recounted, so that would be another 120, that would be 120,000 fish. That would be used as animal research usage, whereas in any other country in the world they would not be counted because they're not biomedical research. So there are large inconsistencies about the way we do it. So, and that's fine, I don't mind what the system is, but we need clarity around what it is we're reporting, and we need to divide it between the different types of use of animals, and then we need greater transparency for the public so that the public understand exactly how animals are being used, rather than just having these very large numbers and then making assumptions about the sorts of research that those animals are being used for.
I know I've just gone over, but quickly with respect to New South and Newcastle, any comments about the statistics and its gathering and its
I think from my view the critical part is the meaning of collecting statistics, right? It is one side to collect numbers, it's another side to produce information to the public, to the informed public, that they get what they want. And I think this needs should be constructed by academics, say in the social sciences, that are familiar with these types of surveys, rather than by a regulator saying, okay, give us this number, give us that number, because that doesn't translate into the public understanding what these numbers are meaning.
Thank you.
If I can just briefly give an example, including the fish, we have some amazing research in the conservation of the bell frog in Newcastle, which has been decimated due to a fungus called chytrid. We have to count the tadpoles. So these are, hundreds of thousands. And so the numbers that are being obviously published are again, like the fish, these are animals being observed, not acted upon. And it really does inflate those numbers substantially.
Well, on behalf of the committee, thank you so much. It's been very helpful to us. First of all, thank you for the submissions in the first instance. But coming along today, I know you're all very busy, so to carve out that time to come along, it's much appreciated. I'm sure there will be some questions arising from this. Once we've read hands up, I'm sure we'll stimulate some further questions, so we'll provide them to you. And once again, thank you very much for making yourself available.
Thank you. Thank you very much. Thank you.
We have a short break now, and we're back at 11:30. Our next set of witnesses, most of whom are joining us remotely, so thank you all very much for doing so in that regard. And we have Professor Charowski joining us in person today, so thank you very much. So what I'll do, I've got a list of the witnesses in front of me. You don't have this, but it's an order that I'll follow in terms of going through the formalities of having you each first of all identify your name, your position title in terms of the organisation you're representing, and swear either an oath or an affirmation, whichever you prefer, the words of which should be before you. If for some reason they're not there, put your hand up and we'll forward them to you. So in the order that I've got, if we could please start with Professor Sloan.
Good morning, my name's Professor Alistair Sloan. I am currently the head of school at Melbourne Dental School and Professor of Tissue Engineering, and I'm representing the Medical Advances Without Animals organisation. And I swear that the evidence now about to be given by me shall be the truth, the whole truth, and nothing but the truth. So help me God.
Thank you very much, Professor. Professor Sharonowski.
I'm Professor Wojciech Szanowski. I'm a Professor of Nanomedicine at Sydney University, and I also represent at the same time Medical Advances Without Animals.
Thank you.
I swear that the evidence now about to be given by me shall be the truth, the whole truth, and nothing but the truth, so help me God.
Thank you, Professor. We'll now move to Ms. Wallace.
Thank you. My name is Paula Wallace. I'm representing Liberty Foundation Australia Limited today.
Thank you.
I solemnly, sincerely and truly declare and affirm that the evidence now about to be given by me shall be the truth, the whole truth and nothing but the truth.
Thank you very much. Miss Stephen.
My name is Nikki Steenham. I'm co-founder of Legal Freedom Australia. And I solemnly, sincerely and truly declare and affirm that the evidence now about to be given by me shall be the truth, the whole truth and nothing but the truth.
Thank you very much. And Ms Burke.
Good morning, my name is Tam Burke. I represent as the co-founder of Legal Freedom Australia. I solemnly, sincerely and truly declare and affirm the evidence now that I'm about to be given by me shall be the truth, the whole truth and nothing but the truth.
Thank you very much. Now, just in terms of confirmation of receipt of the submissions made by the respective organisations, we have Professor Sloan and Professor Jorinsky, submission number 351, generically under the name of the MAWA Trust. You've got your own professional representation as well, but there for the trust. That's been processed and has been duly received as the submission has been uploaded to the inquiry's webpage. Ms Wallace, in regards to your submission today for and on behalf of the Liberty Foundation Australia, that's once again been received, processed and stands as a submission and uploaded to the webpage, and that's submission 213. Thank you for that. And with Legal Freedom Australia, your submission is 241. Once again received, processed, duly stands as submission, been uploaded to the webpage for the inquiry. We've got now time for opening statements from, we normally do it by organisation in terms of the opportunity. So if each of you have your own opening statements, that probably would be worth telling me, but now I presume there is a decision with some organisations there'll be a spokesperson to give the opening statement. Perhaps if we start with MAWA, was there going to be the single opening statement to make, and if so, to provide that?
I believe there are two separate.
No, that's fine. Well, that's okay. That's okay. We'll just go through them then. Just with respect to Liberty Foundation Australia, there's obviously a submission, opening statement there, and with the Legal Freedom Australia, was it going to be one opening statement from that organisation?
Yeah, just one.
Okay, thank you. Well, we'll start then with MAWA and your opening statement. We'll start perhaps with Professor Norim.
So I think that Alistair will go first.
Okay, no, that's fine. Yep. No, that's fine. So, Professor Sloan.
Yeah, thank you very much indeed. So, Bionic Advances Without Animals is a registered charity established in 2000, so it's been going now for some 20 years plus and takes a leading role in representing animals in medical research, with a clear focus on replacement as one of the 3 R's. It operates as an independent medical and scientific trust to encourage and develop research to look at the idea of replacement model systems to replace the use of animals in research. This is probably the 1R out of the 3 of refinement, reduction and replacement that is the hardest to achieve. It is also probably, in my broader experience as a biomedical researcher in Australia, but also the majority of my time in the United Kingdom, the most challenging to actually undertake, because Marwa quite rightly is focusing on the idea of absolute replacement. So that means any culture media, chemicals, antibodies that you would use as part of your research has to be produced or created without the use of animals. That has naturally been the most challenging of areas, but certainly in the United Kingdom has certainly taken a significant step forward, and Marwa's focus and funding research in that area has driven this forward as well. I think Marwa's focus has been to try and encourage young researchers to actually adapt their biological research environments and their research streams to address this significant point and to advance their own research by developing non-animal based models, whether that be laboratory based, mathematical based, computer algorithms, machine learning, and so on and so forth. It's providing leadership in Australia and is actually lobbying significantly for the allocation of sufficient funding and research support and other incentives so that young, early career researchers can address this neglected issue. Thank you very much.
Thank you, Professor.
I'm Professor of Nanomedicine, working primarily in the respiratory health. And if you take the respiratory health as the research, one could say, oh look, animal models should be the gold standard for you. But in reality, there are only two models which represents human physiology, but very poorly pathophysiology, which is baboon and sheep. And if we have to do 100 samples, imagine how many animals would be sacrificed and what is the ethical application of these. Any other model, small animal, rodent model, the correlation to human physiology and a translation to clinical trials is zero, absolute zero. Different aerodynamics, different number of lobes, different cells, no cilia, no mucus. So why do we test using these animals? And for that reason, alternative models are the only option for us. The alternative models building up these artificial, or I should rather say, mimicking structures of human physiology are really great option because we can build a lot of them. So what it means, we can create hundreds of models and do the high throughput screening. They are reproducible, they are reliable. On top of this, they allow rapid screening, and also they allow us to take the cells directly from the patients, which means that we can create models of each of the individual patients for personalized medicine. So why we use still animals? But the global interest in alternative is huge. It's $1 trillion business at the moment with over 300 established companies producing alternatives. DARPA and the DST are good examples of putting millions of dollars into alternatives. Why? Because if they test biological weapon, there is no ethical approval for animals to be used for that. Also, FDA has now iStent process which allows the submission without the animals. First time in the world, first time when we hear about this. CN Bio, one of the companies we actually use these systems, was named one of the top innovation companies 2021 by The Scientist. Big Pharma, Roche and AstraZeneca invested hugely in developing these models because they know that the animal models don't work for them. And also this model allows for the mitigation of the recalls of the product on the market because we test safety very effectively. 3D printing or bioprinting, huge business. Why? Because they produce the models which represent human pathophysiology. We need to build models not only physiology but the disease. None of the animals can represent human disease. On top of this, we are built of cells and microbes. And this is a critical element. None of the animals, we actually have exactly the same number of cells than the microbes. And we are consisting of these two worlds. None of the animals has the same microbes that we have. And they help with treating the disease. They help with recovery from the disease. So the situation here is, in my view, win-win. For those who like the animals, we need them. We need to verify our models. And we still need them in the final stages before we go with our clinical trials. Why? Because we'll screen our molecules, our materials, chemicals, etc. in the alternatives and go with a very high confidence to only a few animals before we travel to humans. I believe we have two options. Just to watch what the world is doing and in a few years wake up and follow what they've done, or be part of the leading team. And potentially, being at New South Wales University, also leading here, I believe New South Wales can lead the development of the Discovery Centre in alternative models and create world-class centre in this state. Thank you.
Thank you, Professor. That's very helpful. No doubt will lead to a number of questions when we get to that part of the session. Ms. Wallace, can I invite you to make your opening statement?
Yes, thank you very much. Liberty Foundation is a registered charity that rehomes animals from research and has been active since late 2017. We're a non-political organisation which does not take any formal position on whether animals should be used for research and scientific purposes. We're not aligned with any other groups or political parties. We appreciate the opportunity to give evidence today about an issue which I believe is relevant to your inquiry. and worthy of consideration in your recommendations and your final report. My submission provided comprehensive information on the number and types of animals used in research and the potential demand for specialist rehoming services in Australia. I won't attempt to summarise that here, but I will make the point that there is no question that providing rehoming presents a more ethical approach as opposed to euthanasia or sending an animal to another research facility. In my experience, there is a high percentage of the animal-based research industry that believes rehoming is a win-win for everyone involved. And like the Australian Code for the care and use of animals for scientific purposes, or the National Code states, rehoming should be considered wherever possible. Ethically, I don't believe there's any question about why anymore. It's more a question of how. And that question is now being answered by the work that we're doing and groups like Beagle Freedom. However, we need to remember that although rehoming has been enshrined in the National Code for some time, actual rehoming activity has only become more commonplace in recent years. There are a number of reasons for this, one of which involves money and convenience, which should not be driving factors in deciding the fate of animals used in research. They should not be euthanized because it's considered cheaper or easier. We believe the industry requires a greater push towards compliance with the code around rehoming and reconsidering its ethics around the fate of and reuse of animals. For introducing a mandatory rehoming and a mandatory retirement age for domestic animals in research. Alongside this, we must also consider how we're going to upskill, fund and support animal rehoming charities to meet this increased demand. We would like to see a rehoming task force formed to bring together animal rehoming groups with the industry and government to develop solutions and build capacity in the rehoming sector. This must be a collective effort involving all players. I was heartened by the news yesterday that the Victorian Government has supported many of the recommendations of the Task Force on Rehoming. It will support a review of the National Code to include mandatory retirement age for dogs and cats in research. It will also consider introducing a mandatory retirement policy in Victoria for dogs and cats. I hope this inquiry considers making similar recommendations and that these receive the full support of government. Thank you.
Thank you very much for that opening statement. We'll now move to Beagle Freedom Australia. I'm wondering who will be making the opening statement in regards to that organisation.
I'll be doing that.
Please proceed.
Good morning, everyone, and thank you for this opportunity to speak today. We're both appearing on behalf of Beagle Freedom Australia. We're a not-for-profit organisation and we specialise in the rehabilitation of rehoming of ex-research animals, specifically beagles and other dog breeds and cats. Beagle Freedom Australia was established in 2005 as Beagle Rescue Victoria. We eventually began rehoming beagles from research facilities across Australia and so launched Beagle Freedom Australia in 2013 to cater specifically for this purpose. At BFA, we take any animal that has been used for medical research and find them a family home. We've rehomed hundreds of dogs and cats and we have had a 100% success rate. Every single animal we've rehomed has been able to be acclimated to a home environment. We provide extensive veterinary care for the animals we take in and we continue to provide this care for the life of each animal if necessary. Since our launch, we've been campaigning to various governments within Australia to implement mandatory release of research animals at the end of their use in research for the purpose of rehoming. In 2018, we worked in conjunction with Biosecurity at what was then known as DTJR Victoria to develop a rehoming guideline document that was then published and sent to all the registered animal ethics committees as a best practice guide to follow. It opened some doors, but rehoming remained a voluntary practice in Victoria. Earlier this year, the Victorian Task Force for Rehoming Pets put forward a report detailing multiple suggestions to improve the lives of pets in Victoria. Two of these recommendations came from our proposal, one being the mandatory release of animals from research and teaching for the purpose of rehoming, and two being the retirement age for animals used in research and teaching. And yesterday, as Paul has said, the Victorian Government released their response to this report, supporting both of these suggestions as well as many others. Today, we're here to discuss these same two suggestions and in the hopes that common sense will prevail and that the New South Wales Government will follow suit. We'd also like to see several other improvements made, including upskilling for facility staff, enhanced enrichment programs for facility animals, staff to animal ratio regulations implemented, and the standardisation of current best practice documentation, as well as a maximum term of no more than 3 years for members to sit on an animal ethics committee. We believe that these improvements will aid in the enhancement of the life of the facility animals, both within the facility and in their new homes. Thank you.
Thank you very much. That's a very concise and clear opening statement. I'm sure there'll be some questions arising from that, so thank you. We have representatives at the table here from opposition government crossbench. So if you're agreeable, we'll share time between those three groupings and we'll get that underway with the Deputy Head on the Wednesday.
Thank you. Thanks, Chair. I'll just start with Ms Steindman. In your submission, you talk about making rehoming mandatory. I understand that there are guidelines. I'm just wondering why a guideline is not enough. Why do we need to move into a mandatory model?
Sorry, could you just repeat that last bit? You was cutting out.
Sorry, so we have guidelines at the moment in regards to rehoming. So I'm just wondering why guidelines are not enough. Why do we need to move to an actual mandatory model for rehoming?
Because the guideline is exactly that. It's just a guide. It's not enforced, and there's nobody checking to make sure that it's actually being done.
So you think that while the work that you and Liberty Project are doing is saving lives, that there are some facilities and some animals still falling through the cracks?
Definitely, and it takes time to convince facilities to release their animals to us. So, it can take, for us it's taken up to 4 years for some places to convince them that we would do a good job at rehoming their animals. So I would hate to think in that 4 years of us, discussing and going back and forth of what's been happening to those animals in that time. So if you took away the need to have to negotiate, it would make it a lot easier for us to actually do our job to rehome. And we could just, they would have to release them to a rehoming facility like ours.
And are there problems with the non-disclosure agreements? I understand they can be quite variable, but does it sometimes make an animal difficult to rehome if the non-disclosure agreement goes too far?
In our experience, we haven't had a lot of issues with the non-disclosure. I mean, it's like any privacy policy, really, where we do tell our adoptive families that they're adopting an ex-research animal. We don't get information on what research they were used in, and we wouldn't, even if we didn't have the non-disclosure in place with the facility, we wouldn't share what facility that animal was from anyway, because it's just a matter of privacy.
Thank you. Ms Wallace, there was an article in the Daily Telegraph on the weekend that proposed that cats and dogs were being sourced from online marketplaces. As someone that's involved in this space, is that something that you have personally heard of, or are there other places that you're aware of that facilities are locating cats and dogs?
I can't really comment on where research places are procuring animals. You'd really have to ask them directly. I'm not privy to that information. And in terms of a research facility procuring an animal from a website like Gumtree, I think the onus here, from my perspective, would be on the unscrupulous people that are exploiting animals for profit on Gumtree. I mean, the reality is anyone can buy an animal from a website, whether that's ethically acceptable or not. Really, that's up to the Animal Ethics Committee to decide, but I don't think anybody should be buying animals off Gumtree.
Thank you. I might go to Professor Janowski. I hope I pronounced that. I probably haven't, but I wanted to talk about, you talked in your opening statement about some of the concerns about lung issues, and we've been talking today about the smoking mice model. Is that something that you consider problematic from a science perspective when you're looking at studying lungs, and these are mice and rats that are being used in these smoking models?
Absolutely. The main problem is that the lungs in rats, mice, do not represent at all physiology in human, zero. And the other thing is that smoking is very different to humans than to animals. You cannot give a cigarette directly to the mouth of the animals. And the same is if you would like to develop a treatment, you cannot use the same devices. You cannot use the puffer for asthma to the animal because the mouth is not fitting there. So physiologically it's very different, and as I mentioned, the aerodynamics in the lungs of animals is completely different than the one of the humans. They have 2 lobes, we have 4, 4 which are actually both of them are different on both sides because we have a heart as well. And this makes even injury completely different, and the deposition of chemicals completely different than what we have in a human lungs. Hallmarks of the disease in the small animals, in the rodent models, are very different to the hallmarks of the disease in humans. So if you would like to now develop a treatment or even study physiology of this disease, there's actually no way of doing this properly.
Are there currently alternatives on the market that you're aware of?
There are some alternatives for this. We buy some of those as well as the controls, but we grow them ourselves in the laboratory. So we try to build human physiology. We take directly cells from patients, or we buy cells from human sources because you can buy them directly, and you can differentiate them to different parts of the lungs. You can do the small airways, you can do the upper airway, trachea. You can create certain parts of the physiology, but this is not entire lung. So that's the limitation. But it is a certain part of physiology which you can study in depth and large number and reproducibly.
And so do you think that, obviously alternatives is something we've talked a lot about in this inquiry. Is it the case that there are sometimes alternatives available, but they're just not being used and uptaken? And is the solution to that there needs to be more funding and efforts towards these alternatives, and also retraining for people who are using animals to actually understand how to use the alternatives?
So funding is the main problem here. And if we look on Europe, US, even Korea, they use a lot of alternatives. I think the funding there is, more for those kind of research. There are huge projects which are funded in Europe, which are purely on the alternatives, building lung and gut axes, brain, gut, and lung axes. This is something which is critical. Organ crosstalk is very important. So it's not only a single model, it's a combination of models, which is very, very important. And DARPA put $76 million into developing the artificial models, which is the multiple organ models. So this is one thing. Funding is limited. I would say it's not existent in Australia. We've never been successful in acquiring any major funding for those kind of models in Australia. Everything was coming from overseas. Retraining, I don't think there's a problem with this, because those people who do histology for lungs is exactly the same histology or exactly the same testing. Sometimes it is maybe education in this space and understanding that they can benefit from alternative models. And some people might be more conservative than the others. Some may say that there is an opportunity actually in these models and drive this even to the next level beyond what we can get from the animal models, and get, in my personal view, more confidence with the products which go to the market.
Thank you.
Thank you. I'm also going to stay with you, Professor Chichanowski. Please tell me how.
Okay, Shanovsky.
Shanovsky. That's
yes.
so much
Ignore
easier.
The first 4, it's a SH instead of the first 4 letters.
Shanovsky. Shanovsky or Shanovsky?
W is pronounced as a V.
Thank you, Shanovsky. Thank you. So, Professor Shanovsky, in your submission, you talk about cultural change and the need for cultural change. And this has been reflected in some other submissions as well. How do you think we actually, you've noted a few things, for instance, researchers and scholars perhaps believing that if they use animals, it will advance their careers. How do we begin to kind of break down that culture to encourage the use of alternatives?
I think that there's one element, TGA in our country and the FDA in the US. If they say that they can approve the submissions without animals and they recognize alternatives as something which is equally good or even better than the animals, this will be a major shift. Because we look at this, we have a product, and can we get to this product to the market? TGA and FDA is the main places where we have to go for the approvals. And if they say, look, you have to have animals, then there is no option. But if they say, look, you can submit without animals, this gives us the trigger, we can actually use our research in the alternative models. The second thing is the publications. Obviously, we aim for the publications, and it is prohibitive, sometimes for us to publish in the top journals without animal models. And it's also working with the publishers and lobbying them and saying, look, these models are equally good. And they are examples of those groups who are lobbying. The one in this institute in Boston is the main one. They do a lot of work in lobbying journals and publishing. And even there was a paper when they said, we are waiting for the moment when we submit the paper with the animal models and the reviewers or the publisher will ask us, but can you please do it on the alternative models?
Yeah.
And we will reach this time. I'm certain.
When you look at the EU's phase out of the use of animals in medical research, my understanding is that the idea behind that is basically to act as another incentive to, like, this is going to be phased out, so now you have no choice but to develop alternatives. Do you think that in Australia we need something similar?
It is difficult to say because I still believe that there is a space for the animals in research, but not in the number which we have at the moment.
Mm-hmm.
So because bear in mind that these models are only models and they represent only certain parts of physiology. You cannot create entire human in these little plastic things. So we still probably need some of the animals with certain type of research, but the number will be substantially reduced.
Yep.
But if we have this kind of declaration that we cannot use, obviously everyone will move immediately to the alternatives.
You mentioned in your submission as well that the University of New South Wales and the University of Wollongong seem to be a lot better than some of the other universities when it comes to animal research. What do you think has led to the cultural change within those organisations that seems to be lacking elsewhere?
So I can maybe speak to the Wollongong, which has probably the country best and the largest bioprinting facility, and they developed these models. So for them, it was easier and cheaper to do their research not on animals but on these models, rather than hassle with the animal house ethics, all these approvals. But also, it is probably recognition that some of the fundamental science can be done at a much better level on the alternative models than on animals.
Yeah.
And sometimes it takes a few researchers who build a culture and perpetuate it to the rest. Some successes, maybe presenting these successes to the public, and some promoting them as well, and having some leverage in different places, including government.
Yeah. And can you clarify for me, when I was speaking at the last panel, I was trying to understand how much students use animals in terms of training as opposed to research. Are you able to give us any kind of insight into whether that is still going on in universities?
So in training, it depends what you consider as a training. If you consider PhD as the training, for us it's a training program. PhD is a training.
Right, okay.
So
in sort of below that, I guess.
Below is negligible.
Mm-hmm.
So it will be very little because of the cost. If we consider obviously veterinary degrees, they have to deal with animals, but this is not in the context of, I think, damaging the animals is actually opposite. But in terms of the training, if we take medicine, pharmacy, pharmacology,
biomedical research, I would say almost non-research on animals, but the one which I mentioned, dentistry as well, negligible, very little. So the animals will come when you do the PhD, when you are a higher degree research student, and I would even say only for those who are lucky and have a lot of money.
Yeah. Thank you. And just one question to the Liberty Project, if I may, to Ms. Wallace. What kind of transparency do we have over the numbers of animals who are dying or being euthanized in research facilities in New South Wales, and what could we do to improve that transparency?
What we have is figures that research establishments are required to produce in different states and territories. I think we can get figures for, I think, all but 2 or 3 states and territories at the moment, but the figures are not particularly transparent in that you can't discern from publicly available sources right now how many animals may be available and or suitable for rehoming at any given time. So, and it's not just me saying that. Like if you ask the Department of Primary Industries or you ask the Minister how many animals are available for rehoming this year, he would be unable to tell you. We simply don't know. We can make a guesstimate about what that might be. We have started collecting in the last couple of years statistics on the fate of animals in research in New South Wales only through DPI. Those figures have started to filter through since 2019. In my view, I'm not entirely sure that they're being accurately reported. For example, in 2020 figures from that were issued by the Animal Research Review Panel through DPI, they state that no dogs were rehomed in New South Wales. Now, I know that I received dogs for rehoming, and I think Beagle Freedom would concur that that occurred in 2020, but they're not being reported in those statistics. So, look, the statistics that are reported are what they are. They're the figures that the establishments have provided. If we want to have a look at what we think might be available for rehoming in New South Wales for dogs and cats, a very rough guesstimate might be 100 to 200 dogs a year and 100 cats currently, but if we introduced a mandatory retirement age or a mandatory rehoming policy, then those figures would go up by several hundred. In terms of the level of euthanasia, I mean, you can look at my report. My submission gives you the overall figures for how many animals are being used in research in Australia. It shows you how many remain after, so that they don't die as part of the process. They don't either die as part of the experiment or are euthanized. It gives you a figure of how many are left, and it's about 33%. So you could roughly say that 70-odd% of animals that are used in research facilities or used for research and science in Australia do die as part of the process. What we're saying is that not all of them need to. And yeah, my submission gives you as much really as we can discern from the figures that are available.
Thank you.
Does that answer your question?
Thank you.
Yes, thank you.
Chris Wright.
Thank you all for your evidence so far. Probably just sticking with, we're going back to Professor Shrinovsky and Professor Sloan as well. Just wanted to, I mean, all of the scientists, researchers that we've had so far over today and in the first day of hearings have all basically said that they are committed to the 3 R's. Is it a case, do you think, that that it's just a matter of everyone agreeing that the final destination of where we want to be is not to have to use animals in medical research? That's the final point that everyone in the medical profession essentially wants to get towards, but it's just a matter of how we do it or the pace in which that transition occurs. Professor Sloan, I think you've got your hand up.
Yeah, thank you. I'm happy to start on that one. I think every scientist will agree with what my colleague has already mentioned as well. Obviously, what you're talking about, we want to get to an endpoint where we don't want to be using animals or have such few numbers of animals in research. It becomes the norm. The barriers at the moment are around actual culture and funding. The real strength where I've come from, and I've only been in Australia 2 years, but the real strength of the NC3Rs in the UK, which has now been in existence for some 18 years, has been a research funding stream that funds high quality science to address the 3Rs and significantly trying to address those 3Rs equally with a focus on replacement, refinement and reduction. Replacement being the most difficult one, but the projects in that area have increased dramatically. They funded my research back in 2006 and 2008, and I spent a number of years on their grant funding panels. What you get then is a culture shift of people thinking how we can use different model systems and actually remove the need for using animals and excessive numbers of animals quite significantly. The other step change that I've been exposed to was acting in my previous role when I was at Cardiff University in the UK, was I was a 3Rs representative on Cardiff University's Biological Standards Committee, which assessed all animal licences. And the UK Home Office had a very strict policy of increasing the need to address the 3Rs in any project licence. And very quickly, researchers stopped playing lip service to that and because their project licences and their grants were not being funded because they were not taking seriously the other options and availabilities that were out there. So it's a culture change. And that culture change is funding. That culture change is how we as researchers and scientists approach the problems we're trying to address. And as my colleagues already mentioned, publication is one of the most significant barriers. To give you an example at the moment, with my collaborator at RMIT here in Melbourne, we have a great piece of research around antimicrobial coatings of implants. It's proven to deliver some superb data. We've tried to get that published now twice in very, very good journals, and we're getting reviewers' comments back and editors agreeing with those comments of, oh, you must do an in vivo experiment. You must put it into an animal model. And the answer is no, we mustn't. We don't need to. That actually would not be a good scientific move. But the barrier to publish that is with the editors-in-chief and the editorial boards of some of these journals. Now that has two effects. One is it prevents you driving culture change, and it comes back to what we've made in our statement with Marla, is it starts to actually have a negative impact on the publication ability of young early career researchers.
But why?
So we perpetuate a myth.
So why would they be so insistent that you'd have to use an animal model? What's the why would they be doing that?
To see how it responds in an in vivo biological setting. Our work is in 3D, it's in model systems, it's on surfaces, but still people perpetuate the view that the gold standard would be an in vivo animal experiment, even though the animal model we would use is so removed from the human condition, it would actually probably provide no additional useful data, because we're developing coatings for dental implants. The oral microbiome, the oral microbiology, the flora of the human mouth is different to any animal model system you would want to use. So the actual argument is flawed. But the insistence is people still think the gold standard will be an in vivo model to get your research published in some of the top journals. Now then, if we can actually start, it is changing slowly but surely. But in Australia, we need significant funding to be focused at this particular stream of replacement model systems. Reduction and refinement as well, but definitely in replacement. And that allows you then to really reduce significantly the numbers of animals that will be used in research. And if you just look at the animal statistics in Australia, the UK, they're significantly high. They're unnecessarily high.
But if you move, look, I agree with everything you're saying, but if you move too quickly, so if you potentially rushed a transition to a world without animals being used in medical research, wouldn't that then also have significant disadvantages in the medical and research benefits? Like, I understand it's more just a matter of what horizon do you think that we can get to that point of not needing to use animals?
I agree with you. I think ultimately you want to use as few a number of animals as possible and as needed.
Yeah.
At the moment, we can try and address that by power calculations to say we will use X many animals, in vivo animals, for our studies. But that's still too many because a significant amount of preclinical, laboratory-based initial development can be performed on lab-based studies in cells, 3D model systems, algorithms, machine learning that can drive a sort of narrative. Look at the pyramid. The bulk of the work at the base in the lab at the start of the project does not need to use in vivo animal systems. If you've got appropriate, we can develop appropriate models. But ultimately, you get to a point where you might need to assess this in a full biological system, so an animal model system. But you want that to be as limited and as small as possible, if you have to get to that point. And whilst doing power calculations gives you that number, actually, let's create model systems that means that you reduce the need for those numbers right from the very start. Systems I've developed have been shown to basically make a 50 to 80% reduction in the use of animals for the point in time that we're developing our therapeutics. An animal system gives you, if you're developing a therapeutic like I am, one animal, one times point, or one animal, one dose point. Whereas model systems can give you multiple dose points, multiple time points, and are highly reproducible. It means when you ultimately get to the top of the pyramid and translation into a true preclinical environment, you're asking the right questions, very specific questions, and the need for animals, if at all, becomes very, very low. And that's the journey we should be looking to progress to.
Yeah.
So I agree with everything which Alistair said, and I think how fast we go. Look, as Alistair said, you can do, instead of one animal, you can do 100 models. You can develop really confident molecules on confident treatment and go with these to one or two animals only. Otherwise, you would have to use 100 of animals. The reproducibility of these animals, but also possibility to create a personalized models. I can take your cells and create you in the dish and test these treatments whether they work or not. And that's the beauty. You will never do it in the animal model. And one of the examples with animals, if you house the animals in two different houses, the results are actually completely different. Even sometimes when you house them in the same house but in two different corners, your result will be different. So how do you normalize it, and how do you know that your result is actually correct or not? Because the microbiomes are different. And also what Alistair mentioned, and I mentioned also in the opening statement, microorganisms in animals are completely different. And we are holobiont at the moment. This is a new theory, which is how we exist in the planet. And we have to understand and consider always microbes and the cells together in the system. We also promote not a 3R but a 6R. And the additional 3 R's are rapid, reproducible, and reliable. And this is not something which animals offer.
Yeah, you're right. It's probably a matter of controlling the variables, which is basic in any any research you do. So from what the evidence you've given so far is that you're saying that Australia is a bit of a laggard in this regard. What would be some jurisdictions around the world that you think would be best practice, or that are doing a much better job than we are in terms of the 3 R's or the 6 R's that we might be able to look to as a committee?
I'll refer to Alistair, but for me as a researcher, if we apply for funding overseas, the models are accepted. If I apply for NHMRC funding, I get a comment, you need an in vivo model. You cannot progress anywhere because you need an in vivo. And then the argument which Alistair made, it is irrelevant. This model is irrelevant. If someone tells me that I have to use the mice for my lung model, I said, no way. But this is the problem. And I think the UK, Europe in general, European funding is a huge amount of money for safety assessment and alternative models. I'm actually part of the two large funding which was submitted recently, and the US. Military organizations like DARPA and a DST in Australia, I mentioned, they also put a lot of money into alternative models.
Yeah, Professor Stein?
I'd add to that. I think Australia is lagging behind significantly. I think one of the strengths in the UK has been the NC3Rs, which was initially funded by taking certain amounts of funding from the MRC, which is the Government Research Council, and the BBSRC, which is another Government Research Council, and directing that into this centre for the 3Rs, so it would fund development of model systems and approaches to address the 3Rs. Over the last 18 years, what we see in that, and certainly I stepped down from the grant panel on the board as I left the UK to come here in 2020, we would be getting, on average, every year, in the region of 120, 130 grant applications that were pre-screened to get down to about 45, to which we'd probably fund about 8 or 9. But the quality of the science was significantly high. The quality of the 3Rs was very, very high, and the approach that was being taken. And when I applied and got my funding in 2006, you were getting 10, 15, 20 applications. You then go up to 2019, 2020, and 120, 130 applications. You can see how that has shifted the culture. The NC3Rs works in partnership with the other funding bodies from the UK government. So it's not in isolation, which is a real strength. The Dr. Hadwin Trust, it has strong connections with them, and Frame as well. But the other argument that I would make is that, as researchers, like the evidence I give to you, is that big global pharma want to avoid animal model systems. So I work significantly with large global institutions, and people that have funded my research from GSK, or Philips, Renshaw Limited, out of 3, these are big global institutions. Not one of them really wants to entertain an animal-based project. They might use some of our model systems that may be using some animal material, it could be culture media, it could be antibodies, but they really avoid doing fundamental research in R&D with academic partners on animal model systems. And that's been very clear advice back to me. So you have to adapt, but I think funding is one part of it. Driving funding in a direction is the other critical line, as Wojciech has been mentioning as well. And then I think we can make some big, rapid changes in Australia and take some big leaps.
All right, thank you.
Thank you. Just my first lot of questions to the professors, and either one can jump in. In regards to Australia, obviously we're a federation, so we obviously have the state of New South Wales. We have regulation at the state level primarily. At the national level, we've got the approval mechanism for research and funding. And it seems there could be an argument advanced that in terms of trying to address this issue and progress this matter along, whereby we are focused on using over time less animals in research, that there needs to be a clearer line of sight between the Commonwealth and the state in this whole area of animal experimentation, and that there is a greater synchronicity in the way in which the whole thing operates. At the moment, one is getting a sense that they're almost operating at separate levels. They do intersect at points, but they are in their own domains and operating almost separately. I'm just wondering if you'd like to comment whether you think that's a fair observation. I guess that's my first question. And secondly, how does one drive change where that's less of an issue over time?
I think it's a very fair observation, certainly what I've experienced in the 2 years that I've been here in Australia. So I think you're absolutely right with that observation. How you drive change, I think, a state, and my view would be, if the state of New South Wales can show leadership, and that leadership leads to significant step change in culture or practice, or suddenly you're getting a whole collection of researchers in New South Wales winning highly competitive but excellent grant projects that actually drive things forward, then actually that should, and in fact I'm fairly certain it will send quite a shockwave across the other states. Because here in Victoria, certainly I can imagine my postdoc going, how can I do that? If they could do that in New South Wales, in UCID or UNSW, how can we do that? How can I do that? So you almost send a shockwave across the sector and the biomedical research sector that way. So I think someone to take a step change and leadership, and then to show quite dramatically, and could be done quite quickly, that leadership, that decision making, this focus, whether it be funding, whether it be direction or awards or whatever it may be, is making a step change, then I think it will create other states will want to follow suit, or certainly from a federal point of view, the federal government and your NHMRCs will start to go, okay, we need to take notice of this. I think someone has to plant the flag and show significant leadership, and it should nudge everybody else to go in the same direction. But I say I've only been in Australia for 2 years, but I get a sense that from my 2 years here, someone shows some leadership. Certainly from a young researcher's point of view, they will want to know, how can I do that? How can I get this funding? How can I get my research going in this direction?
Thank you.
Yeah, I certainly agree. Leading by the example and having this leadership would be phenomenal, and showing the example to the other states that we can actually achieve this. And bear in mind that Alistair mentioned and I mentioned about the global companies which can come here and work with us and set up the centers with us to develop these alternative models. They are looking for the researchers like ourselves to work with us, and we have a good examples of these. So definitely an example, funding or developing the centers and initiatives within the state, which hopefully will be picked up later on by the other states and the government as well, especially NHMRC in terms of the funding, would be probably something which we would like to see.
The culture change issue has been raised by other witnesses, and I think it's featuring as part of the narrative that's coming forward with this inquiry. I have to say I'm a little bit surprised, but perhaps maybe I'm just naive, that with respect to the national bodies like the NHMRC and the TGA, at the very later level of research and the determination over which research projects should be given the green light and which are not, those individuals who are involved in that decision making presumably are aware of these developments overseas, particularly with respect to the use of models, as opposed to this almost insistence of the, I think, Professor, the in vivo model as being almost a requisite to green light a research project. What's the cause, if I may ask, of what appears to be an inertia? Because clearly they are aware of these matters that you've both posited today and explained, quite articulately. Is it fair to say there is inertia there and resistance, or is there some, dare I say, ignorance and wanting to turn one's face away from what's happening overseas because we are well used to what we're doing and we don't want to change?
I think there's some inertia. I also think currently the way that grants are reviewed and approved for funding in Australia at the moment is deeply flawed. We have gone away in Australia for having grant panels sitting with designated panel members speaking about a project, where you've got key expertise in a room declaring conflicts, where you can talk about a grant and the rebuttals an applicant might make and the reviewer's comments, and you can have a fair, open discussion about the strengths and weaknesses, to a situation now where grants are just simply reviewed by anonymous reviewers, anonymous to the applicant, and an award is simply on the score that is given. I think you've taken away the critical part of grant funding, which is discussion. And I think currently the situation of grant funding in Australia is deeply flawed. And it means quite simply, money simply follows money. You've had 20 years of funding, you've delivered on that funding, you're likely to get your next grant funded. You're an ECR with a very poor, an emerging track record, depends who you work with, and it depends very much on how lucky you are with your reviewers. And I think that is one of the biggest problems at the moment. And by not having panels sitting, and I think we have to go back to that, you don't get a real focused discussion around the quality of the science, the quality of the project, and could things be done differently. You're simply in the hands of a reviewer. And I review for the NHMRC. And I've said you need to go back to grant panels, and I think it's a very big problem at the moment. I've not seen this anywhere else in the world. NIH in the US meet, on an annual or biannual basis for the grant calls that come out. MRC, NC3Rs, BBSC, EPSLC meet with their calls twice yearly for their grant calls or the grant plan. You're a designated panel member to argue for or against or pick up comments on a grant. So everybody putting an application in gets a fair hearing. And comments like, this model's not particularly good, they should look at a 3Rs model, or they're using 400 animals and the power calculation is way out. If they've been lucky to get some good friends to do their grant, that might just get through on a review. Or you've got someone that kicks a grant because it's you've got a competitor reviewing it. And that's a different argument. But I think the way grant research funding is reviewed now in Australia doesn't help shift the inertia that we see and a step change to try and change things.
And I think you raised a very important question, and sometimes I ask myself the same. And honestly, I don't know what's the answer. Because the same people who are criticizing these alternative models and they work with the animals, they consider using the alternatives. But still, if I apply for funding, they will kill my grant because I use the alternatives. As Alistair mentioned, the system is flawed. But one of the problems is we don't have the international experts on the panel. We don't have the international reviewers. Why? Why these scores, which are completely independent, are not considered here? When I review for Singapore, my score is worth 50% of the total score of the panel. 50%, because I'm independent of all the people there. Here, the country is very small. Everyone knows everyone. But definitely inertia and some resistance. Maybe people become really comfortable and quite conservative, consider something new. And I think if we have some successes, spectacular successes, showing that it works, maybe this would drag more people behind. But definitely what I see at the university, maybe with slightly younger academics, there is definitely a strong push toward the alternatives and the better models. And they give a lot of chances and more opportunities for fundamental discoveries, which you cannot do in the animal models. And the fundamental discovery drives the outcome and drives the commercialization. Without the fundamentals, we cannot do anything.
And with respect to specifically New South Wales, because obviously this is a committee of the New South Wales Parliament, do both professors have any thoughts around possible recommendations that we could make from this inquiry? We're going to produce a report with recommendations, specifically about driving the change and dealing with some of these matters that we've been contemplating. In light of your submissions and your comments today around driving change, some recommendations.
I wonder whether you as a parliament could consider awarding or creating an award, not a research grant, not a research project grant, but a 3Rs award. So the universities in Cardiff, Bristol, Bath and Swansea did that when I was there. It was led by the Welsh Government and predominantly because Swansea and Cardiff drove it. But maybe there could be an element of the New South Wales Parliament having a competitive award, 3Rs award. So you're reviewing projects that have been undertaken in the universities in your state that are addressing the 3Rs challenges, or projects that make a step change, that have a step change towards 3Rs advancement and bringing 3Rs into their research. And that could be working. You could actually target that at even at a young career investigator award, a young researcher coming through, not an experienced professor like myself or Woodcheck. You actually try and do it at an ECR level, even a PhD level. You could probably have to, but you actually, I think, and it would just have to be big funding, but you're rewarding excellence of science and a step change, or something that results in a step change to 3Rs. So a 3Rs award would be something that I think would be my recommendation to come from this.
So I would go probably even further. With the 3 innovation precincts which we have now, why not to establish a innovation precinct in replacement animals or a discovery center and bring together really powerhouse, brains which we have in the state, bring them together and establish something which is world class, the first in the country, probably the first in the world. I'll give you a very simple example. A few years ago, I started working with a company in Korea, Rocket. It's a 3D printing company, focused at certain moment on the bioprinting. Today, they have a factory which is producing the skin replacement after the burns, after injuries, and they have the size of literally one of the buildings, which is a university building, is only production of the skin replacement. Company with 5 years, they moved from literally one bioprinter to hundreds of bioprinters printing the skin. Why we cannot do it here? Why we cannot have a replacement for liver, heart, lung? We have a wonderful research happening across the board. Why not to bring it all together and create a world-class facility, discovery center, or even innovation precinct in this space?
You'll likely find yourself attracting researchers from across the country into New South Wales then, because it's something that will attract half a mile out to new states.
Yeah, especially that you build now next to the new airport, the company which is focusing on the therapeutics.
Yeah.
They would love to test them straight away. Any nutraceutical company, any cosmetic company. Look, I was attending a conference 2 years ago, unfortunately virtually, which was in replacement in Maastricht. 3,000 people attending. Honestly, this is probably the largest conference I've ever been to. And every big company which is producing cosmetics, which is producing shampoos, all those things, they were attending. Johnson & Johnson has a massive unit which is producing artificial skins for testing because they don't want to test on animals. Because we don't want to have a shampoo with the label tested on animals.
Yeah. Well, that brings us to the end. I have to say, we could easily spend another hour and there's many more questions we could ask, but can I thank you all? The fact we didn't get a range of questions to everyone, please, that's no reflection. There's a time pressure. But what will happen, and hopefully you'll be agreeable to this, that there'll be, no doubt, some supplementary questions once you've had a chance to read the Hansard, which, if agreeable, our Secretary will provide to you and invite responses and come back to us. So, on behalf of the committee, thank you very much, first of all, for your submissions and your very valuable testimony today. Thank you.
Thank you.
You're welcome. Thank you very much indeed.
That brings us to our break now, and we're back at quarter past.
Sorry, Chair, I just noticed
I
that Ms. Wallace
apologise.
has her hand up. I don't know if she had something to, or if that was an accident.
So, no, we're finished. So, it's now breaking for lunch, and we're back at quarter past. Thank you very much.
Thank you.
So, welcome to our session this afternoon and our next panel of 3 witnesses. We'll go through the formalities. In fact, Dr. Our first witness doesn't need to. Yeah, so you're appearing. I just double checked. You've already been sworn or affirmed today, so thank you for that. With Dr. Stevens and Dr. Johnson. Oh, thank you. Yes, no, thank you for the right. So, Doctor, no need to be sworn or affirmed again, but could you just state the capacity upon which you're appearing this afternoon?
Yes, Dr. Susan Maastricht, and I am now representing the Australian Veterinary Association as a member of that association and also a member of the Special Interest Group on Animal Welfare.
Thank you very much, Doctor. Dr. Stevens, I'll get you to identify yourself by name, your position title, and who you're representing this afternoon, and swear either an oath or an affirmation, whichever you prefer, the words of which you should have. If not, just let us know. So, Dr. Stevens.
Yes, Dr. Tanya Stevens. I am representing the Australian Veterinary Association, and I am the immediate past president of the Welfare and Ethics Group.
Thank you, Doctor.
I solemnly, sincerely and truly declare and affirm that the evidence now about to be given by me shall be the truth, the whole truth, and nothing but the truth.
Thank you very much, Dr. Stevens. And Dr. Johnson. Same name, position, title, and oath or affirmation.
Okay, Dr. Peter Johnson. I'm a retired veterinarian. I was formerly an inspector with the Animal Welfare Unit of New South Wales Department of Primary Industries. And I solemnly, sincerely and truly declare and affirm that the evidence now about to be given by me shall be the truth, the whole truth, and nothing but the truth.
Thank you very much, and once again, thank you for making your time available. We know you're very busy, and we're looking forward to your evidence this afternoon. So we effectively got 2 representatives from the Australian Welfare and Ethics Committee, members of the AVA, and I understand, Dr. Johnson, you're appearing in your own capacity as a...
That's correct.
Yes. So, Now, just to confirm, and I'm sure you're aware of this, but just to confirm for you anyway, with respect to the AVA's submission, it's submission 242 to the inquiry. It's been received, processed, and stands as a submission to the inquiry, and has been uploaded to the inquiry's webpage. And Dr. Johnson, yours as well. Yours is submission 248. Once again, be processed, uploaded, and stands as a submission to the inquiry. So thank you very much. So those submissions you can take as read, but I'm now going to invite for opening statements because in addition to your submissions, your opening statements, that will open up, I think, nicely for the question from committee members. So on behalf of the Animal Welfare and Ethics Committee members, who are here today, from the AVA, we wanted to share the opening statement, or Dr. Mastrick, you will do that, or?
I'll read the opening.
Okay, thank you, Doctor. Please proceed.
Thanks. My name is Dr. Susan Mastrick, and I'm joined today by Dr. Stania Stevens. We are representatives of the Australian Veterinary Association, the AVA, and we're both members of the AVA's Special Interest Group for Animal Welfare and Ethics. Collectively, we have, for ourselves, more than 40 years of veterinary experience, particularly in this field.
Yeah.
I would like to begin by thanking the committee for the opportunity to contribute to this inquiry and to congratulate the government on its ongoing dedication to improving animal welfare in New South Wales. Of note, the AVA has been in consultation with the government about animal welfare reform process since October 2019, and we have made several submissions throughout the various reviews. The AVA has over 8,500 members made up of veterinarians across Australia working in all areas of animal science, health and welfare. Veterinarians are key experts in animal health and welfare, so it is important for our views to be heard on any animal welfare legislative or policy amendments. A key priority of the AVA is animal welfare, and we acknowledge the ethical dimension of animal welfare in medical research. The AVA has a statement of principles that articulate the ethical basis for all our policies and advocacy on animal welfare matters. These include the AVA Animal Experimentation Policy and our policy on the role of veterinarians in the care and use of animals for scientific purposes. The AVA submission makes several recommendations to this inquiry, particularly focusing on supporting and encouraging a continuing commitment to animal research in New South Wales and the important role that it plays in the knowledge and improvement of health and well-being for humans, animals and or the environment. Calling for a greater emphasis to be placed on the provision of research grants to address the 3 R's of reduction, replacement and refinement and alternatives to research. Highlighting the need for veterinarians to be included in all aspects of animal research to enhance the applications of the 3 R's, foster animal welfare and maximise quality of research. Seeking an AVA representative on the Animal Research Review Panel, ARP. Improving competency training of researchers and involving institutional or facility veterinarians in assessing the competencies of researchers to undertake procedures on animals and advocating for better harmonisation and standardisation across jurisdictions for annual animal abuse statistics and animal research legislation, particularly calling for a uniformed Commonwealth legislation aligned to the Australian Code for the care and use of animals for scientific purposes. We thank you again for the opportunity to present, and we welcome any questions from the committee.
Thank you, Doctor. That's a very good opening statement. It sets up the question very nicely. Dr. Johnson, I invite you to make an opening statement.
Dr. Peter Johnson, retired veterinarian. New South Wales introduced the Animal Research Act regulating the use of animals in scientific research and teaching in 1985, requiring that all institutions and organisations conducting such business must be accredited as animal research establishments. Inspections by external government inspectors commenced in the early 1990s and generally included members of a statutory body, the New South Wales Animal Research Review Panel, appointed from their respective fields of expertise to monitor the application of the legislation, providing independent advice to government and reporting annually to the New South Wales Parliament. Initially, the departmental inspectors were all experienced veterinary graduates recruited from private practice, government research or field veterinary services. Routine inspections were carried out at intervals of approximately 3 to 4 years, or more often where there were problems identified, or particular issues needed further attention. Regular external inspections, augmented by site inspection reports containing conditions and recommendations, provided animal research establishments and their AECs with clear objectives for achieving compliance with legislative requirements and benchmarks against which they could assess progress in the implementation of best practice. To maintain this important monitoring function, inspectors must be selected on the basis of appropriate qualifications. They must be trained, mentored and resourced to ensure rigour in the inspection process. They must be aware of both animal welfare and ethical issues, and in particular advances in research technologies that promote replacement, reduction and refinement in animal-based research in accord with community expectations. The progress made through the monitored self-regulatory and inspection process has been supported by the development and maintenance of evidence-based resources for the respective stakeholders. Maintaining these resources requires the ongoing provision of appropriately qualified and experienced departmental staff and sufficient guaranteed funding supporting the regulatory framework. I thank the committee for the invitation to appear and I wish it well in its deliberations. Thank you.
Thank you, Doctor. And your submission is very detailed, and I'm sure in conjunction with the opening statement there'll be some questions to you. So we have representatives here from the opposition government and crossbench. So if you're agreeable, we'll, between now and the end of the session, share time equally between ourselves and present questions. If there's any questions that you need to take a notice, feel free to do so, but hopefully you can answer much of what was presented to you this afternoon. So, to the Deputy Chair.
Thank you, Chair. I'll start with Dr. Johnson. It's good to see you. It's been a long time. Can I just ask quickly what years you've worked as the vet within the DPI conducting inspections on animal research institutions?
Yes, I started in 1998. I had a brief sojourn between 2000 and 2001 with the Commonwealth, then came back in 2001 and finally retired from the Animal Welfare Unit in 2014. So it was around about 15 years.
So quite a long time then. Did you ever hear about, during that time that you were an inspector under the Act, did you ever hear about research that was potentially being abandoned? Say there was a chief researcher that might have changed roles or moved to another institution. Is that something that happens, or is that something that's somewhat of a concern within the research community?
There would have been perhaps one or two occasions where research that was being undertaken at an establishment might have ended when the person who was responsible for that research transferred to another institution. But I don't particularly recall any specific issues where in midstream of a project, for example, someone left and then there was a problem with animals remaining that were no longer under a protocol. I can't recall that being a particular issue, no.
Okay. And what about university students doing projects? One thing, we've heard from in this inquiry, there's been some concerns that sometimes there are projects, and even though that project in and of itself has a supervisor, obviously if somebody's still as a student, but that some of those projects themselves are just replicas of research with sort of known outcomes. Is that something that you witnessed during that time? And I'm not asking for names or institutions or anything like that, but is that something that was occurring?
I can't honestly recall any specific instances of exact replication of research. I would have to say that in inspecting across a number of institutions, which all of us who were appointed as inspectors did, there would be commonalities in research that was being conducted at different institutions. But generally speaking, there would also be differences in the method in which the research was being conducted, and also some differences in the proposed outcomes. So if you think of situations such as cancer research, there are certainly commonalities between the types of projects that are conducted, but the end results may well be different. So there are certainly instances where there are commonalities, but I wouldn't say that there were
exact replicas.
Situations of exactly the same research being replicated, and not by students. I think my recollection is that in the majority of cases they would be postgraduate students who are engaged in a specific part of a project that is being supervised by somebody within the institution. And the outcome of that particular part of the project would often be their master's thesis or their PhD thesis.
Thank you. One thing that's come up a lot in this inquiry, and I think it's really difficult for people to really sort of get a grasp of the different types of research that happens when you're not sort of really looking in and you have not actually involved in it. And we've heard from, researchers that are talking about, observing wildlife and that falling under research, and then veterinary research, and then there's all these different categories of research. There's been an enormous emphasis on human life-saving research. Considering all of the categories that there are, how much, as a rough estimate of the research is around human life-saving research?
That's a challenging question.
Could I help?
Please.
Only because I happen to have some data that's come from, not from the ABA, but indeed from Sydney University, where we have about 51.5% of our research which is actually biomedical, and the rest is not.
But that's just your university.
Correct. I can only speak to that.
No, thank you. No, I appreciate that, and thank you. But I might just throw back to Dr. Johnson for a more holistic view.
I think there is considerable variation across institutions. So, for example, at our regional universities, there would be substantially less medical research. There would be some, but at the regional universities you would tend to have a lot more in the production animal and wildlife area than you would, say, perhaps at the University of Sydney or the University of New South Wales or Macquarie. But having said that, all of those institutions also have a substantial amount of research that is not human-based. So there's a lot of wildlife research. There's quite a bit of research onto domestic and companion animals as well. So I honestly couldn't give you a figure any different to what Sue has given. I would think it would be roughly equal if you took it across all of the institutions. The other thing to bear in mind is that it's not only universities either, it's commercial companies that are producing vaccines, and it's generally producing vaccines for domestic animals and livestock. There are other companies that are doing environmental research, and that's essentially wildlife-based observations and surveys, that type of thing. So all of that needs to be taken into account as well when you're looking at the overall breakup of what's human what's directed towards human health and what's directed towards a range of other purposes.
Thank you. If I could ask perhaps the representatives from the ABA, in your submission you talk about and we've heard this a lot, that every project that gets approval from the ethics committee needs to have justified that the 3 R's have been addressed, and that the impact on the animals is managed to maximise their welfare. And we keep being told that. But how does that work in practice in terms of, is it really just a tick box? Or is there at what point does the funding bodies decide that actually enough is enough? You need to go and do some more research into alternatives before you can carry on doing this sort of treatment. Is it all sort of very siloed? Or, if you could give me a bit of a colour as to how that process occurs.
Look, I've sat on 4 or 5 animal ethics committees since they were established, so I have a long-term interest. The ones I sit on at the moment, one is very basic research, the Children's Medical Research Institute one. The other one is the Department of Primary Industry, which is mostly wildlife and observational studies and independent research. So I've got some, across the board, I've sat on different committees that do different things. And to be honest with you, I think animal ethics committees work very well. I think the concept of conflict of interest, because somebody's paying for it, even if that comes up, I think that that's really not an issue. I think they're very robust, every committee I've sat on. I don't think they've ever made decisions on justification based on funding. I think they work very well, and I think we have to remember that they were established so that the community could get involved in, have a say in animal research.
And perhaps I could clarify, my question was not about the behaviour or the decisions made by those ethics committees. It's about the processes and procedures that get inputted into those committees. So, for instance, if a research proposal came forward where a particular, a particular model of research was used, where perhaps there could be alternatives to that, but the work had not been done, presumably the ethics committee approves it on the basis of that particular research in that particular context, as opposed to looking and saying, well, more broadly, we should be perhaps refusing these so that people do go and invest more in alternatives. Do you see what I'm saying? Is that...
Yeah, that happens, definitely. Every committee I've sat on, we always talk about alternatives. Have alternatives been considered? And that's not a ticking boxes, I can assure you.
No, but there's also no consideration of if we were to actually prevent this particular research from going ahead because of the use of animals, it might encourage investment in alternative methods.
Depends on the research. I think very much there's always a discussion on alternatives. I've sat on committees where research projects have been knocked back, and there's always a discussion on alternatives. I think it's very difficult to generalise like that, to be honest with you. I think it just depends on the specific situation where you would say, well, what are the alternatives? Well, you do that automatically. Every animal ethics committee does.
Perhaps then I could ask Dr. Johnson, I guess, from that more holistic perspective then, if we have a situation where the sort of commitment of researchers to the 3 R's is done through a grant-by-grant process, what is needed to create that incentive, do you think, to invest or to follow alternative methods that don't rely on animals as much? Do you have a view on that?
I think that the granting process could perhaps be improved to encourage more of those types of studies to be undertaken. You would be aware, I'm sure, that it is a highly competitive process at present. Many of the good grant applications that are put up don't get a run, and a lot of very well-trained people end up not being granted funds to do research, and we lose them. Now, amongst those grants that go up, I'm sure there would be proposals that would be looking at the development of alternatives to animal research. The question is, how much research funding is there available to go around, and how much can be directed towards developing those alternatives? And my answer to you would be, I think there is scope for more resources to be directed into more projects being given a run.
Thank you.
And they would be, at this stage, most of those types of projects would be at the fairly fundamental level of finding out what the best alternative is. So it's a question of whether you're funding some of the pure research that's necessary to get those things up and running, as against some of the applied research that might be looking at the development for treatments, against existing diseases.
Thank you.
Just wanted to ask about the regulatory regime in New South Wales. How would you describe the regulatory regime? Would you say it's robust? I think was the word used before. But in particular, I wanted to know about the enforcement of the regulations that exist, and in particular the inspections as well of the facilities. If you could provide a bit more context in that regard. To all three.
Do you want me? Okay. So it is a very robust and piece of legislation directed towards animal research, which means that from the point of view of people who are undertaking research and animal ethics committees, the DPI, Animal Research Review Panel, they've got a very specific place to go to find out what their obligations are. I think that it needs to be very well resourced for all elements of it to be attended to, particularly the issues around inspections, the inclusion of veterinarians in many of the elements of it, who could provide a fairly, a really significant interpretation of the animal welfare issues. And I know that when Peter was part of that, there were a number of vets that were actually part of the animal welfare unit, and functionally it was fantastic, because when those inspections were conducted, they were very robust and they were done by the department. There's been changes in terms of the allocation of resources, and things have changed within the department, and I think that there's probably something that this group could do that would really effectively help that to return to perhaps the level that it was before. Because let me tell you that I've been in this industry since before 1985, and the improvements that I've seen since the legislation has come into place have been extraordinary. Part of that is because the responsibility for what's going on in the institutions was placed absolutely on the animal ethics committees in the institutions. They've taken those responsibilities incredibly seriously. I remember having some chairs who were extraordinary in being able to say no to very powerful people doing research, and say, no, that's not how you'll do this anymore. Yeah. And it changed things. It really changed things. We haven't finished the journey yet. We've got lots of work to do on the 3Rs and alternatives. There's still lots to be done. But we've made some significant improvements. And this Act has been the means of doing that, because it embedded the National Code. It embedded all of the things that are the best parts of how we do research and get quality outcomes, and looked after the welfare of the animals that we were using. So I think that it's been an interesting journey that we've been on, but a very effective journey. But I do think that there's more resourcing needs to be done if the state system is going to be the one to carry this forward in a really meaningful way. And I'd love to see, with my AVA hat on, I'd love to see more vets doing this and being involved in this. And I know that somebody has said, if you don't mind me saying this, but I know that there's been a question about whether or not vets are trained. How many vets have we got out there to do that? We're doing things. One, vets can do their memberships in animal welfare and indeed in the medicine and management of laboratory animals so that they can become fully equipped to function in this space. They can do external training from overseas. Our intern is currently doing Royal College of Veterinary Scientists endorsed training from Edinburgh University. So there's lots and lots of things that can happen, and that's what we need to also see happen. We'll have university students doing placement with us so that we can give them something around what it is to be a vet working in this sector. And that's one of the things that I think that we do need to do as well. But these are all parts of how we can make this better. And I have to say to you that the universities are very keen to be part of that, and the AVA particularly is very keen to be part of this whole process because we bring unique expertise to the table.
Yeah, I think in terms of the animal ethics committees, so it's a bit hard for us, and sort of similar line of questioning to Ms. Boyd before, where because we've never sat on any or seen them work in practice. How do they work in terms of how often would a research proposal involving animals be rejected, for instance? Like, is it a matter of that? I assume that researchers won't come to an animal ethics committee saying this is what we want to do, and then it's just a simple yes or no. There's probably a lot of back and forth in terms of refining the process. It's not a it's neither a tick the box easy process to get through, but it's also not that no and then it never happens. It's more
in fact, it can be no. No, I'm sorry, you can't do it. No, it's not the right model. No, you need to go back to basics. No, you can't do it. That happens. More commonly it is, no, that's not sufficient. The welfare of the animals is not being addressed. We see the benefit, we see what you're trying to do, but no, that's not what animals, you can't do that for the welfare of the animals. The impact is far too great. There's a lot of back and forth, a lot of requests to go back and revisit, redesign. And we often say we want the animal welfare vets to be involved in the design process to make sure that we get this right. And then it comes back. I think there's some work that could be done that vets could contribute to about the right models. Is this the right model for this type of research? Show me the evidence. Wouldn't it be lovely to have a 3-hour centre? One of their systems was to look at the models that are available. And so that we've got somewhere to go, so that ethics committees have somewhere to go, researchers have got somewhere to go. These are some things that we could look at that would make a huge difference and help the ethics committees and help the researchers. None of the researchers want to be going back and forth. They don't want to get a grant and then be told, sorry, you can't do that work. You'll need to think about something else. And that happens. Because that's how seriously the ethics committees take their role in all of this.
I think, too, just to add to that, research funding in Australia has been going down and down and down over some years. It's so difficult to get a research grant that you're not going to put forward a proposal to go to an animal ethics committee without having done the groundwork. With, considering the difficulty of actually getting a research grant to do that research in the first place. And Australia really needs to better fund research into the 3 Rs, but into basic research as well. I mean, we're really falling behind. There's an enormous loss of talent. If you've been studying, you do a PhD, and you want to do research in Australia, there are very limited options nowadays. And I think what we need to do is to make this process simpler. So we're talking about going to animal ethics committees. You're not going to go there unless you've got your research grant. And at that stage, you've done your homework. And you're going to try and make sure that that research is humane and it's worthwhile. And I think those of us who've been around this area for a long time appreciate that.
Thank you for that. In light of that, however, given that there is such a rigorous, regulatory regime in the state, given that the dollars are seen to be quite tight, and so you would expect that researchers have a plan in place already before they approach not only the application but also the work that goes into the research. And given the outcomes that we've seen, we've already heard the benefits to the populace that the research can deliver. Is there really an issue? Like I'm failing to see exactly where the concern is around this area and the need to, I think, strengthen it. It seems to me that it's almost a self-managing situation, given the current legislation, the restrictions, the ethics bodies in place. That it's very rare to see any issues at all. I fail to see the problem. Can you enlighten me, perhaps, as to where this urgent need for change would be?
It is, in fact, enforced self-regulation. This is the system that was set up, absolutely. And it was, if you look at the National Code, it says every single person who's involved in every project using animals has a personal responsibility. Everyone. Every animal care person, every person in the research project, the Animal Ethics Committee, everybody who's involved has a personal responsibility. And we take that very seriously. It's one of those key things that the code calls for and it's what the legislation calls for. So in a way, we're not saying that the system is broken and needs to be fixed. What we are saying is there are some other elements that need additional funding. I think that, to be frank, the regulator could do with more resources to be able to do their part of it, because I think that there's some real merit in them being able to do what they need to do, and to give more opportunities for veterinarians to participate on ARP in the DPI in this particular sector. I think that would be fantastic. I think if we could have more research dollars around the 3 R's, around the alternatives to research, because believe me, everybody would be really glad if we didn't have to use animals to do research. I know that there's this notion that we all want to do this, we all want to be using animals for research. It's not that. We use animals for research because we do not have an alternative at this time. At this time. What we want is to be able to find those alternatives and implement them, because they, let me tell you, from being completely pragmatic about dollar cost, it would be so much cheaper to use alternatives than it is to use animals. And the cost to the animals themselves, when we're talking about cost benefit, we're talking about the cost to the animals, and that's what we balance up. We don't really care about the dollars at all. It doesn't matter if you've got a $5 million grant, that's lovely. Thank you very much. Bring that into the university or wherever it may be. The truth of the matter is the ethics committee says, what is the impact on the animals? What's the cost to the animals? And balance that against the benefits that you're anticipating you'll get from the research.
I was going to add that the animal ethics committees were established so the community could have a say in experimentation. I think we've got to remember that. Anybody can apply to sit on animal ethics committees.
Yes.
So they're not detached from the real world.
No.
If you want to sit on one, you can get a list where there's vacancies and you can apply. And this is why they were established, and they do play a really important role there for the community to make sure.
They've been key to actually helping the committees to grow and develop and be better at the job that they do. They've been an incredible inclusion. And of course we sort of led the way on this by bringing community members in, and this state led the way with this legislation. So
yeah, I would agree with that. I'd agree with what Susan and Tanya have said. I can tell you that in the early stages of when inspections were being conducted, and I was in after it had been in place for a while, but at the fairly early stage, there certainly were instances where things were detected at various establishments that were clearly outside of the Act, outside of the Code. Did we stop projects at those institutions? Yes, we did. Did I have researchers sitting on the other side of the table from me under caution? Yes, I did. Did we prosecute anyone? Yes, we did. But as time progressed and as ethics committees progressed and as institutions progressed with their approach to implementing the code, as they adopted the evidence-based guidelines that were becoming available, those kinds of instances dropped away dramatically. The number of times that we had to implement conditions on institutions and establishments dropped substantially. And you could see over time that right across the industry there was progress towards a much more directed approach to animal welfare and animal well-being, and a much greater acceptance of the evidence-based guidelines and information that was becoming available. And I think the important thing is that process needs to be properly resourced, and the monitored self-regulation is the way of doing that. I can't speak for how it's been in the last 6 years or so, because I retired in 2014. I understand there have been some changes, as Susan has alluded to, in the way that the licensing and inspection is done. And my deep concern would be that if there are changes to the legislation and changes to the process, that the gains that were made over those early years, over the first 2 decades of the Animal Research Act, that those are not lost.
Thank you very much. Can I, I've just got a couple of questions, just following on from that to elucidate that. But being able to establish, as a committee looking in on this as an inquiry, to see and identify if there has been, I'll use this word colloquially, slippage backwards, compared to where things were when the Act commenced, and that perhaps time immediately thereafter with its implementation and all that went with it. Is there any way in which we can see that the integrity that you've argued is there and was there is still the case these days? And if the answer to that is yes, you believe it is, how do we validate that integrity? Or is your answer you're not sure and that really requires an investigative exercise to establish the integrity of being maintained?
Yeah, my answer to that would be I'm not sure, and I really think one of the things that the committee could look at is what the current situation is. And I'm not sure whether anyone from the department has made a submission or not. But yeah, look, I think it's something that the committee needs to consider carefully, is what the situation is now, whether it has changed from the way it was 6 years ago or 10 years ago. And in the light of the proposal to combine legislations and create a new Act, I think it's very important to be certain that the gains that were made are not lost in that process, or have not been lost, in recent times.
Given that the regulation effectively is the key domain for the state in this area of animal experimentation, Dr. In fact, I think you've all commented on this about the matter of particularly inspections. Are we able to look at the issue of regulation, do you think, and be able to make some judgment about whether or not, and it's a subset of the question I think I just asked, that the regulation has somehow diminished and become less robust, or it's basically the same, or I get the general impression that some witnesses, I'm not going to sort of say yourselves, but others have presented, have been at least implicitly suggesting that there has been a diminution perhaps in the robustness of the regulation in New South Wales. And I'm trying to tease that out to establish whether, as experts, you would come to that conclusion from your own observations, or say more work needs to be done to establish that.
I don't believe that there's been a diminishing in the rigour with which the research is being reviewed by ethics committees, or in fact monitored by institutions. But there's a lot, and the code calls for an yearly external review to be conducted, and we've certainly, we see that happening. That used to be the responsibility of the department, and they would go in and and in a way that meant it brought the regulator in to see what was going on. And this is to say nothing about the competency of the external people who are conducting that now. Sure. But there was something quite meaningful about the fact that a regulator and the people from that group were coming in to do those inspections. And of course we have had that, but we haven't had that all the time, and it's been particularly challenging over the last 2.5 years of COVID. But I do, we would certainly not lean back from the notion of the regulator and a group of people, particularly qualified, that Peter described the competencies of the people who undertake that review coming in and doing it. We would certainly not be stepping back from that, and we've had some very, very skilled people come in to do the reviews. But in a way, it gives the legislation strength if the regulator actually plays a role in that and is able to do that more readily, and we have really highly competent people who are part of that process. I think that's saying, yes, we are going in and we are looking, and we're going into all of those institutions and we're checking what's going on, because how else are you going to do this?
Yeah. Just on the question of the enhanced and improved role of vets, and what we've been discussing this afternoon, just from your own point of view, what could we do as a committee producing some recommendations to this inquiry with its terms of reference be suggesting or proposing to the government to enhance the role in terms of presumably the number and the general availability of vets to be involved in this animal research work that's currently underway in New South Wales?
I think if we could engage more vets, give them opportunities to do the learning in this space, because it's not a large part of what a vet would actually receive as part of their undergraduate training. So making sure that that's available to them, supporting them through that, allowing them to do memberships, doing all those are the sorts of things that could be done. I really would love to see that happening. Doing in-house training, that used to happen all the time, and it's that's the sort of stuff that would be fantastic. Making opportunities for that to happen, going to all the vet schools and saying, what else can we do? How can we make that happen more readily, so that this becomes part and parcel of what veterinarians actually can contribute to this space? Because that's what our training is. Our training is about the animals, we're understanding animals, all about them. They're not just their health and well-being, but the environment in which their whole of life is actually given the greatest merit and benefit. I mean, it's just something that we carry with us. It's part of our integral training that we bring, and I think that it would be such a unique opportunity for New South Wales, who's led the way in this space. It would be lovely to see New South Wales do something really meaningful to provide opportunities for veterinarians to receive further training and to get employment in areas such as this.
So just in regards to this opening up of more opportunity for vets, perhaps take it on notice, but be very interested to see what your suggestions might be in terms of, is there postgraduate study, or are there units they do within their actual undergraduate degree which actually provide opportunity? Just the range of things that perhaps we could be looking at as potential recommendations to put forward, subject to how we go.
I think they're not really exposed to it. too much. And of course, the curriculum's pretty crowded.
Yeah.
Yes.
But I think unless they're interested in research, they don't quite understand that they can get involved in this without actually being researchers.
Yes.
Now, I mentor new graduates, and I suggested to one that they might sit on an animal ethics committee, and they weren't aware too much of what they do.
Mm-hmm.
So I think there's also this lack of understanding of you can make a contribution without actually being a researcher.
Yeah.
So I think it comes down to education and not just postgraduate training. I think the universities have a role here to promote the idea of vets getting involved. One of the problems is, of course, the veterinary profession, the same as the medical profession, has become a bit splintered, as it were, and specialised. Yeah. And I think that is that to me has become a big issue. When I graduated, I could sort of think vets can do anything, and you could go out and do community work. I think that's changed a little bit, and I think we've become a little bit too much specialised. Mm-hmm. But I think it goes back to education. I think giving students the opportunity to think outside the square.
Yeah.
Really.
Well, thank you very much. We've just gone past the time, but listen, it's been most valuable to be able to ask you questions that elucidate the content of your respective submissions. I know there's going to be some supplementary questions arising from having the opportunity to read Hansard once that becomes available. So if you'd be agreeable with those supplementary questions, our Secretary will liaise with you. But once again, thank you very much for making yourself available.
Thank you.
Thank you.
Thank you, Doctor, coming back a second time.
Pleasure.
Okay. Good afternoon, Professor Knight. How are you?
Good morning. I am okay, thank you.
Oh, good.
Calling in from Europe here.
What country are you in?
I'm in France at the moment, and it's awfully early in the morning. I hope your afternoon is going well there.
Well, can I commence by thanking you very much for getting up. Obviously, I don't know whether you've had any sleep overnight, but thank you so much, and we do appreciate you making yourself available, because I know you're going to be able to make an important contribution to our inquiry, so thank you very much. Now, we just need to get through...
It's a pleasure, Mr. John. Thank you for the invitation.
Thank you, Professor. Now, we'll just get through the formality, which is to have you identify your name, your position title. I understand that you're this afternoon representing yourself as a private individual to the inquiry, and we'll come to your submission in a moment, and then swear either the oath or the affirmation, whichever you prefer, the words of which should have been provided to you, and if not, just put your hand up. So I'll get you to just proceed with that, please.
So my name is Andrew Knight. I'm a Professor of Animal Welfare and Ethics, and my affirmation is that I solemnly, sincerely and truly declare and affirm that the evidence now about to be given by me shall be the truth, the whole truth, and nothing but the truth.
Thank you very much, Professor. Just returning to the subject of your submission, and thank you very much. It stands as submission number 250 to the inquiry. It's been received, duly processed, and uploaded onto the inquiry's webpage, and you can take it as read with respect to the members of the committee who'll be wanting to ask you some questions shortly. But before that, can I invite you to, if you wish to do so, to make an opening statement to get the ball rolling, and then once that's done, we'll then move into the questioning, if that's agreeable with you.
Yes, thank you. So I'm obviously a veterinary professor of animal welfare and ethics, and I studied animal research extensively within my doctorate and since time. And I think it's very clear to anyone that does this that there's a very sizable and consistent body of evidence. In fact, the systematic reviews, which is a type of scientific study that aims to appraise all of the evidence in a particular research area and do so very thoroughly and systematically. So there's a very sizable body of this evidence looking at how useful has animal research been for advancing key human outcomes, in particular the advancement of human healthcare. And it's clear that unfortunately, although there have been some times where there are positive links, it's clear that unfortunately overall the efficiency of this research method for attempting to achieve this social objective has been very low. And there's a well-identified set of reasons that seem to be limiting the utility of animal research for advancing human healthcare. So in light of these, as I've said in my publications, a range of measures are strongly warranted to increase the scientific quality of animal research, the reliability of subsequent results, and to overcome some of the barriers that currently prevent reliable extrapolation to human outcomes. Compliance with each of the 3 R's and other best practice standards during the design of animal research, the conduct and reporting of animal research must become mandatory. Such standards should cover the animal sourcing, housing, environmental enrichment, socialisation opportunities, appropriate use of anaesthetics and painkillers, handling, non-invasive endpoints, and a range of good measures designed to minimise sources of conscious and unconscious bias and to ensure scientific quality and safeguard animal welfare. Compliance with standards such as these should be a necessary condition for securing research funding and ethical approval, licensing of researchers, facilities and experimental protocols, and publication of subsequent results in scientific journals. Implementation of these sorts of measures would require cooperation and coordination between researchers, regulators, licensing bodies, ethical review committees, funding bodies, journals and authors, and of course the necessary willingness among all parties to change. But given how sizable and consistent the evidence in this area is, showing us that the current culture of how animal research is designed, conducted and reported has produced such a low level of benefits in light of the resources that have been invested in it, a paradigm change is clearly warranted. Instead of uncritically assuming the benefits of this research, we should be subjecting it to much more rigorous and critical evaluation. Where animal research continues to persist, a broad range of measures must be implemented to substantially improve its scientific quality and compliance with the 3 R's and to maximise the reliability of subsequent results. When such research fails to meet the harm-benefit standard expected by society, it should cease and the resources consumed by it should be directed into more promising and justifiable fields of research and healthcare.
Well, thank you. That's a very helpful opening statement, which sits very nicely with what is a very quality submission that's been made. So thank you very much, Professor. We have representatives at the table from opposition government and crossbench. And if you're agreeable, between now and the end of this session, we will spend time moving questions around between the 3 groups. So are you agreeable to that arrangement?
Yes, of course. Thank you.
Thank you, Professor. Start with the Deputy Chair, the Honourable Emma Hurst.
Thank you, Chair. And thank you, Professor Knight. One argument in this inquiry has been the continued need for experimentation on primates. Would you agree that primates should still be used for experimental purposes? And if not, why would you propose that they shouldn't be used for further experimentation?
Yeah, primates are particularly concerning when used in animal research for a couple of reasons. One is that they have particularly advanced cognitive, psychological, social characteristics, which mean that it is particularly problematic when they are confined in relatively small environments where they're not able to exercise their full natural behavioural repertoire. The disruption of their social networks is also important. These factors create a particular set of stresses which may be less commonly experienced by other species. One characteristic, for example, is their long memories. They've got the ability to remember that certain people, as in laboratory technicians, certainly certain tools and certain procedures may be associated with pain and stress, and to anticipate those occurring in the future. So these sorts of characteristics may place primates at particular risk of suffering when subjected to invasive research and confined within these environments. Should they ever be subjected to experimentation? I mean, certainly some level of research is justifiable. Non-invasive, observational or behavioural research in sanctuaries or free-living populations is obviously an example of that. You could argue that if there was a primate that needed some kind of veterinary medical intervention, a treatment or a surgical intervention or a pharmacological intervention, existing treatment modalities were unlikely to work. There was reason to believe that a new test intervention might work. Then in a situation like that, if the need was great enough, it might well justify such research. In general, though, unfortunately, the studies, including the systematic reviews that I've actually conducted and published myself, have shown that research on primates for the normal purposes, which is attempting to test drugs and vaccines and so on intended for human patients, unfortunately is just not sufficiently reliable or predictive enough for human beings to be of very much use. One of the reasons for that is perhaps simply logistical. The accessibility to this species, because of their cost, because of their relatively low numbers and difficulty gaining access to them for research, is so limited that the number of animals used in this research is relatively small compared to sometimes rodents, for example. And this means that it's often not possible to reliably extrapolate results from very small samples to much larger groups for the same species, and certainly to other species as well. So unfortunately, there are a well understood set of reasons explaining what the evidence clearly indicates, which is that this research has not been reliably and consistently useful in helping us to advance important human healthcare objectives. So I don't think that kind of research should be proceeding, particularly in light of the unusually high animal welfare impacts and also the costs of that research in terms of financial resources, research resources, skilled personnel hours.
Thank you. There's evidence that in New South Wales, and I'm assuming here that maybe you know what the forced swim test is, but if you don't, please let me know. But in New South Wales, there was evidence that there were forced swim tests of up to 90 minutes, where animals were forced to swim for 90 minutes, whereas I know a lot of these forced swim tests are 45 seconds to 90 seconds. What are the welfare impacts of a 90-minute forced swim test? And do you think that with those animal welfare impacts that experiment should have been approved in New South Wales?
That's a very interesting question to me, because with scientific colleagues we actually published a systematic review of the forced swim test in a scientific journal within the last year. And we looked not only at the animal welfare aspects, but in particular the scientific benefits accruing from the forced swim test. And one of the major reasons that this test is conducted is to study depression, major depressive disorder, one of the top human psychological disorders worldwide, affecting many millions of people. And the idea is that by putting these rodents into water with no ability to support themselves, eventually they will give up and enter a state which is thought to be similar to learned helplessness, which simulates aspects of major depressive disorder. What we found by studying forced swim tests published in the scientific literature was that unfortunately the results of this research was almost never actually cited by clinically focused papers aimed at tackling major depressive disorder and other important psychiatric disorders in human beings. So this research, if it was cited, tended to be cited by other animal research, but human clinically focused research was continuing almost in a parallel world without making very much use of this forced swim test data whatsoever. So it's not achieving its intended objectives scientifically in terms of contributing to the combating of those major human diseases. It has a particularly severe animal welfare impact. So based upon the harm-benefit analysis that this research is meant to be complying with, it should not be proceeding.
Thank you.
Thank you. So just following on from that question then, on that basis, do you think that there is any justification for using the forced swim test? Should it be outlawed?
Yes, I do, and indeed that's what we called for in our recent published scientific study of the forced swim test, after examining, as I say, after conducting a systematic review, looking at how often this kind of research does go on to produce human clinical benefits, and looking at the alternatives, looking at the level of contribution of those compared to forced swim tests and so on, we concluded that it clearly did not meet the harm-benefit analysis that animal research is required to meet, and is expected to meet by society at large. Forced swim test is a particularly, I think, egregious example of animal research that has particularly high animal welfare impacts and a low level of actually achieving the benefits that are so often hoped for and so often claimed actually in attempts to justify this kind of research. And that's usually made to grant bodies that are funding research, funding researchers. There are various justifications made, but unfortunately the evidence does not stack up in this particular case. So the research rarely, if ever, produces those kinds of benefits. So yes, I think it should not be continued.
So I guess in a similar vein, the other thing that we've been hearing a lot of evidence about is smoking towers with mice and the nose apparatus and the forced inhalation of smoke for a prolonged period of time. Is that something you're familiar with? And again, do you think that the supposed benefits outweigh the animal welfare concerns when it comes to those smoking towers?
Yeah, I would have the same kind of concerns that we have identified in systematic reviews of many fields of invasive animal research, and they are concerns about aspects which limit the productivity of this research for wider populations, both of the same species, but also for other species, notably human beings. Unfortunately, the problems are very prevalent with this kind of research, and they concern, they're very numerous, but they concern things such as lack of statistical justification for sample sizes, lack of sufficient sample sizes, and both of these mean that you can conduct the experiment, but it doesn't mean that you'll necessarily be able to reliably predict outcomes in the entire species of mice, for example, or primates, or whatever the species may be. So that's a problem. Lack of randomisation, of allocation of animals to different treatment groups and control groups, and that's something that is absolutely standard within human clinical trials, because failure to do that is known to distort the outcomes of the research. Lack of blinding during the assessment of the research results, so that means the researcher should not be aware of whether the animal being studied has undergone a test intervention or has been in a control group. And it's well known also from human clinically focused research that failure to do that also distorts scientific outcomes. So, I'd be taking at this kind of research and looking at the prevalence of those kinds of errors. We know from conducting large-scale studies, systematic reviews, that these kind of errors unfortunately are very prevalent, and these are some of the reasons, some of the key reasons why the results are not very reliable. They don't tend to reliably predict the same species and certainly other species as well. So, the level of benefits unfortunately low. The reasons why that occurs are well understood and well studied. And in light of that, should this research be proceeding? Well, when it's particularly invasive and when the animal welfare impacts are particularly high, as they are in smoking towers and forced swim tests, then the research needs to be especially justifiable in order to be proceeding. But it needs to be particularly good scientific quality, and the likelihood of benefits should be very high in order for there to even be a case. And that's not true, unfortunately. Those conditions are not met by this kind of research. So it should not, should not be proceeding.
Thank you. One of the themes of this inquiry has been the sort of lack of funding for alternatives in Australia. I understand in the EU they're working towards a phase out of the use of animals in medical research. Do you think that sort of phase out timeframe or plan is necessary in order to properly incentivise adequate funding and focus on research alternatives?
Yes, I think that's certainly very beneficial. Obviously, there is a long history of animal research. There is a lot of scientific culture which has been focused around setting up research institutes, getting in equipment and facilities that have used invasive animal research, and it takes time to transition those to alternative methods. But it's absolutely something we should be doing. Within Europe, there are the National Centres for Alternatives in EU member states, which are government funded, which provide grants for people working on the development of non-animal alternatives, provide opportunities to present their work at conferences, and help to provide career pathways for scientists working in these areas, and all that's necessary to help to generate a new scientific culture focused around alternatives. And from a scientific perspective, perhaps the most important thing is simply use of the research methods that are most likely to predict human outcomes, if the objective is to advance human healthcare, or to accurately answer research questions. And it happens to be the case that when you use things like human cell cultures in microfluidic circuitry systems, you have different cells from different organs connected via tiny microfluidic circulatory systems, you predict human outcomes better than if you use rodents or primates, because you're actually using human cells. So from a scientific perspective, it makes sense to be using the research method that is most appropriate to your research questions, and most likely to answer the research questions that you have. And I think it's, if Australia wants to produce a world-leading research culture in this area, it needs, I think, to be resourcing that. It needs to be encouraging researchers that want to develop those methods and to use them, funding that research, setting up centres, providing conferences for people to present that work, and helping to support career pathways for researchers who are keen to push the envelope forward in these sorts of research areas.
Thank you.
Thank you. Thank you, Professor Knight, for appearing today. Do you think it's fair to say that every dollar that would go towards funding new RRR solutions potentially comes out of the budgets that would go towards funding cancer research, say diabetes, heart disease, all the issues that Australians are living with day to day now, would likely have to take a back seat in order to have those research dollars realised?
Yeah, thank you for the question. No, unfortunately, I wouldn't say it's fair to ask that. And those key public health concerns are so important, affect so many people. I think the important thing from a public health perspective, and also scientifically, is to pick the research methods that are most likely to produce benefits. So if we have a budget for the treatment of cancer, for example, I think it's important to spend that budget in a way that's most likely to produce outcomes that are actually going to help human cancer sufferers. And so it's very clear from the systematic reviews that have scientifically studied how often does invasive research produce those sorts of public healthcare benefits. It's very clear that although there are some benefits and some links, the level is unfortunately much lower than is usually hoped for, and often what is expected, and is in fact very low overall. So it's a method that we can use, but the efficiency of this method or this tool for achieving that public healthcare objective is very, very poor. So if we want to spend our budget more effectively, we're certainly not taking from that budget, we're just seeking to spend that budget more effectively, then we want to be using methods which are much more likely to be predictive for human patients and likely to produce those benefits. And myself and other researchers have published papers looking at the effectiveness of different research methods for achieving these sorts of objectives. And human clinically focused research is currently underfunded in many areas and believed to be more likely to produce those objectives. So it's not a matter of taking from those budgets, it's a matter of discontinuing the wastage of much of those budgets and redirecting it to more effective research methods, more likely to produce those public healthcare benefits.
The evidence you've just given is in total contradiction to the evidence that we heard earlier this morning from Dr. Ted Raw, who was talking about the requirement for animal research to aid children who are undergoing cancer treatments. And I asked him specifically about what other methods might be able to be employed to supplement or replace the animal research that's undergone in his clinics. And he said that it wasn't possible, that it was effectively only to be able to trial and error on the child would be the only other method that he could see that would work. So in that instance, I think that there's been a clear demonstration that there isn't another pathway. Obviously, there's a thought that we need to be doing more in researching triple R, but if we weren't able to do that sort of research, then does that not have an impact on those patients?
Yes, that's a good example, and that's exactly the kind of point I was making, that we often have expectations of animal research and hopes that animal research will produce certain kinds of benefits. The trouble is that when we conduct the systematic reviews to examine very thoroughly the evidence, how often does this research method produce those kinds of benefits? Every systematic review that's ever been published in any field of animal research has demonstrated that the majority of the research in that field has a very common set of scientific flaws, which seem to be limiting the reproducibility and the reliability of this research, and consequently the ability to produce the hoped-for benefits in terms of human healthcare advancements. So the trouble, I think, with the sorts of claims that you heard before is that they are claims that are talking about the benefits of a certain kind of research without systematically and carefully and rigorously evaluating that research in a sort of study that then is published in a good quality scientific journal. And that is a systematic review. And the problem with a lot of animal research is that it's been uncritically assuming benefits, and it's not been undergoing that kind of scrutiny. And researchers such as the gentleman you heard from before and other research fields should be subjecting their own research to that kind of scrutiny. It should be undergoing systematic reviews so they can get an accurate impression of how often does that research produce hoped-for benefits, and also what are the barriers that are holding it back from doing things better, from becoming a more efficient research method.
Sorry, go on.
Very often we hear claims and anecdotes about the importance of certain kind of research for advancing human healthcare, such as in that particular case, as a means to try to justify research funding or to seek to obtain ethical approval, or perhaps in your case to speak to an expert committee assessing this issue. But what we're not seeing in those sorts of narratives is evidence from systematic reviews and other thorough scientific studies that looked at how often does that research actually produce those benefits, and what is the level of compliance of that research with best practice standards designed to minimise scientific errors creeping into the research. And unfortunately, we know whenever we do this, there's not yet been a systematic review published that's found that the majority of the research in an area of animal research complies with good scientific standards. There are a very well understood common set of problems which were identified many, many years ago in human clinical research and have been effectively weeded out of human clinical trials to make those more predictive for human populations, but unfortunately still very persistent within animal research because the culture has not improved. So, you hear those stories that have been given to us many times, but they operate, they make the assumption that that kind of research is highly efficient, highly useful for producing its outcomes, and it's the only way that those outcomes can be achieved. Whereas the reality when you study it is that the research is normally, unfortunately, scientific errors are very common within that research. The efficiency for producing those outcomes is very low. And indeed, methods using things like human cell cultures, simulations, epidemiological studies are more likely to produce benefits for actual human beings.
Professor Knight, if it was your child, would you want the animal research to be trialled for cancer treatments for your child, or would you be prepared that that not be conducted in order to satisfy perhaps an objective, to further triple R treatment? Because I know in my case, I would absolutely want what's proven for my children.
Yeah, again, a good question. Look, if it was my child, I would be, I think, even more motivated to try to ensure that the research method chosen was most likely to be predictive for my child, and I wouldn't want financial resources, skilled personnel hours, other scientific resources, resources being essentially misdirected into fields of research that are unlikely to be very effective or efficient in producing what I needed. I'd be wanting the use of methods that are most likely to be predictive for my child, and that unfortunately is not animal research, certainly as a general rule. It's certainly not studying cancer in rodents or even in primates, I'm afraid.
We'll agree to disagree. Thank you, Chair.
Thank you, West. Doctor, I've just got a few questions myself. Sorry, Professor. Towards the end of your opening statement, you may have it in front of you. I didn't write this down fast enough, but I thought it was a very salient point. You spoke about the matter of paradigm change, and then you actually went through a number of items: funding, regulation, research, journals. I think there might have been 2 or 3 others, but they essentially were sort of, could I describe it this way, the sort of research universe as a totality. Can I ask you, in regards to that, and it's my sort of description as a universe, but would you agree that what we've got to try and do is move forward on all of those fronts concurrently to push this thing along and drive it, as opposed to focusing on any particular one? Or do you have a view that there's a particular element that we should be giving particular focus to?
Absolutely. So, as I said, it's with respect to the last speaker, it's not just my opinion. What I'm giving is the results of systematic reviews, which are numerous and published in scientific journals, and they have identified very commonly a certain set of flaws within animal research. We're talking about the design of the research, how it is conducted, and also how it is reported thereafter in scientific journals. This is certainly much more than just my opinion. And in order to address that, we need to change fundamentally how we assess our research and how we conduct it. We need to be subjecting it to scientific scrutiny. Animal research is not somehow exempt from the need to be subjected to scientific scrutiny, particularly when it consumes so many research resources that are unavailable to other research fields. We need to be subjecting it to scrutiny, and we need to be changing the way that we conduct animal research. And I mentioned a range of measures that need to be undertaken to change how animal research is designed, conducted and reported. And if we did those things, theoretically we would be able to eliminate many of the problems that are so prevalent within animal research, which give it such poor reproducibility when you conduct the same experiment in a different lab. You very often, unfortunately, get different results. And also the poor productivity of that research for wider populations. So it wouldn't fully remove the barriers to extrapolation to human beings, but it would certainly improve the quality of research and the welfare impacts as well. Now, in order to achieve that, I said that there needs to be cooperation and coordination between all relevant parties. So that's animal researchers themselves, regulators of research, bodies that license research, and also facilities. So we're talking about laboratories, ethical committees, funding bodies, journals, and others. So everybody involved in the field. We're talking about changing a field of scientific research. We're talking about cultural change. The need for this is very clear if you look at the evidence in this area. The same needs were identified and recognised and addressed within human clinical research some decades ago. The same kind of scientific problems were found to be highly prevalent. The same kind of problems it was causing with the reliability of the research results, the inability to accurately predict outcomes in wider populations was widely recognised, and in order to fix that up, the guidelines were published that human clinical trials needed to comply with moving forwards, and appropriate measures were taken, such as the establishment of clinical trial registries so that future researchers could look at all the existing and past research in an area and then not miss key research. So these things were done in human clinically focused research decades ago, and they've been effective. Now, in recent times, we've had the publication of similar guidelines within the animal research field, following wide recognition that the same kinds of problems are highly prevalent within animal research. But unfortunately, there has not been the cultural change. Follow-up studies have taken place, have been published in scientific journals, demonstrating that even though guidelines have been put out there, requiring research to be conducted to a much better standard, unfortunately, the reality is that those problems and errors have persisted, and there has not been a widespread change the way that there was in human clinically focused research, and accordingly, the efficiency of this research method for achieving these key social objectives, such as the advancement of human healthcare, remains very low. So there does need to be a change in the culture of science in this area. Animal research should not be immune from scrutiny, it should not be immune from change and improvement, and in order to achieve that, all those different bodies need to work together and have the willingness to change. That's not been the case to date, unfortunately.
Well, can I be a bit of a devil's advocate and invite you to be as frank as you possibly can or wish to be about what is, in your considered opinion, Professor, the cause or at the root of this inertia? So can I just say,
sorry.
I make the sort of assessment that from the witnesses that have come along and given evidence to the inquiry thus far and the submissions we've read and what have you, we're not talking about unintelligent people, people who, dare I say, are not very fine minds in their particular fields, and they can presumably see, if we could use this sort of phrase, what you can see if they're looking like you're looking. But the question is, to invite the response is, why aren't they not looking like you have looked? I'll put it that way.
Yes. I guess one reason is they have not been compelled to do so. There's also not time to do so. Researchers are extremely busy people. Well, it's been said that animal researchers are sometimes scientific giants, but ethical infants. It's also true, I would say, that they may be very intelligent and expert in a particular area, but unfortunately not in the crucial area of subjecting this research to the kind of scrutiny that systematic reviews subject the research to. There's been, surprisingly, this research is proceeding somewhat unscientifically in the sense that it's not been subjecting itself to scientific scrutiny adequately. There's been uncritical assumptions that this research is likely to produce the sorts of key societal benefits that have been so often claimed in applications to grant committees, to ethical review bodies, and even to journals. And that needs to stop. This community needs to recognise that even though they may have great deal of expertise in their own very narrow area, that is not a substitute for being willing to scrutinise the field with respect to how often is it producing those kinds of hoped-for benefits. So we're talking about things like prospective and retrospective analyses of the research to see how often it went on to produce the kind of benefits it previously hoped for. And there's a list of around about 20 or so key scientific flaws that research in this area often known to run afoul of. How much of the research is suffering from those flaws? What can be done to change that? People are just not thinking about these aspects. They may be brilliant in a very narrow field, but they're actually not being sufficiently scientific enough. The scientific thing to do is not to uncritically make assumptions. It is to critically scrutinise and to question and identify flaws, and then the responsible thing to do is to try to fix those flaws. And that's not been happening. It's not been happening, I suppose, because they're incredibly busy people, because it's always been done that way, and because they have not been compelled to change. There was stronger steps were taken in the human clinically focused field decades ago. You could no longer publish your research in key journals in your field if your research did not measure up to standard, if it did not meet the required standards. That's compelling change. You couldn't necessarily get grants for your research if it did not meet the necessary standards. This compels change. These kinds of steps have not been taken, unfortunately, in the animal research field, and so change has not occurred.
Well, it's a shame we don't have more time. It'd be great to be able to follow that up with further questions. But nevertheless, time's got to us. So we will, no doubt, certainly I will have it. I suspect other members may have some supplementary questions to follow up on what's already been asked, and I'd like to follow up on that particular line of questioning I had just to end it off. But on behalf of the committee, can I first of all thank you once again for getting up so early. I understand you may be on a holiday over there with your family, so if that is the case, may you and the family have a wonderful break and a bit of a rest. But once again, thank you so much for making yourself available, and we appreciate very much the evidence you've been able to provide the committee. Thank you.
Thank you very much indeed. Best of luck for your deliberations. It's always a pleasure to talk to Australians. Thank you.
Thank you, Professor. Okay, we now have a short break for 15 minutes. Well, thereabouts, back at 3 o'clock. Again, thanks to all those participating this afternoon, and particularly those present now with our next session. Now we have three of our witnesses joining us by video conference, and Dr. Steele's in the room. So I've got a list here just in the order that people have been listed. So I'll work through that list in that order. You don't have it in front of you, that's okay. I'll call your name out as we go. And for each of you, get you to state your name, your position title with the organisation you're with, and then swear either an oath or take an affirmation, whichever you choose, the words of which should be provided to you. If for some reason you don't have them, put your hand up and we'll provide them to you. So we'll get things underway. So we'll start with Ms. Torrance.
Yes, so I'm Prue Torrance, Executive Director, Research Quality and Priorities with the National Health and Medical Research Council. I solemnly, sincerely and truly declare and affirm that the evidence now about to be given by me shall be the truth, the whole truth, and nothing but the truth.
Thanks, Ms. Torrance. Ms. Bates.
Good afternoon. I'm Mary Bate, Assistant Director, Ethics and Integrity Section at National Health and Medical Research Council. I solemnly, sincerely and truly declare and affirm that the evidence now about to be given by me shall be the truth, the whole truth and nothing but the truth. Thank you.
Thank you, Ms. Bates. Ms. Peckin.
Hello, my name is Kathy Pitkin. I'm the Executive Manager, Social Responsibility and Ethics. I have responsibility for the Ethics and Integrity team at CSIRO, and I'm also a board member of ANSCART. I solemnly, sincerely and truly declare and affirm that the evidence now about to be given by me shall be the truth, the whole truth and nothing but the truth.
Thank you very much. And Dr. Steele.
I'm Jack Steele. I'm the Director of Science Impact and Policy in CSIRO. And I swear that the evidence now about to be given by me shall be the truth, the whole truth and nothing but the truth, so help me God.
Thank you, Dr. Steele. Can I, first of all, thank and acknowledge the respective organisations for your submissions. With the NHMRC, your submission's been received, processed, and stands as submission 238 to the inquiry, and it's been uploaded to the inquiry's webpage. And with respect to the CSIRO, your submission has been equally received, processed, and stands as a submission uploaded to the inquiry's webpage, and it stands as submission 237. So thank you very much for those contributions. What I'll do is invite both organisations, and I presume one person has been delegated or agreed to make the opening statement. That's my assumption, unless there is a plan for everyone to make an opening statement. Is it a plan for each to have one person make the opening statement? Is that a fair?
Yes.
Okay. Well, we'll start then with the NHMRC, the opening statement, then we'll defer to the CSIRO, and once that's done, we'll open up for questioning. But let's get the opening statements done first. So, who's going to make it for the NHMRC?
So for NHMRC, that would be me, Prue Torrance.
Thanks, Prue. Please proceed.
So thank you for the opportunity to present to this committee. I'm joining today from the Noonawool land. I pay my respect to elders past, present and emerging. The National Health and Medical Research Council is the Australian Government's lead agency for supporting health and medical research in Australia. NHMRC's legislative functions under the National Health and Medical Research Council Act of 1992 are to fund health and medical research and training, to issue guidelines and advice on improving health outcomes through prevention, diagnosis and treatment of disease and the provision of healthcare, and to promote the highest standards of ethics and integrity in health and medical research. NHMRC provides national leadership on the use of animals for scientific purposes through the Australian Code for the Care and Use of Animals for Scientific Purposes, which I will hereafter call the Code anytime I need to mention it. This national leadership and provision of ethical guidelines on the use of animals is a long-standing role which NHMRC has undertaken since 1969, but it is in addition to our legislative responsibilities for improving human health. The current code is endorsed by NHMRC, by the Australian Research Council, by the Commonwealth Scientific and Industrial Research Organisation and by Universities Australia, and the code is incorporated in legislation in all states and territories, including in New South Wales. The code outlines principles for the care and use of animals and the responsibilities of all those involved with the use of animals for scientific purposes. Any research involving animals must be approved by an Animal Ethics Committee before it begins, but the day-to-day operation of these ethics committees is the responsibility of research institutions such as universities who maintain those committees. NHMRC has also produced guidance for best practice in animal-based studies, and as a funder of health and medical research, NHMRC requires that any use of animals in any NHMRC-funded research complies with all relevant legislation, the code and NHMRC guidelines. Thank you.
Thank you very much, Ms. Torrance. Dr. Steele, over to you.
This will complement that opening statement.
Thank you.
So I thank you for the opportunity to attend the committee. I'd like to acknowledge the traditional custodians of the lands on which we're attending this meeting and pay respects to their elders past and present. So the CSIRO submission to the committee is basically set out in terms of the specific terms of reference of the committee. All of CSIRO's research activities are under a strong policy structure and also audited by CSIRO's internal audit process, which is quite active about looking at our science integrity performance. The governance structure includes our science and delivery policy and our code of conduct, and they adopt the code using the short form of the word that Ms. Torrance has used. Also, the best practice methodology that Ms. Torrance referred to as well, and also the NHMRC Australian Code for the Responsible Conduct of Research. So we're on a very common footing in terms of policy positioning. Ms Pitkin's team supports the operation of 4 animal ethics committees in different states of Australia that review and approve the use of animals in research. In New South Wales, our research involving animals is conducted at Armidale and is largely into livestock management, sheep and cattle. Really importantly, Ms Pitkin's team provides advice to researchers in preparing their proposals for committee consideration, including and emphasising implementation of the 3 Rs. That team also supports the Animal Ethics Committee members, including access by those committee members to ongoing training. And each year, those 4 committee chairs meet with Ms Pitkin and myself to go through and talk about best practice that they've been seeing, issues that they've been seeing, contemporary matters that need to be considered for best performance of those committees. So each year we also provide targeted training programs for human and animal research ethics to staff and ethics committees. Ms Pitkin's doing one of those in Canberra today, to support and maintain our best practice. And of course we provide online services and individual advice. And as Ms Pitkin's observed, we are active members of ASCAR. So finally, I'd just like to acknowledge the members of our animal ethics committees for their service on those committees. It's a really crucial part of ensuring animal ethics is appropriately considered and all of our animal research is up to best practice. We're deeply appreciative of their care and diligence in the way in which they do that and approach those responsibilities. Chair, over to you.
Thank you very much for that opening statement, which complements the previous one very nicely. So we'll get questioning underway. We've got representatives for the opposition, government and crossbench this afternoon, and if it's agreeable, we'll share them around for the duration of time, which will take us to the conclusion. So we'll go underway with the Deputy Chair, Honourable Member Hurst.
Wonderful, thank you, Chair. I should start with some questions either to Ms Bate or Ms Torrance, whoever feels more able to answer the questions. I have read through your submission, and in the submission you give quite a detailed breakdown of the amount of funding given to animal research projects in the last 5 years or so. I'm wondering if that information that was in the submission is actually publicly available elsewhere, such as in an annual report. Sorry, Ms Torrance, I think you're on mute.
Sorry about that.
That's all right.
So, no, our annual report, given the range of activities NHMRC undertakes and our broad remit, doesn't get into details about animal research numbers. So, no, those numbers are not published in our annual report. We're able to provide them on request. The total amount of funding shown in that would be the amount of funding for all of those projects, not the amount of funding that goes specifically to the animal research component. We're only able to provide the total amount of funding for each project. It's not itemised in that way, so just thought I'd put some clarification around those total figures.
We've heard a lot in this inquiry about concerns around transparency, particularly when it comes to the funding for animal research. When you say that you would provide this information on request, have you ever had requests specifically for that information, and then, you know, are you voluntarily providing that information to any requests that come in? And why is it not sort of published annually?
We do get questions on notice through the Commonwealth parliamentary process, through the Senate estimates process, around the number of projects and amount of funding that includes animal research components, and that's usually where we would respond and publish those numbers when requested, and they would be, you know, available through the Australian Parliament House website. We could publish the numbers regularly on our website, but my sense is it's not the key data that people are looking for. The key data that people are looking for is usually the number and types of animals that are used in research and actually used in research, which would be information that would come from animal ethics committees that monitor those projects.
So you don't have that information, the number and types of animals that are used in the projects that you fund?
We request at the grant application stage whether or not animals are proposed to be used and what type of animals and the proposed numbers. But those numbers do get refined later, post-funding, through the Animal Ethics Committee approval process. And we don't collect the final data, we only collect the proposed. And we do that to monitor trends in what we fund over time, not to kind of provide any national reporting, partly because we're only capturing NHMRC-funded research. We're not capturing all animal research that happens. You know, we're only funding health and medical research as well, so it is really a subset of what happens nationally. So I think it is really the animal ethics committees that have the best knowledge of what actually then happens in each research project, both that we fund, but that's funded from anywhere, because everything must have approval and monitoring and oversight of those committees.
And it's an interesting thing that's come up in this inquiry. There's almost been an emphasis that from some of the people that have come as witnesses to the inquiry that they've talked a lot about NHMRC funding as though that is the primary funding source. You know, I'm not sure if you know the answer to this, but, you know, you sort of talked about the fact that you're not the only funding source. Do you know how much of animal research as a percentage that you are funding from all animal research?
No, because I don't.
Sorry, Ms. Torrance, you've just sort of broken up there a bit.
How much others are doing. I just know that there are both other Commonwealth departments. Sorry, my audio cut out. I didn't hear that.
That's right. I was just that your audio was cutting out. I was just letting you know. But I think that there's a little bit of a delay. If it keeps happening, it might be good to turn your camera off. I think it might be a streaming issue. Hopefully you're still there. Yeah, okay, great. I'm being told that you're still there. One experiment that's been flagged as controversial in some of the submissions and witnesses in this inquiry is the nose-only smoke model or smoking towers. Does the NHMRC fund any of these experiments?
I would have to take that on notice. I don't have details of projects like that with me.
Is there any specific projects because of, you know, there's been quite a few projects, I suppose, that have been flagged during this inquiry as some researchers say that they're not valid, like forced swim tests and smoking models, and a lot of people have flagged them as highly controversial because of the animal welfare impact. Does the NHMRC have any sort of process in place where they wouldn't fund certain experiments because of the high animal welfare impact or because of concerns around that scientific validity?
That's a good question. The code, as well as the peer review processes that we use and the things we fund ourselves, really do set in place the key principles that should be taken into account when deciding what methods to use. And so there's a whole series of processes that should happen well before an application is received by us, where the researcher is taking responsibility for considering whether the method meets best practice, considering all relevant aspects of, say, species-specific biology, physiology and behaviour, considering the best available scientific evidence, including international evidence, and including the potential adverse impact on the well-being of animals used and any strategies they might need to minimise adverse impacts. Like there are responsibilities in the code for the researchers, the investigators themselves, to be considering all of that before they even propose to use a test. Our peer reviewers in reviewing a grant application would also bring that lens, and we work very hard to try and match peer reviewers with applications where they actually do have the right skills and expertise but are not conflicted on the application and are able to provide those kinds of judgments on whether they're the right types of studies to be used. And if they're not the right types of studies to be used, they're not going to make it through the peer review process because the quality and significance of the research is what they're looking for. And then even when we've funded something, We then require the Animal Ethics Committee approval before the project proceeds, and that can result in variations to the grants that we've funded to take into account these sorts of issues as well. So there's a lot of kind of checks and balances already in the system, and if the code is well applied by all the responsible people, researchers, institutions, animal ethics committees, etc., it should come out well. That said, we obviously look at when people report issues with particular methodologies or procedures, and I think we have in the past made some statements about previous procedures, and we can look at those sorts of issues in the future. We have an animal welfare committee who advises us on policies, so they're not an ethics committee like the ones that review actual research projects. They're a policy advisory committee who might look at whether we need to produce particular guidance if needed. So we can look at those sorts of things, but in general, we think that there needs to be a lot of confidence placed in the whole process and methodology of review, of ethical review, right through from the researcher deciding to the Animal Ethics Committee approving, and most of these kind of tests you're talking about are being phased out over time because of that review process.
A lot of those, so you're relying a lot on the Animal Ethics Committee process, which occurs after you've provided the grant. Is that what you're saying? That you're relying on, first of all, the researchers to have looked into the welfare issues, and then relying on the Animal Ethics Committee after you've approved the grant to identify if there's animal welfare issues?
You are right that the Animal Ethics Committee's formal approval happens post-funding in most cases. Experienced research teams would have a good idea of how their proposal would be considered by the Animal Ethics Committee and would be seeking advice if they had any concerns early on before they're putting in applications. And again, our peer reviewers would have a good idea. But it does formally happen after, where the Animal Ethics Committee obviously recommends any changes. That's taken very seriously. Some small changes, like they will look at the 3 Rs and the application of the 3 Rs, which includes reduction and then use of animals. If they're reducing the numbers and it's, relatively small reduction in numbers, then that will just happen. If it's a large reduction in numbers that might impact the scale and significance of the research, they would have to come back to NHMRC to seek approval for a grant variation to make sure that we're still achieving value for money out of the research. But that's a fairly standard process that we would look at any variations that arise, so long as the original objectives of the research being funded and are going to be maintained and achieved. We would look favourably on any variations that came about as a result of ethics review.
In regards to the forced swim test and the smoking models, the smoking tower models, I'm not sure if you're aware that the Animal Research Review Panel has been reviewing those particular experiments. Has your Animal Welfare Policy Advisory Group also highlighted those or other experiments that could come up for funding with the NHMRC? Are they ever flagged as an issue of concern?
Yes, the forced swim test in particular, we are taking some evidence and consideration to our Animal Welfare Committee for advice later in the year. We're currently reconstituting the membership of the Animal Welfare Committee because some of the people's terms are expiring, and once we have it in place, they'll be looking at that. But we'll also be looking to see whether there's already any international evidence or any international guidance, because we don't look to duplicate things that are readily available elsewhere. So yes, we have had that particular one brought to our attention, and our Animal Welfare Committee will be looking at it to see whether it is something where we need to do anything specific to provide any further guidance on NHMRC funded research and using those tests.
Thank you. And that's only for forced swim tests, so the smoking models haven't been brought to the attention of the NHMRC? Because that's the other one that's come up, obviously, in this inquiry.
No, it hasn't been brought to my attention recently, but I'll make a note of that now and we'll see whether we need to look at that one as well.
Thank you. stakeholders within the research industry as well as animal protection groups have all agreed that there needs to be more dedicated funding for alternatives, and Australia's really falling behind other countries. But the NHMRC doesn't have any sort of very dedicated funding streams. I know you don't exclude them, but there's no actual dedicated stream for funding. I'm wondering if the NHMRC has considered actually ensuring that there is some sort of specific dedicated funding for alternatives, and if that's an area that the NHMRC will be moving in at any point.
What we have done is have a look retrospectively to see if there have been any innovations in alternative models that have arisen out of NHMRC funding, and we produced an information paper that highlights some of those examples, because it does turn out, while we haven't had a dedicated funding stream for it, it has certainly been a feature of some of the research we fund. So the real limitation on what we fund is, under our legislation, we only fund research that improves human health. So it has to have that focus, but there's certainly ways in which research that has exactly those objectives to improve human health and proposes to use animals or to move from using animal models can be framed within that objective of improving health, so human health. So we are seeing things coming through. The other complication is it's not always obvious when somebody is using a methodology that is replacing animals, because they're not talking about replacing animals, they're talking about the new methodology that they're developing. So we have seen some really exciting projects using mathematical models and computer simulations that are essentially things that would have previously been done using animal models, but that's not really drawn out in the reporting, so it's not obvious and easy to find and count all those things. In terms of whether or not we would do a target funding stream, as I said, the complication is that it needs to be for the purposes of improving human health, and whether it be possible to design something that achieves the broader objectives people were looking for. So we don't have any plans to do that at this point in time, because it is a little bit complicated, but we would absolutely welcome others with funding sources putting in those sorts of initiatives.
Thank you, Ms. Price. Ask government members if questions you'd like to direct to the witnesses.
Thank you. I'm curious to know how you feel about the current regulatory regimes that we have in the state, and if you believe that they are on the money for effectiveness, or if you think that there's a strengthening that's required by the Parliament to achieve an outcome. Dr. Steele, if I could start with you.
Sure, thanks. And in the CSIRO context, the answer in relation to the state is in terms of our committee, which overlooks the Armidale activities that we have, which, as I say, is largely in the livestock space. A general observation, as my opening statement inferred, it's the dedication and due diligence and sophistication that those committee members, and the chair in particular, brings to the matter that's the first line of defence, if I can put it that way. The second line of defence is making sure that the organisation that's conducting the research has good, robust policies, but really is attentive to the intention of the code, and so treating it vigorously as opposed to treating it as a permission step. Our experience, I believe, is when we talk to the chair of the committee, that it's a very robust process that's very effective and leads to improvement in the research activities in the way in which they're conducted. So I believe that our experience is that it's pretty effective. There's a design element here, which is how close do you have to be to the individual experimentation to be able to make effective judgments about whether it's an appropriate use of research, of animals in research and whether or not the 3Rs are being complied with, and whether or not there's any better way of actually going for the aims of that particular research. That has to be done at the committee level and at the organisation level that's conducting the research. And so it's the intensity of those activities that's crucial. Of course, the overall legislative framework sets the context for that, and of course, DPI's oversight of that process is important to ensure that it's going the way in which was intended when the social licence was granted by legislation. But I think our initial view would be the New South Wales process is robust and effective in our experience, as indeed generally is the case with most other states, by the way.
I have concerns when people talk about social licence because it's such a arbitrary measure. It has no determination. It has no framework. It's somebody's belief over somebody else's. What I'm looking for is evidence, shall I say, that either the system is working or the system is deficient in some way.
Yeah.
So far, over the hearings, it's been my interpretation from the evidence that while some people may have concerns and may point to one or two historic events, that currently the framework is working. And so we ask the same questions of each witness, and we're waiting. I'm waiting for an indication that it's not, but I'm yet to get it. Does anybody else on the panel have, some view as to in the space that New South Wales has legislative control over, has there been either a failure or is the system working in a way that would provide confidence to the New South Wales public that it's operating in the way that it should?
Kathy, would you like to comment?
I would just say that my observation of the deliberations of our animal ethics committees and the approval processes that they work through, that system is actually very robust and it provides a very solid review and feedback to our research teams, and they work very hard to ensure that best practice in terms of animal welfare is maintained at all times.
Okay.
So my view would be that that part of the system is working well.
So in that instance then, if the regulatory framework and the legislative framework is such that the system is operating and people like yourselves who I think have that higher level view and would also see what other states are doing in this space, aren't saying that these things are perhaps being done deficiently or these things need to be improved immediately. What we're talking about then is really, I guess, a question of ethics and perhaps that social licence, as you spoke about. It's that where do we sit on this issue? Is that perhaps a fair way to present the issues that we're facing now? That which it's really about the involvement of animals in research.
I think you're right about. I think you're absolutely on the money there. So the question is that having been legislated as a conditional possibility and a robust process that then tests that on each occasion. If the question that's on the table is, are we satisfied that whether for medical research or other purposes, that's an appropriate use of animals, that is a judgment that goes up to what is the Parliament's view about what is acceptable in that regard.
And so part of the evidence that we heard in previous days' hearings and also somewhat today, that without the use of animal research and animal testing, things like the COVID vaccine and things like children's cancer treatments would have been either delayed or the treatment would be impacted if we didn't have those regulatory regimes in place. Ethically speaking, and again the social license aspect, do you think that that is a reasonable use of the legislative framework and the robust nature of the ethics committees, and do you think that that's an acceptable way to look at the issue? That, you know, it's really, if we didn't do this, people would potentially die.
So I have been watching a little bit of the evidence, and you've certainly heard evidence that there are aspects of medical research for which there is currently a high dependency upon the availability of conducting animal research, notwithstanding the fact that for reasons of ethics or economics and the cost of research, there's also, for a fair portion of it, quite a bit of motivation to look for in vitro methodologies, screening methodologies, etc., to reduce the number of animals and get to the point where the animal experimentation is minimised. So those dynamics are certainly there, I can tell you. Animal experimentation is quite expensive. The production of animals for use in research is not really a viable economic business. So it's always the case that institutions are concerned about the cost of those activities and are keeping an eye open for that. So I think those things are absolutely right. You do get to the end of the day, I'm afraid, to a situation where actually there are some things you can actually only do with animal experimentation, even if it's right at the end of a non-animal using lineage of research.
Yep.
And in part that's because you actually don't know what you don't know.
Yep.
And so there comes a moment where you have to test it in literally in real life.
Yep.
Now, what you'd hope, though, and there have been improvements in the Australian context in this regard over time, what you'd hope is it's really value-delivering research. So it's not cosmetic testing, for example. It's not chemical testing, etc. And I'd just make the observation that over the last 40 years, to my direct observation, there's been a very significant tightening up. I think you heard from an earlier witness on this afternoon. Very significant tightening up on the use of animals, partly because of the implementation of the code.
Yep.
But I will say to you that at the end of the day, you get to the calculation being made by a group of committee with carefully designed skill base that's brought to the committee, and so on each case, to be able to know whether it's justified, that's the point at which actually the right skills in the room with the right level of detail to be able to make that calculation. And I'm afraid that is always going to be where that decision gets made. It would be tricky to make it in a legislative framework, although you can set the principles, obviously.
Yeah, and with the Chair's indulgence, there was one last part I wanted to ask you about that. Is, a lot of the evidence has said we need to be spending more on research in the RRR space. You know, we need to be doing more to make sure that, you know, we move away from animal testing and animal research. And from a pragmatic standpoint, that's probably correct. However, I imagine that you've been in research for quite a number of years, that this isn't your first rodeo, should we say. In your experience, if money is spent, or a large amount of money is spent on perhaps pursuing that avenue and that goal, is it reasonable to say that it's likely that that money will have to come from another space and will not be new dollars that are actually put down that path? I.e., would it be at the expense of other research that, say, the CSIRO might do?
So let me come to CSIRO in a moment. I'm not surprised the committee has had the experience of people giving evidence that more money for research would be a good thing. That's a general phenomenon.
I don't think anyone ever says it shouldn't be.
Because there is tight funding for research generally, including in medical research, although that's gone up fairly significantly over a few decades, it's inevitably always the case that unless you can identify a particular application of research where you get both goals satisfied at the same time, you're making a choice between one option and another option. And so, yes, you have to do the calculation on where you're putting your preference and where you're getting your best returns in terms of the nature of the research outcomes. A fair portion, but not all of the funding for medical research, because of the context in which you've got most of your evidence, I believe, comes from NHMRC, and Prue can talk to you about that. I think she's already told you the legislative framework in which NHMRC is funded. In the case of CSIRO, we do some investment in research which reduces animals. We've got a number of different business units that do different sorts of research. So, for example, the work that's done in Armidale is done by our Agriculture and Food business unit. There's another business unit which is Health and Biosecurity and the ACDP facility. They are actively investing in vitro tests like organoid type testing to look for new and better ways of doing testing activities that would reduce animal use. That's part of CSIRO's portfolio of activities. Even there, of course, I'm sure they would say to me, have you got some more money for you, Jack, in order to be able to do more of this? Yes. But yes, it's an active investment that we're making, but you're always making those decisions, those preference decisions, I'm afraid.
Yes. Thank you, Honourable. Can I just, limited time left, but direct this particular question to both organisations, and who answers it, matter for yourselves. And it's drawn from some of the evidence which has come to the inquiry thus far from submissions and witnesses in their oral testimony. It's sometimes been explicitly suggested. In other instances, it's more implicitly suggested. And it's this proposition, and I'm going to invite you to comment on this, even though effectively you're working in organisations which are operating in the Australian context, okay? But the proposition that's been put forward, implicitly or explicitly, is this: that if you look at the matters of the research being done in Australia that involves animals, that the way in which this has been conducted hitherto, up to this point, has in effect become entrenched as a status quo position. That's the way we've always done it. And that creates, in and of itself, an inertia or a tardiness to look seriously at other methods that could be available. In other words, if I could put it this way, we're in our own research bubble involving animals in Australia, and that's the way we've always done it, and there's state, obviously, regulation, there's funding from the Commonwealth, and the model's well understood. But that in and of itself is creating this difficulty of and the phrase has been used, cultural change, to look beyond what we're doing and how we've done it, and look to see what's being done elsewhere, particularly in other jurisdictions overseas. Can I invite both organisations to comment about whether you think there's any validity in that proposition, that we struggle with the fact that we're doing it the way we always have, and that's become entrenched? And if you disagree with that, could you perhaps elucidate and explain, perhaps from examples or particular research projects you might have been involved in doing, a demonstration why that's not the case, to try and give us a sense that these propositions put need to be challenged a bit further?
Okay. Prue, do you want to go first?
Sure. So when we look at how we compare internationally, I think there's a few things to say. One is the regulatory framework that we have in place is international best practice. It does compare favourably. The only main difference is that in a number of other countries, they have national legislation, whereas here we have legislation in each state and territory, but there is legislation in each state and territory, and it does pick up the national code. So the framework's quite good. We were concerned for a while that there was a gap in information about how well that framework is applied, which I think gets to some of the transparency issues that have been raised a few times already. And the project we undertook in around 2019, looking at the implementation of 3Rs, tried to address some of that gap, and again did find that actually, comparatively, internationally, the 3Rs are very well understood in Australia and are implemented relatively well. And those 3Rs, of course, do all those things you're talking about. They do try to move us into more innovative approaches by actually questioning the use of animals. Can they be reduced, replaced? Those questions actually start to drive those innovations. But I think to really get to your question, the example that I would use, I can't give a specific example of a project, but NHMRC's Ideas Grant scheme, introduced in 2019, is all about innovation and innovative research projects. And it is through that scheme that we are seeing the examples of new methodologies, including mathematical modelling that I mentioned before, that really is starting to replace the use of animals. And the system is actually driving some innovation through those sorts of schemes, and there are other ones across government that really do focus on innovation and research. So I can't really talk to how a particular facility might run and whether there's entrenched behaviours there, but as a funder, we certainly do try to drive the incentives that would lead to those sorts of innovations and keeping on top of the absolute best practice in scientific methodologies. We have a research quality strategy and an action plan under that, where we're also trying to drive and improve the quality of research in Australia more broadly, which does include consideration of the use of animals in research, and that's an ongoing strategy and an ongoing piece of work for us, but it's a framing that we have about looking at this. So that, combined with our grant schemes, I think it is unfair to say Australia is kind of falling behind in being innovative about how we approach the use of animals in research and moving away from them. But I'm sure there are pockets of some stagnation, as you've suggested.
No, thanks, Victoria. Just testing the proposition. I don't know whether Dr. Steele would add to that.
I'll give you my personal experience in a moment. Kathy, did you want to make any comments?
I would just observe that the application process and review process by AECs actually does ask our researchers to show and justify that what they're proposing is best practice and how the application is meeting the 3Rs. And we undertake professional development with our ethics committees, particularly with the lay members of our ethics committees, to ensure that they are across expectations and best practice so that they are adequately able to assess applications. And there have been instances in the past where ethics committees have said, no, we're not prepared to provide an approval to that because we think there is an alternative or there might be a better way of doing it. And those better ways of doing it are then developed and found. So I would agree. I don't think this is a case where we're kind of lagging behind international standards. And I think the process that we have does expect best practice to be shown and demonstrated as part of the approval process.
No, thank you. That's very helpful.
And so I just supplement to say, and you'll see it in CSIRO's submission, we're keen on the openness agreement being implemented, and the truth of the matter is I think the answer to your question would be much better placed if we had longitudinal data on the number of animals and what they're used for and what's the nature of it, whether it's wildlife, observation or whatever. My own experience is CSIRO's number of animals that were used in New South Wales when I joined CSIRO and became a secretary of a committee back in 1982 would be at least an order of magnitude more than it is at this point in time. Part of that is because there's been changes in various technologies. So we used, for example, use animals in order to produce nerve growth factor and other growth factors for various experiments. That would now be done by molecular biology techniques. we used to use mice to grow up high concentration ascites fluid with monoclonal antibodies in. That would all be done by in vitro methodologies. So what's the driver of that? Was the driver of that reduced animal usage? Well, I can tell you that it was certainly something we were looking for at the time, and it was the implementation of the code that was part of that. But there's also been a fair economic impact of the cost of conducting animal experimentation, which has also been at play here. I'd love to tell you that it was slightly more virtuous than that, but the truth of the matter is that's one of the factors that's actually quite significant about determining whether particular animal experimentation even goes to the question of whether it's justified or not.
Well, that's been very helpful, I have to say. The submissions were very good, and to have the opportunity to augment that evidence with the oral testimony has been very, very helpful. So I'm sure that after reading hands, some members will have some supplementary questions. So if it's agreeable, we'll provide them to you through the committee secretary and liaise over the timing of those. So once again, we know that you're very busy in your different ways and had to make the time available this afternoon. Thank you very much. Much appreciated. Thank you.
Thanks very much.
Thank you.
Well, listen, we'll get underway if everyone's ready. On behalf of the committee, as the Chair, can I welcome you all for coming on this afternoon. It's much appreciated. This is an important inquiry for Portfolio Committee number 2, and we'll value very much the contribution from yourselves as witnesses for and on behalf of the New South Wales Government from your respective agencies. What we need to do first of all is to go through the formal part of having you sworn or affirmed to give your evidence, and probably the easiest way to proceed is to sort of move from my left to right to get each of you in order to just nominate yourself, your name, your full name, position, title, agency that you're from or part of the organisation you're from, and to swear either an oath or an affirmation, whichever you prefer, the words of which should be before you. If not, just raise your hand and we'll provide them to you. So with that short introduction, I'll commence with Dr. Tracy.
Thank you. So John Tracy, I'm the Deputy Director General for Biosecurity and Food Safety for New South Wales Department of Primary Industries. I solemnly, sincerely and truly declare and affirm that the evidence now about to be given by me shall be the truth, the whole truth and nothing but the truth.
Thank you very much. Is it pronounced Vacarthy?
Vacarthy, yes.
Please.
So Gregory Vacarthy, I'm the Director of Compliance and Integrity Systems at the Department of Primary Industries. I swear that the evidence now about to be given by me shall be the truth, the whole truth and nothing but the truth, so help me God.
Thank you very much. Dr. Filmer.
My name is Dr. Kim Louise Filmer. I'm the Chief Animal Welfare Officer at New South Wales Department of Primary Industries.
Thank you very much.
I solemnly, sincerely and truly declare and affirm that the evidence now about to be given by me shall be the truth, the whole truth and nothing but the truth.
Thank you very much. Associate Professor Garcia.
Yes, my name is Associate Professor Roger Jonathan Garcia. I'm an employee of the Sydney Local Health District, New South Wales Health. I'm also the Chair of the Sydney Local Health District Animal Welfare Committee, Animal Research Ethics Committee.
Thank you.
I swear that the evidence now about to be given by me shall be the truth, the whole truth and nothing but the truth, so help me God.
Thank you very much. Professor Phillips.
Hi, my name is Professor Jacqueline Phillips. I'm here in my capacity as the Chair of the Animal Research Review Panel, and I solemnly and sincerely and truly declare and affirm that the evidence now about to be given by me shall be the truth, the whole truth and nothing but the truth.
Thank you very much. And last but not least, Distinguished Professor Hennessy.
I'm Professor Anne-Marie Hennessy. I'm the Senior Scientific Advisor to the Sydney Local Health District and member of the Animal Ethics Committee at Sydney Local Health District since 1998. I'm a member of the Animal Research Review Panel by ministerial appointment from 2008 to 2010 and again from 2013 to current, and I've been the Deputy Chair of that committee since 2019. I solemnly, sincerely and truly declare and affirm that the evidence now about to be given by me shall be the truth, the whole truth and nothing but the truth.
Thank you very much. Well, we'll move on now to just acknowledging the government's submission to the inquiry. You probably would be aware that the government has made a whole of government submission, which is quite common to inquiries. So it's titled the New South Wales Government Submission. It stands as submission 239 to the inquiry. It's been received, processed, and uploaded to the inquiry's webpage. So that's before us. But I'll invite now, in addition to this, which we already have, an opening statement. Now, normally we just have the one opening statement from an organisation, but given the significance of New South Wales Government's contribution, I don't know whether this is going to be shared across more than one person. So I'm in your hands in that respect. Probably wouldn't be suggesting that we have 6 opening statements, but how would you like to proceed?
So I can make an opening statement from New South Wales DPI.
DPI, that's fine. Would anyone else like to make one? If you feel like you wish to do so, or had prepared. Okay, so that's fine. Well, let's just start with Dr. Tracy.
Great, thank you, Chair. New South Wales DPI is committed to ensuring best possible animal welfare outcomes. We have a strong, effective animal welfare system underpinned by the Animal Research Act and Regulation and the National Code that apply to all teaching and research involving animals. This Act also legislates the operation, composition and role of the Animal Research Review Panel. This panel provides independent oversight of the conduct of animal research and any complaints related to it, and includes equal representation from industry, government and animal welfare sectors. We support that panel in DPI. We provide administrative and technical support. We accredit establishments in accordance with the panel recommendations and collate statistics on their behalf. We also investigate complaints, inspect accredited establishments and licensed animal suppliers, report to the panel on outcomes of those activities and carry out delegated investigations into statutory complaints for the panel. The system provides independent statutory oversight of animal research activities in New South Wales with input from the animal welfare sector, while also bringing to bear the technical, investigative and compliance expertise of New South Wales DPI. As members will appreciate, the term animal research is extremely emotive, and it's therefore important we prioritise transparency in this area so the public confidence in the system reflects the very high standards of care and conduct that are built into it. New South Wales continues to provide a very high level of transparency concerning animals used in research within the legal parameters in which we operate, including the publication of animal statistical reports and the annual report of the panel. The information published in these reports goes above and beyond that of many other national and international jurisdictions, including information on the level of impact, the field of research in which they are used, such as medical or wildlife research. The statistics for the 2020 reporting period again show that the vast majority of animals, so that's 98% of cats, 91% of dogs, are used in studies with minimal impact on the animal. These might be anything from trials to assess pet food, observational behavioural studies, educational activities in which students learn how to care for and handle animals, or even scent detection work in wildlife surveys. These studies are important to our health, community and environment and for animal welfare, as the committee's heard in previous testimony today. New South Wales DPI will continue to work collaboratively across government and with all stakeholders to support the principles of the 3 Rs and to ensure the best possible animal welfare outcomes. Thank you.
Thank you very much, Dr. Tracy. Who would like to make the next opening statement? Can I invite...
So I'll take that opportunity if I may.
Please proceed.
Firstly, thank you to this inquiry for the opportunity to present. Very much appreciate that. So I thought what I would do is try and give some perspective from a chair of Animal Welfare Committee, which is the reason I'm here. The committee that I chair is a committee of 11 members, which includes 2 Category C animal welfareists, 2 Category D lay people, 3 veterinarians, and 3 members of Category B who are researchers, plus myself listed as a Category B member. My actual professional role is as a clinical consultant physician in immunology at Royal Prince Alfred Hospital, and I'm an immunopathologist within New South Wales Health Pathology, and I hold a research PhD in medicine. I've been a past president of the Australasian Society for Clinical Immunology and Allergy and served on a number of national granting bodies related to multiple sclerosis, hepatitis and HIV. So I'm just trying to give you a perspective on the sort of background, and my situation will be similar to very many people who chair these committees. The Animal Welfare Committee that I chair oversees research that's conducted by the Anzac Institute, the Centenary Institute at RPA, the Heart Research Institute laboratories in Newtown, the National Baboon Colony, and the on-site facilities used in teaching, training and research at the RPA Animal House, the RPA Robotics Training Institute, and the Institute of Academic Surgery. And the research we scrutinise predominantly involves laboratory-bred, natural and transgenic strain rodents. These are mainly mice, which are studied in animal traditional models and novel models of human disease. And to a lesser extent, we oversee the conduct of animal research involving zebrafish, pigs, and baboons at the National Baboon Colony. The facilities we inspect are in some cases also authorised as animal suppliers for research, and they conduct breeding for research and training purposes as well. There's no research currently under our committee's governance that uses companion animals, dogs and cats in our facilities at the moment, though there have been in the past many years ago. Many of the researchers use animals sourced from the Australian Bioresources in the Southern Highlands or from the Animal Resources Centre in Western Australia. And many of our facilities have quarantine rooms that can be used for direct importation of strains from sources in other countries, interstate or locally. And some of these facilities operate as pathogen-free facilities with enhanced biosafety infrastructure and strict surveillance for entry to prevent contaminating microbes. One facility has PC3 level, which is a high level of biocontainment, enabling studies of virulent microorganisms, including the SARS-CoV-2, the pathogen that's been causing COVID-19 and has been actively involved in that research. All the facilities we monitor are staffed by qualified managers and a mix of full-time and part-time staff. And of course our researchers are the investigators working within those facilities who have primary responsibility for the animals that they are researching on. As well as being under the scrutiny of the Animal Welfare Committee, they also come under the internal scrutiny of their line management within their institutes, facilities and the organisation of Sydney Local Health District. So in my clinical role at the moment, I don't currently use animals for research because I'm spending most of my time working with humans. That's my clinical role. But I did my PhD work in a project that could not possibly have been done without the use of animals at various stages of that work. It required the use of monoclonal antibodies, which at the time were produced in ascites, as you heard earlier. Now they're done in vitro. So the changing profile of animal use reflects the advances in technology. I went to the United States in 1989 and worked on humanised mice which had implanted in them human immune systems, which enabled studies to be done which were able to recapitulate the human response. And those sorts of humanised mice have been absolutely critical in a different form, not exactly the same types of mice, but those approaches have been critical in the early development of SARS vaccine and other vaccines that pathogens to prevent infections in man. So I won't go on into any detail to describe some of the other experiences that I've had close involvement with, which have been involved in the taking of basic science discoveries made in the United States, where I had the privilege of being a postdoctoral student, through to the point where some of those entities became drugs which were critical for the prevention of transplant rejection in kidney transplants, heart transplants, lung transplants, and have even been used recently in multiple sclerosis. Now, better drugs have come along, but for 20 years they were very important in the maintenance of the health of people right across the spectrum with those sorts of conditions. So what I'm trying to do is just give you the sense that as a chairman of an animal welfare committee, where one is trying to balance the impact on animals versus the need for advances in health, that sort of perspective, it assists me, at least in my consideration of what information I need to ensure is presented to the committee in the justifications that are actually provided in the course of the submissions of the projects that come to our attention. And I do emphasise that the committee is a committee, and as the chairman I see my role as to ensure that the right questions are asked, that the documentation is thorough and verifiable, and that when we then go through the steps of modification, that those adjustments get to the point of implementation, and that in our inspections of facilities we're applying the highest best practice standards in our expectations of the adherence to the protocols that we've approved. So I think I'll leave it at this point.
Very good. That's laid out a very nice base for us. I'm sure there's going to be some questions arising from that. So thank you. Professor Hennessy.
If that's okay.
Please do.
Thank you. I will just add, can I just start by acknowledging the Gadigal people on whose lands we meet in this, their NAIDOC week. I'll just add quickly, because Roger's set the stage for me. I'm a renal physician, a kidney specialist, and I deal with the complexity of patients in the New South Wales system every day. I see the complexity of our responses to those patients, and I deal with distressed and bereaved families with early death, the loss of sight, the loss of limbs, and the death of a baby. I've worked in the COVID wards, and I've seen the death that COVID can bring and the scariness of being unable to breathe at the end of your life. In fact, I saw a COVID death this morning. I bring a perspective as a clinician scientist. Also, I have a PhD, as Roger indicated, as part of being a clinician investigator within the system. It's my job to review evidence, to ask questions, and to think about the best way to answer them. I really wanted to just make a statement that I've seen in your questioning about the idea of how do you get a linear journey from a problem to an animal experiment and then out the other end to a solution. And I see that as much more of an iterative and a complex process. And we would ask a question. The tools we have are family, teams and data. We then move to pills and surgeries, but we're every day asking for better solutions to the problems that we see and what's the best way to get the best information to do that. We would start with the data that's based on a human population. We would think about the question that's relevant to those families. They would donate their tissues to us, and we start with that. We might use complex computer modelling to come up with a particular pathway or an approach, and then we have to try that out and see if that would work. We would, at all costs, try multiple tools to do that, and any application that I've ever looked at for funding, I review hundreds of those every year, would have a multi-pronged approach to doing the work, of which animal research was only a part. I wanted to use the example of the COVID research, and so having come up with 20 years of work towards mRNA therapies, having used the computers to give us the best possible sequence for COVID-19, and signaled and worked out what that compound could look like. Do we coat it in cholesterol? Do we put it in something else? What do we do to try to deliver it in the right way? 200,000 Australians, humans, subjects stepped up to be part of the research that would enable the global vaccine to go forward. But how much more comforting to be able to go to those 200,000 volunteers and say this has been tested in a few dozen macaques, so it was done in non-human primates, but in a way that enabled the next step to go much more quickly. And you heard from your witnesses on Monday last week about years being shaved off the journey towards a vaccine success by doing an iterative and integrated approach to research that takes into account all the advances that we're looking for. And my final comment is the research quality is underpinned by a strong regulatory environment. That means that the animal welfare is paramount within the context that I speak about trying to improve these massive problems that we deal with every day. But the quality of that work and its regulation and its support through the mechanisms described today means we're all striving for the best possible outcomes for both. Thank you.
The defence rests, Your Honour. That's very good. So thank you for that. The combination of those three knit together very nicely and I think provide the basis of some questioning. So we have representatives at the table from opposition government crossbench, and we'll share that around if that's okay for the duration, which is just hour or so. So perhaps start with the Deputy Chair, Wayne Hurst.
Thank you, Chair. I'd like to begin my questions to both Professor Phillips and Dr. Filmer. We've received evidence on the first day of this inquiry from an animal care technician who raised some quite serious concerns. One problem was in regards to overbreeding inside institutions and the culling of potentially thousands of animals that are healthy and have been bred in excess to experimentation. And I wanted to understand if this is a problem that you're aware of and if it's something that the Animal Research Review Panel or the DPI have actually investigated.
Kim, did you want to comment on that first?
The regulation of the breeding has an oversight by the Animal Ethics Committee. So any breeding that goes on should be monitored by the Animal Ethics Committee that breeding sits under. So animal ethics committees look after and control and regulate and monitor the breeding as well as the research that occurs. So if there was a suggestion of someone having a complaint about that, then that should be lodged as a complaint and go to the compliance area for investigation.
One of the documents that we received as part of an SO 52 showed one facility killed 807 animals that were bred in excess. Is that normal that such a large number of animals are being killed because they've been overbred, or is that quite a specific situation?
Can I just appreciate the question? The SO 52 obviously is a process of taking information from government agencies. Could we perhaps guide the witnesses a little bit to the detail of, because it might be for the first time they've heard this, I'm not sure. So
I was going to suggest, Chair, that if we're going to question the witnesses on evidence, that we should perhaps procedurally and observing procedural fairness, provide them with copies and provide them
I
also
have
with
copies
time.
here, Chair. So it's part of the it's part of an SO 52 with it was actually an agenda item of the Animal Research Review Panel, so it's part of the Animal Research Review Panel minutes, so it might be something Professor Phillips will have a better understanding of.
No, I'm just trying to ensure due process and procedure here. So
I think, Chair, we also need to allow the witnesses time to read it, but also absorb the information, because as you said, it may be the first time they've...
Sorry, Chair, but this is an Animal Research Review Panel document, so I don't think it's the first time the Animal Research Review Panel has their own minutes.
Just order. Just to make sure that everyone has an opportunity to be aware of what is before them, and if they need to read it, they obviously need to read it. There's no assumption in my mind that people necessarily study the document or are aware of its content. So I'm not trying to be obstructionist, I just want to make sure that everyone has the opportunity to be aware of what's put before them. If you need more time to study it, and I'm not being obstructionist, if you need to take it on notice, you're able to do so, but if you're able to make some comment, I'll invite you to do that, but as you see fit.
I'm unable to answer that question. I'd have to take it on notice, but the Animal Research Review Panel, I'm not on that panel, so I'm probably not the best person to answer that anyway.
So I'm just reading the material.
Got to give the witnesses an opportunity to study the document.
So can you remind me, please, sorry, and just in terms of the what you're wanting to know about this particular, I'm assuming it's item 9 on the agenda item that you've highlighted for us?
Yes, so item 9, it says 807 mice noted as culled in annual report. I'm just wondering if that is a common number that you would see coming from as a bred of access of animals, or do you think that that is quite an unusual figure to see?
Right, okay. So I'll just have a look at what we've said in the discussion at the ARP again. So we have questioned this, obviously, and so it was brought to our attention and a point of consideration for the ARP. In terms of other numbers, I probably am not in a position to quote. I wouldn't know off the top of my head what other numbers are from other committees. or other reviews that we've looked at. However, because we have looked at this, we've obviously gone back to ask questions and to dig further information about it.
So it was alerted to you as a high
noted by
number.
us. Yeah, it was the fact that we've looked at it and discussed it would suggest that the ARP has had some concern about it. So.
Is there an actual reporting requirement on the culling of excess bred animals that goes directly to ARP? I know that Dr. Filmer said that the AOC oversees it, but is there actual reporting requirement that goes back to ARP specifically for excess culled breeding stock?
So in terms of our reporting requirements, in terms of numbers, each year the institutions have to report the total number of animals that have been used, and they'll be classified under various categories. So if animals are reported under excess breeding protocols, I'm not quite sure what the category is that they would come under, Kim. In terms of our annual statistics report this year, I don't think we actually break that one down specifically in terms of a number that we get from the AECs in their annual reports.
Okay, so that's not something that's recorded?
No. So we get the animal numbers and we get their use per se, and that would be the type of category of usage that they've gone under in terms of the research. And I don't have a recollection on the top of my head as to whether we've got the...
If you could confirm just on notice, that would be great. But I think probably from the statistics that I've seen coming from it, that I haven't seen it either. So I think that we're probably on the same page there. Given the concerns that were raised by the witness that came, is that something that either the Animal Research Review Panel outside of this incident have investigated or are planning at any stage to look at in regards to healthy animals that are killed because they've been bred in excess?
So the way we would be aware of this type of information other than through a report like this, so we have annual reports that come through from each of the ethics institutions. And we also undertake inspections of institutions, and these would be the type of things we'd look at. And then we would have engage a conversation with those institutions, particularly with the Ethics Committee, and sort of try and understand the rationale or the reason for those numbers at that time, and work through with each institution individually.
So it would be something you would deal with on a case-by-case basis, but it's not something you've ever sort of looked at holistically across the board?
No, but we would certainly be something that we look at when we look at reports coming through.
And you've probably also heard, possibly also heard the evidence that that same animal care technician gave. She gave quite a few horrific examples of a facility she had worked at. She talked about toes and tails cut off adult animals, animals not giving proper analgesia, researchers culling animals because they didn't like the data that they were getting. Is this something that ARP is looking to investigate into now that she's come forward with this information?
So we haven't been provided with that information, so I can't comment on that. I'm not aware of that information personally, so I can't comment on that.
Okay. She said that she put in a complaint to the facility, the Animal Ethics Committee, and that was ignored. And so she left the institution. She said she felt that there was nothing else that she could do from there. Does that concern you that she didn't know where else to go beyond that, and that an animal care technician had concerns that weren't dealt with?
So one of the things that we do look at when we do inspections and we liaise with the organisations is about their complaints processes, because that's one of the procedural issues that we think is really important. So there should have been the opportunity for her to have an internal complaints process. We also have the opportunity for complaints coming through to ARP directly. Greg can perhaps speak on that in a little bit more detail. So those opportunities and those avenues are available for members of the public or for anyone within those institutions.
I might talk about, we've heard a lot about smoking models and forced swim tests throughout this inquiry. And we did hear, particularly with the smoking, so I'll talk about the forced swim tests first, because do you have, and I'm aware that the ARP panel, from minutes from the ARP panel, that the ARP is actually looking into both forced swim tests and the smoking towers. Do you have some concerns that, we've heard evidence that some facility have outright banned the approval of forced swim tests, while others are continuing to use them? there seems to be a lot of variability in their acceptability. Is that something that's become a concern within ARP about that sort of variation, and that some people say no, it's not justified, and other people say it is justified?
So as you mentioned, we've undertaken a survey of the institutions in New South Wales in terms of the use of forced swim tests and the smoking procedure, as you indicated. We are in the moment collating that information, and we're also looking to review the literature around the forced swim test and identify what the debate is with the welfare considerations and also with the research outcomes associated with it. So that's a work that's currently in progress, and it's something that ARP hasn't quite landed on yet. It's something that we're hoping to sort of finalise with a guideline recommendation in the next 6 months.
Thank you. There was also some specific concerns about one particular forced swim test that was done at the Victor Chang Institute for 90 minutes, where a lot of other forced swim tests are between 45 to 90 seconds. And I think we heard from one of the universities this morning that they were having deaths from these 45, 90-second ones, and that another facility was doing them for 90 minutes. I mean, it's quite, 90 minutes sounds, I suppose, to a layperson outside of this quite extreme. Has the Animal Research Review Panel taken any action on these 90-minute forced swim tests, or any investigation taking place?
So I can't reveal the confidential aspect of that at the moment because it hasn't been discussed at the ARP panel, but we have had a meeting with the Victor Chang Research Institute and have talked to them about that, but at the moment it's not a position where I can probably say anything, as ARP hasn't discussed it since that meeting.
And in regards to the smoking models, the ARP panel minutes talk about their concerns with the fact that NHMRC was still funding smoking experiments. Has anything come out of those concerns and any communication between the Animal Research Review Panel and NHMRC?
So I think that the minutes would probably actually reflect that a member of the ARP had those concerns rather than the ARP as a whole agreeing to that statement. And as I indicated, that we have undertaken a review of the forced smoking procedure, and we are in the process of finalising a guideline associated with that. When that guideline has been approved by ARP and for submission and publication by the DPI through the ARP website and with the institutions and informed researchers, one of the groups we are going to circulate that guideline to is the NHMRC.
Are you concerned that, we talked about guidelines for both the smoking mice and the forced swim tests. One issue we heard today was, and it was more about rehoming in the discussion we were having, but that a guideline is simply a guideline. And that even though there's guidelines put in place, some people who are trying to make positive changes adopt that guideline, and some people simply don't because it is a guideline. Does the Animal Research Review Panel have the power, or is there even the ability to actually sort of submit that to the Minister to make it something other than a guideline? Or is the Animal Research Review Panel just looking at creating guidelines on these experiments at the moment?
Those capacities are there, but with regards to the forced swim test, I'll say with regards to the smoking procedure specifically, we undertook an extensive consultation and review around that, including getting independent expert advice, and at the moment the panel decided that it was appropriate to have a guideline which really raised the awareness for animal ethics committees specifically about the implications associated with the procedure, with some fairly strong recommendations about what those AECs should consider if they were to approve. What we've also done, and this is a guideline that's going to be available soon, is we've also made the reporting around smoking a higher level. So it will actually have to be reported by institutions in their annual report to their governing body, which the ARP will also review, which will increase the level of transparency around that, and will also report it at a higher level of invasiveness than what it would have been reported and required before. We're then going to look to review that process in 12 months.
Okay, was there one suggestion that's been made in this inquiry was, obviously there was people sort of suggesting that those procedures should be outright banned, and then there was some people taking a softer view that they should have ministerial approval. Is that something that was also considered by the Animal Research Review Panel, that they would move into the category of ministerial approved projects?
No decision was made.
No decision on that has been made. We also heard evidence at this inquiry that actually somebody on an animal ethics committee for one of the smoking mice experiments had actually objected to the project. Does that, have you heard of that as well? And is it possible for an animal ethics, somebody on an animal ethics committee to object to a project but it gets approved and runs anyway? Is that actually possible? I'm just trying to follow up what the process is there based on the feedback that we had received from another witness.
So in terms of decision making for animal ethics committees, Roger, you might be able to help as well, but the committees will tend to work towards consensus with regards to decisions for approval of projects or otherwise. Roger, in your committee, is that the process that you work for?
Yeah, very much so. This whole process works on the basis of finding and, I suppose, articulating what the issues are that the person is actually objecting to, if there's an objection to a protocol, and working with the researchers to establish, whether that protocol can proceed with mitigation strategies that can reduce the impact on the animals, or that will increase the validity of the model, or whatever. There are various reasons people object. In some cases it's scientific. They feel that that isn't the best model, there's a better one, or that they feel that some aspect of that protocol needs adjustment. It's using too few animals to reach conclusions, or using more than is necessary. So without looking at the specifics, you wouldn't know why someone actually objected. But it is the chairman's role, I believe, to try and establish exactly what their concerns are, and give an environment where people feel in a committee environment they can articulate their concerns and work to establish whether that committee can reach a consensus about approval. It's not uncommon to defer an approval to the next meeting, and in between meetings to do quite a lot of work with the researchers, and sometimes it is a matter of them giving much more detail about what they're doing. Very often they have considered some of the additional aspects that they haven't written, and people then are much more comfortable approving a protocol where they're sure that the experience and the international experience with this model is such, or that the value of the work is such, because ethics committees are trying to always do this balance. We're depriving animals of liberty. We need to balance that by a gain in medical and scientific knowledge.
I guess what I'm trying to find out is technically, I guess the accusation was that one of the Animal Ethics Committee members objected to it and the project went ahead anyway. So I guess I'm trying to find out, is that legal? Is that possible that, say, a Category C member says, no, I don't think that this is acceptable? Can the rest of the committee override that? I understand you're saying that there's an attempt to get a consensus, but if there isn't a consensus, can the project be approved and run anyway?
Look, so it's a situation that must be extremely rare for that to actually occur, where a consensus can't be reached. My understanding is it could, that it doesn't require a unanimous decision on behalf of the committee, but I think one would find that the minutes would very clearly articulate that issue, and that those reports that went forward from that committee to its administering institution would identify the fact that there is a conflicting view about the value of the research or the issues, and these are part of the reporting that happens in a regulatory environment with annual reports to institutions. So ethics committees are required to provide reports to their administering institution.
Sorry, Professor Phillips, you had...
So I was just going to clarify from the code. It is actually a provision there for if a consensus cannot be reached. So this is 2.311 in the code, that if consensus is still not achieved after, as you've described, discussion and attempt to resolve the differences and exploring with applicants ways of modifying the activity or the project, the AEC should only proceed to a majority decision after members have been allowed a period of time to review their positions, followed by further discussion. So it is possible for that to proceed.
Thank you. And is the ART panel aware of this accusation in regards to the approval of a smoking mice model, where an Animal Ethics Committee member had apparently opposed? Has that information in front of the ART panel?
I don't think I'm in a position to state that in a public forum. My apologies.
Okay, that's all right. One concern that's also been raised in this inquiry, sorry, I'll move on to the DPI now. One concern that's been raised throughout this inquiry is around transparency and how funding is going to animal research from state and federal government. And the NHMRC has supplied us with some figures today, which is quite helpful. But we still don't have any specific number on how much state funding is actually channeled directly into animal research. And I have asked the Health Minister previously in budget estimates and haven't got any answers. Is there any insight into how much funding is actually going into animal research? And if that data is not available, why isn't that being recorded and why isn't it accessible?
So I'll have to take that on notice in terms of the funding amounts because I don't have that on hand. I guess for transparency, I mean, we strongly support transparency in terms of animal research and this activity. So I think we do everything we can within the powers available to us to get information out.
Thank you. Honourable Chris Ray.
Thank you so much for all your evidence so far. I wanted to ask about how strict the regulation is or the sort of rules in place for medical research on a non-human primate. I assume it's not exactly easy to get that sort of approval, but how strict is the regulatory regime in place should a researcher, scientist want to go down that path, or what's the process involved? I don't know who to ask that to, but whoever's...
Well, I might give it a go to start with. With medical research and any animal research, the framework in place, which is the Animal Research Act and the code, apply. Going down to the detail of how it gets issued is quite a specific component of the Animal Ethics Committee. So the regulation around those committees is quite stringent, and also the colonies and the use of primates falls under two codes, and there's one specific code that applies to the use of non-human primates as well. So what the DPI does is we do inspections and work with the Animal Research Review Panel, and we conduct inspections of those facilities to ensure that both the Animal Ethics Committee and also the site where the animals are housed in the baboon colonies are essentially maintained to the required standards. So that's our component. I think you may be referring a bit more to how do you actually get the research approval, animal ethics approval, which sits with the kind of the Animal Ethics Committee that's responsible for issuing those authorities.
Yeah, because I assume it's a lot stricter or harder to get approval for primate than it would be for mice, for instance, and just sort of wanting to ask about the differences in what the regulations that might exist between those different levels, if I can put it that way.
Perhaps I could add to that. So this, I'm just holding up a document here, which is I think about 30 pages or so, which is the Principles and Guidelines for the Care and Use of Non-Human Primates, Scientific Purposes 2016, which is in addition to the code, which sets out all of the additional requirements for the use of non-human primates. As you've heard, there's a whole series of considerations, including where the animals are sourced from, because currently we scientific community do not import non-human primates, etc. Without specific approval. higher than the Animal Ethics Committee. We don't, as an Animal Ethics Committee, have the authority to approve a project where that will involve the importation of non-human primates without that authority having been obtained at other levels. So there's right through from the sourcing of the animals right through to their ultimate fate. Every step has a stringency because the threshold for working with those animals is I think augmented by the reality of the proximity of that species to man. But we also take the view in the Animal Ethics Committee that every animal is an animal and is valuable, right through to zebrafish, and the smallest vertebrates. So the principles are no different, which is that the researcher has to justify the use of that species, and effectively the threshold for the use of non-human primates is that this experimental work could not be done effectively in anything other than a non-human primate, and that it is a necessary step to advance an important field of medical research. So just to give a sense that in our work that we supervise with non-human primates, the numbers of animals that are used are very few, because each animal is studied so intensively to try and generate the maximum amount of information. And some of the animals will participate in a number of protocols, some of which are observational, behavioural, etc., right through to experiments which might be involved in transplantation or something which is a very major impact intervention. But as you've heard from Professor Hennessy, the conditions that are actually being studied are some of the most prevalent conditions in the community, and the ones which have been what you'd say the wicked problems, trying to impact on preeclampsia and premature birth and premature death of neonates in man, to get treatments. One of the protocols we deal with is trying to develop a new treatment for leukemia and is very close to going into human trial. Another is looking at a mechanism of delivering previously injected drugs in a different mechanism which would allow oral dosing. And these are conditions for which the beneficiaries are not numbering a few, they are numbering hundreds and thousands of people in this country, let alone internationally. And those approaches cannot go into the next stage, which is human administration, without having had a step in a subhuman primate, because the next step will be the administration into human volunteers. So this is a very major threshold that has to be passed before those sorts of protocols would get approval from the committee. And the committee then has to be very satisfied that the researchers have the expertise and are using state-of-the-art technology. So in all those examples I just gave you, what is being used is absolutely cutting-edge state-of-the-art technologies, for which we should be very proud that there is the opportunity for those sorts of medical advances to be made, and for our researchers to be contributing to those developments. But it's on the basis of need, that is the step which must be done if that line of human advancement is to take place.
So we heard, you sort of mostly answered my next question, but I'll ask it as well, is that we heard from Professor Andrew Knight earlier about his view that we shouldn't be using non-human primates for medical research. What do you think would be the impact if we were, for instance, if the government were to ban non-human primates for medical research? What medical research impact would that have?
So can I just ask clarification? Do you mean the New South Wales government or the national government?
Because say New South Wales, or it's more like what if you weren't able, or express it in a different way, if you weren't able to use non-human primates, putting aside what level of government and what regulations are being changed, if you weren't able to use non-human primates for medical research, what impact do you think that would have?
For the sorts of projects that are currently running, I think that would potentially mean that project could not continue to the next stage in Australia or in New South Wales. In some cases, the researchers may be able to find a funding partner which would enable that to be done with collaborators elsewhere, where there was a different regimen of governance. We have been proud in this state and this country that we believe we have state-of-the-art governance for subhuman primate research, and we very much believe that it's better to do that research in this country, in this state, than to have that research done in places where the stringency and the supervision and perhaps the quality of the research may not be up to the same standard. And so the reliability of the findings may not be at the same standard either. And then if you go to that and take that argument further, then what you lead to is a situation where you may administer things to people in human volunteer trials, which you might not have had they been done in a more stringent, scientifically advanced environment. So I think there would be commercial pressures in some circumstances to do the research in other places. For some types of research where the funding pathway is meagre or more meagre, it might be the end of that whole line of work. And that would be, I think, actually for some drugs it may be some pathways of treatment that might be very many years before it's able to be picked up again using alternative methods.
Smoke inhalation, which we've heard about constantly today and on the first day of the hearing, first hearing as well. Benefits of using it versus the ethical issues with continuing with those trials. What are the benefits and what would be the problems if that weren't allowed to happen in the future?
I think that's directed
to
myself. I'm happy to pick it up because that type of research is under one of the places it's done is under the supervision of the committee that I chair. Well, there's over 8 million deaths a year from smoking worldwide, a minimum. That's WHO estimate. 1.2 million of those are passive smoking, so it's occurring in people who have no choice in many cases in that smoking exposure. In many countries it's over a third of the population, and worldwide it's closer to 20% of people smoke. So it is a absolutely massive problem, and whilst there have been gains in reducing the rates of smoking, the harms are going to continue for many decades, even for those people who've stopped. So we have absolutely a critical need to, I'm sorry about the phone ring.
That's okay.
We have a critical need to make some advances in trying to stop the progression of disease in those people who've already been exposed to smoke, and to those people who will expose themselves to smoke, and the passive smokers. And so it's diseases like emphysema, which leads eventually to respiratory failure, it's lung cancer, and it's not just smoking. We're in a country in which bushfire, wood smoke, particulates, all of these actually contribute to the environment that we live in, and that affects not just humans, that affects animals as well in the wild. We lost billions of animals probably in the recent bushfires. So getting some basic science understanding of the effect of inhalation of smoke, a greater understanding, is in fact a very important issue for this country as it is internationally. So that's the balance. So the question then becomes are the animal models able to answer some of those questions to justify the addition of smoke to the air that the animals are breathing, whether they're in towers, or whether they're in a cage with smoke, or whether it's administered to their air in some other way. And so that's the question that has to come up to an ethics committee every time they approve a project. Where is the state of knowledge up to? Is that knowledge already known? And if so, we may not need it. We may have great doubts about it, and there may be a situation where it still needs to be verified with a different strain of animal or whatever. So that balance has to be struck. And that weighs heavily on the minds of everyone on a committee that's approving experiments which expose animals to anything that is actually something that they would otherwise avoid, which is natural behaviour would be to avoid that environment. So then it comes down to how that exposure is delivered, how certain we are that any distress those animals are experiencing, any health deterioration is being picked up in monitoring, that there is an opportunity for rest and recovery, and that the endpoints that have been chosen for assessing whether that has achieved the scientific, answered the scientific question, that those are valid. And so where that is the case and where those I suppose, requirements are met, then a committee can feel comfortable that the gain potentially is such that it's justified and the researchers have justified doing the work.
Similar with the similar question, I suppose, the forced swim test that we've also heard about would be a similar thing, or what would be what's the benefits of using that versus the cost if researchers weren't able to in the future?
Look, I don't feel I should answer the question on forced swim test because I actually haven't, that has not come to our committee at any stage, so I haven't really looked at the literature to see whether there are alternatives that are adequately reliable in the conclusions that are actually reached. So I would take an agnostic view myself as to where that balance actually best fits in that particular investigation. And as at our committee, there may be people on our committee who have had from previous committees they've sat on experience. I'm not aware of that, but it's not something that's been discussed. That forced swim tests have not been discussed in our committee.
So, if you like, I can just add to that. I think, the key to this is the justification for the research, and I think, what we've got in place within the animal research system here is a thorough one in terms of looking really carefully at the justification of each of these steps. In addition to that, considering alternatives, is there any other way this research can be conducted? So it's quite thorough, the system, both through the Animal Care and Ethics Committee, overseen by the panel, is a thorough way of looking for alternatives, looking at whether the numbers of animals are sufficient or are too large, and as well as minimising impact to animals involved. So I think that the structure in place is set up to ensure that research is conducted in that way.
I just had one question, if I could, to Associate Professor Garcia. Just in relation to some of the questioning from the Honourable Amherst earlier, is it fair to say that in instances where there are objections, perhaps during a meeting of animal ethicists about a test, that perhaps an objection isn't necessarily about the procedure itself, but more that there might be a concern about the scientific rigour around the test, i.e., like you said, there isn't enough subjects or animals being used, or the methodology in the science, as opposed to an objection around the use of the animals. Is that a fair assumption? There's a number of reasons why it could be objected to.
Absolutely. The number of considerations that I suppose the lens we put each protocol through is to look at all of these aspects, including the statistical power, the power of that experimental protocol to answer the question that has been posed. So there have been, and contextualising that research So it's not uncommon for us to refer researchers to a literature paper which might take a different slant and say, comment on this, how that information modifies what you're putting forward to us. That's certainly something that's commonly done. Just with respect to that's internal discussion in the committee, but many committees, and ours is one of them, often invite a member from the research team to present to the committee, particularly if it's a new technique or a new line of research, and to justify their work, their written work, face-to-face, and answer questions, and it's an iterative process. A vast number of the protocols are, I think, improved by the actual review process that they go through. You have, in our case, 11 minds that have thought through every aspect of this protocol, lots of questions raised and addressed, and in some cases, that will actually point the researchers, to things that can improve their actual approach. Sometimes it's even awareness of other people working in the same field who they can be put in touch with. All of this is part of the 3 R's. It's about replacement, reduction, and refinement. And refinement is an important part of what we do. And all of those 3 R's, we've got very good evidence that actually is taking place, adherence to that. And it's not uncommon for our researchers who had proposed to do experiments with larger numbers of animals to go on the basis of some of their early findings to the point that they're not going to pursue that line of research any further. Occasionally that will lead to the culling of animals if they have been breeding them in-house for that work, but that work is not going to proceed because they found that it's futile or they got an answer they weren't expecting. That sort of comes to one of the issues which was discussed earlier, about and committees will look at the reports coming back from each protocol, including the breeding and the culling and the reasons for it, and will ask, why that took place. There has to be justification for the proposed numbers of animals, and also justification for why those proposals weren't actually met. Was it a technical issue? Was it a change in knowledge? Has someone else done the experiment, reported it, and it's no longer a necessary part of advancing the field?
Can I add, if that's okay? I saw it mentioned by other witnesses, the notion within these committees of pilot experiments or side-by-side experiments with alternate techniques are not uncommon. So Roger might like to speak to there. We don't necessarily go down a pathway that's going to be a futile pathway, not that you were known in advance, but there are other options that you've also explored in these investigations about ways that you can refine the numbers to get the best and safest outcome. Do you want to speak to that?
Yeah, look, I think that's certainly part of our role. Is trying to have an environment where we can keep coming back to the committee. It is once again not unusual for a protocol in its life of 3 years to come back to the committee with modifications, amendments, and some of them come back sort of every second or third month, with a refinement or a change in technique or something else that actually is an improvement. So that is the regulatory environment we're in, where it's not approve and leave it and forget about it and come back 12 months later. It is let's keep this process as a live process so that we can get refinement and reduction and in some cases replacement. And the committee does keep an eye on international developments, in particular the European Union's Commission, which has a group who validate assays that replace the use of animals, and that's an important aspect of and that's been possible, and that's particularly those in vitro and non-living animal experiments are an important step in the drug development process now of a lot of drugs, but eventually it gets to the point where the ones which have had the best safety profile in the test tube get to the point that they need to be tested in a living animal to establish whether there are off-target effects, unanticipated effects, and whether the notion that has been the basis of that drug's development is in fact correct before moving to the next stage.
Thank you very much for providing that context around the answers, Chair. Thank you very much. I know we're running out of time.
Thank you, Dr. Westling. I'm going to be referring and dipping into the government submission, which you may or may not have, but it doesn't matter. I can quote from the specific page. If you've got it in front of you, all the better. Can I just start on page number 8? It's under E, and it's talking about the regulatory framework. So, Dr. Tracy, it's to you. So under E it says the adequacy, this is one of the terms of reference terms, of the current regulatory regime regarding the use of animals in medical research, particularly in relation to transparency and accountability. It's got regulatory framework, and then there's a series of dot points. And the third dot point says the New South Wales Department of Primary Industries, NSW, is the regulator responsible for compliance under the Act and Regulation. In terms of, perhaps if I put it in this way, if you're asked to explain, to scope out what are the boundaries or the elements of what the compliance work that DPI does under the Act and the Regulation, is there somewhere where you point one to go to see what that scope is? In other words, to understand the broad term of what the regulation means. What, as the regulator, whose role importantly involves compliance, how do we understand what that is as activity inside DPI?
Yeah, so look, I can make a start and then see if Mr. McCarthy wants to add anything to that.
Just to give us a sense of the landscape that we're looking at.
Yeah, so I mean, we do underpin the sort of oversight of the research panel. So we provide support for that panel in terms of technical, administrative, but we also accredit establishments in accordance with the panel's recommendations. So that's our compliance role there, is the accreditation of that. We also investigate complaints. And also we're delegated investigations to us to investigate complaints on behalf of the panel. And we also have an audit and compliance program in place in relation to that. So one of the key sort of advantages of what we've moved towards is a risk-based compliance approach in New South Wales DPI. And that brings significant advantages to getting the most in terms of being able to target the highest risk areas. I might just see if Mr. McCarthy wants to add to
if that's okay.
Yeah,
I might
please.
just add to this. So we do have a wide range of activities that, as Dr. Tracy has said, we deal and we can point you to the Animal Ethics Info link, which gives a bit of information about the inspections and how we do those inspections. But there's also components of that around the complaints and the approach that we take under formal complaints that are legislated, and also general complaints where somebody doesn't wish to put in a formal complaint, and this kind of goes back to what was discussed before. We always encourage people, if they've got concerns and they might be, on the Animal Ethics Committee or any general public, to make the complaints to the department for investigation. So we have a role investigating complaints. We are sometimes delegated the functions of the Animal Research Review Panel to investigate those formal complaints and to assist in providing those reports back to the Secretary to make a determination on those complaints. Within my team, we also administer the licensing components of animal research establishments, as well as supplier licenses. So there's a number of different components that the compliance, the range of compliance that's undertaken. We have the inspector that does the inspections of animal ethics committees, as well as working with ARP to go and do those inspections, and that's also detailed, I'm pretty sure, on the Animal Ethics Info link. So it's quite a broad range of compliance activities that the department's involved in administering the Act, and so it's not only providing those inspection functions, but also assessing the applications, presenting them to the Animal Research Review Panel for assessment, as well as providing statistics, assisting in the statistics that are collected and presenting that information. So, and I think there was a discussion around the secretariat function, kind of an assisting ARP. So they're all key components, I guess, of the system and the regulatory function and administering the Act that DPI plays.
On the issue of inspection and audit, if I could, there's been what I discern from some witnesses a concern or some rising doubt, if I could put it this way, of the integrity of audits and inspections which are not done by the regulator per se, but may be done by another institute on the basis that you do the audit of my facility and I do the audit of your facility. I stand to be corrected, but I'm quite sure we've heard that example from witnesses, I think, from the last hearing. The argument being there is a suggestion that that is not as rigorous as actually having, dare I say, the cop on the beat coming in and doing the audit and running the ruler down how things are being done. Could you just explain whether this now has become the typical way in which auditing is done via the sort of parties perhaps involved in research, one for one doing it, or indeed that DPI still is involved in doing this sort of auditing work of institutes and facilities?
So I can please to that, yeah. So the Australian Code provides that there's a requirement for independent external reviews, so the institution needs to undertake that activity. That's supplemented by these inspections that we do with ARP members of facilities. So it's not, it's provided for that you can conduct those independent external reviews where another institution, for example, reviews your performance, and that's really a key component of the self-regulatory model as well, because there's oversight by ARP and the DPI, but it's in addition to these independent external reviews that are conducted as well by the institution to make sure that they're, working within the requirements, and it's a critical component for them to keep under review their compliance with the code and the Act. So the department still undertakes inspections in conjunction with ARP. As Dr. Tracy said, they're focusing that based on risk and increasing the inspection frequency of that, because under the code there's a requirement that the institutions undertake this on a 4-yearly basis. And so having looked at where risk-based compliance has come from and also the requirements on government to ensure a risk-based approach is taken in relation to its compliance and enforcement efforts, we see that it's having worked with ARP, looked at what's the best way of making sure that we're focusing our efforts in the right place and at the higher risk or high impact areas of these institutions. So it's a tailored approach that we take, but it supplements, I guess, what's already been done and is allowed to be done, I guess, from that, the independent external reviews that are conducted by the institutions.
So the key to that is that you've actually got that additional layer. DPI also does inspections, and in addition to that, you've got the oversight of the Animal Research Review Panel. And at any time, so this sort of approach allows, yes, institutes to be under the requirement of some self-imposed review. But in addition to that, you've got DPI doing investigations as well as the panel overseeing that activity. So you've got essentially all elements of that make it a stronger system. And I think the key point about the risk-based approach here also allows a greater focus where we see higher risk or where the panel see higher risks in relation to that. So it actually is a much stronger way forward as we're progressing. It's getting stronger and stronger in relation to that.
Can I ask the next question, and perhaps various people might like to answer? The matter of the ethics committees, and we covered that a bit this afternoon, and it's been explained, I think, in some detail by yourselves or some of you, and also with other witnesses about the way in which the committees function and how they can function well in terms of their role. But I'm wondering, stepping back and just, if I didn't say, well, listen, how many ethics committees are there in the state of New South Wales operating? I don't know the number there, but let's call it X. So you've got X number of these committees. In terms of, I'll use this word advisedly, the standards and the quality of the work it does as an ethics committee, assuming that clearly people who are involved in ethics committees are individuals with expertise, knowledge, obviously desire and motivation to play that role on the committee. There surely must be, though, some variance in the output or the quality of the work done by an ethics committee, which leads me to this question about how can we be sure, and if we are sure, please say so, but that the ethics committees are functioning in the best way as they possibly humanly can, bearing in mind nothing is infallible. And if the answer is, well, there are issues, but this is what can be done to enhance the ability of them to function in the best possible way, this is what ought to be done. So it's an open question.
Sure, maybe I can start.
Please,
there.
yes.
So firstly, in terms of, we were talking about inspections, one of the things that we do when we undertake inspections is we actually virtually, as in cases we have occurred, or really attend an ethics committee. And we will sit and we will observe, see what happens, and then have a discussion with both the committee and perhaps also the chair afterwards on both their compliance with procedure associated with the code and also the function of the committee, how well they're interacting, the engagement of all the different committee members, and is the actual committee working effectively in the way that it's supposed to in the code. So that's one of the things we do. The other thing we do is every year, as I mentioned, we receive an annual report from each of the accredited institutions. This is a report that they have to submit internally and to us. And in that are questions about the function of the Ethics Committee, and also it gives the Ethics Committee the opportunity to indicate where they feel they have issues and compliance, or they have concerns with the institution. And so that's another avenue where the Ethics Committee can have a voice to indicate where they feel that they may need support. So that's two of the main ways that we engage with ethics committees, to have oversight of how they're functioning and also from the arts perspective to support them. So we will then go and support and provide advice and help with perhaps its templates to help them engage better with the researchers. There's a number of different ways that through the ARP we provide that support and oversight for ethics committees.
And when one comes onto an ethics committee or joins an ethics committee, is there understood training or educational programs that are undertaken to enable a person, whatever their particular professional right, be, or they might be a layperson, to carry that out well?
Yeah, look, absolutely. So we actually provide a guideline for induction of new members onto ethics committees. It's also part of the code that's an important thing of what ethics committees do. And the process there is something, again, we will support ethics committees in doing that. And so there's support around how the committee runs, what the work of the institution is, and really bringing those people up to speed in terms of their functions and their responsibilities as being members of that committee.
We heard from a witness, I think earlier today, or it might have been on another occasion, about people joining the committee who are very much drawn from the citizenry. They're people who have an interest and put their hand up, so to speak, to enter onto a committee. Are they advertised, those positions, when they come up? Just out of interest, because I don't know the answer to this. Are those vacancies, as they become available, something which are advertised and people can apply? How does that generally work?
Different institutions will do that differently. Perhaps, Roger, in terms of how your institution works to receive this particular, I suppose you're referring particularly to the Category C and Category D members.
Yes.
Yeah.
A variety of mechanisms. We have advertised, in newspapers, local newspapers, etc. In the pre-COVID era, the practicality of attending, meetings and giving aside what is a significant contribution of time for volunteers.
Yes.
We try and make sure that they understand what they're seeking to do. Interview them mainly to make sure they can take the time off from their employment if their employer is giving them time off or whatever. But I think the thing is to make it known that these vacancies are available, and there is a listing that the department also keeps, because people contact them, and periodically we'll contact them to know if there's someone who's put forward their name as wanting to do this sort of work, so that there is that avenue for appointment to committees. As was alluded to, the most difficult categories to fill are those who have a track record of working for animal welfare and organisations, because whilst they are not representing those organisations, the requirements are that they be endorsed by an animal welfare organisation to sit on the committee. So that step does, I think, mean that they have credibility as members in a Category C. So very many of them have already done committee work in some role in other settings, sometimes have sat on other ethics committees. But the induction is important. In the last week, I spent half a day with our newest member of our committee, giving them an opportunity to see one of our animal facilities firsthand, so that they're not in a situation where they might be making decisions about housing or other things of something that they weren't physically aware of in terms of what resources were on the ground. So I think that is actually an important part of these committees functioning well, is that they, the people on them do their homework, do their inspections, and have the opportunity when they're inspecting facilities to interact with researchers and to ask questions that they want answers to.
Next question, I'm just going open to across the board. Evidence about the involvement of vets. There's been some examples where, I can't think of the precise case itself, whereby on the ethics committee there was, a few vets, or 2 or 3 vets. There's been counter evidence that from some witnesses that there's not enough vets, or there is a dearth of involvement of vets interfacing with the committees or being on the committees to sort of play that role that a vet would be capable of in terms of input. And we actually had the Australian Veterinary Association giving some evidence early today, expressing a desire and interest to try and encourage the other stakeholders, because we use that phrase in the area of animal experimentation, to give consideration of engaging more with the Australian Veterinary Association to have more involvement of vets. I'm just wondering, is there a sense that any of you can give that the involvement of vets in this area of animal research has been something which has been declining over time, or stable, or any general observations? Because certainly the Australian Veterinary Association were putting their hand up and saying we're keen to see how more can be involved. And as I said, there's been some evidence where some committees seem to be well represented with vets on them, and other ones perhaps that may be a bit light on. So I'd be interested in your observations.
Well, I guess I could probably answer some of that question. The animal ethics membership is determined, and there's a set formula, I guess, if that's...
Yep.
So there's a category A, B, C and D member as a minimum. So you must have one of each of those.
Yes.
A category A member is a veterinarian.
Okay, so there's got to be at least one.
So each animal ethics committee must have one veterinarian. There's also other rules around the category C and D members must also make up 1/3 of the membership. So you can have more than 4 members, but you can't have 5 vets and 5 A's, 1 B, 1 C, 1 D.
Yes.
So that gives an unequal sort of balance.
That's helpful, even though that, yeah.
Yeah, so there was reference to some committees have 2 members of 2 A's, 2 B's, 2 C's, 2 D's, but whatever the formula ends up as, you can't have any more than, you must have 1/3, C and D, so that you don't get an over-representation of vets or researchers. So the category B members are animal researchers.
Can I just add that the change in personnel of those committees has to be endorsed by ARP. So there's a second endorsement of that ratio and the CV of the proposed addition, and it would not be uncommon at ARP for us to vet out and vet in applicants for those committees.
Okay, okay, that's helpful. Well, listen, I'm sure we could go on, but listen, you've been very generous with your time. I know you're all very busy roles, and we do appreciate making yourself available this afternoon. So thank you very much for it on behalf of the committee. We'll study the Hansard after it's produced, and I'm sure there'll be some supplementary questions, and if you're agreeable, through the Secretary, we'll liaise with you over the questions and return those questions. So once again, thank you very much for making yourself available this afternoon.